live now? Are we live?
Yeah, we are going live.
Yeah, thank you. Very good evening to everyone connected to us in India, and very good morning to our attendees from abroad. I welcome you all to the second day of the two-day virtual Centenary Conference, organized to celebrate the establishment and success of the Department of Biochemistry ISC. We have amazing talks scheduled today too, and the organization of the talks and Q&A sessions will be followed similarly as yesterday. I would like to request my colleague Kirtana to coordinate the third session of the conference. Over to Kirtana.
Thank you, Shistaza. Once again, I'd like to welcome you all to the virtual centenary celebrations of the Department of Biochemistry. Before we begin the first session of today, I'd like to request all our speakers to kindly adhere to the timing. Apart from the centenary lecture, each talk will be of 25 minutes, followed by a 5-minute question and answer session. Attendees may verbally ask their questions or post their questions in the Zoom chat box or the YouTube comments section. The session chairs will coordinate the question and answer session by calling on the attendees to verbally ask their questions or by reading out the questions themselves. I also request all our speakers to disable attendee annotation from the More Options tab on the top right of your screen after screen sharing mode. Now I'd like to welcome Professor H.S. Savitri, who is chairing the first session of today. Professor Savitri is a distinguished alumnus of our department and was the first woman faculty and the chair of our department. She is well known for her studies on plant viruses. Currently, she is an NASI senior scientist in our department. We warmly welcome you, ma'am, and we invite you to start the session.
A very warm welcome to all of you for the session 3 of this virtual centenary conference of the department. At the very outset, I would like to thank Professor D.N. Rao and Rangarajan for inviting me to chair this session. It's indeed an honor for me to be part of this celebrations of this temple of learning. The greatest strength of our department has been the training, the excellent training it gives to all its research scholars. And all those students, almost all the students who have gone out of this department, have passed out of this department, have invariably come back and told that they owe their entire success in their careers to the fundamental foundation of research that they received in this department. And it is indeed true in my case. And this, I'm sure, all of us, all the alumni, will cherish the time they spent in this department. There are 4 speakers in this session. I know all of them personally, except Professor Mohan Babu. I'm looking forward to meet him now virtually and to listen to his talk. The first speaker of the session is Professor Sandhya Vishweshraya. She is my friend for over 35 years. She obtained her PhD in 1987 with Professor Adiga. And unlike most of us, she moved in 1987, she moved to Astro Research Center for her postdoctoral fellowship. And in 1993, she joined the department, the newly formed department, Molecular Reproduction and Development and Genetics, as a faculty, and is currently a professor in the department. Her focus of research in the laboratory relates to signal transduction mediated by cyclic nucleotides and investigation of the molecular mechanisms of signaling by receptor guanyl cyclases, phosphodiesterases, and novel nucleotide cyclases in bacteria. So I'm sure you're all looking forward to the talk from her. So I hand over the talk to her. I request her to present her talk.
Thank you very much, Savitri, for your very kind introduction.
And at the beginning, I'd like to extend a really warm thank you to Professor D.N. Rao for having sent me this invitation. I consider it a great privilege to be able to speak at this meeting. It seems like yesterday when I interviewed in the Department of Biochemistry, and I know that I was absolutely flabbergasted that I was selected into the department for my PhD. I think, as Savitri mentioned, we all benefited greatly from the discipline that the Department of Biochemistry instilled in us, as well as the wonderful teachers that we had, which include, of course, Professor Padmanabhan, Professor M.R.S. Rao, and of course, my own supervisor, the late Professor P.R. Radiga. I hope as I go on in this talk, you will realize how closely linked I remain to biochemistry, because I honestly believe that a greater understanding of biology comes if we can apply biochemical approaches to answering the questions. Now, the title of my talk is Gut Instincts and Gut Reactions, and I'll be talking about the role of cyclic GMP in mediating fundamental aspects of gut physiology. Now, it was Hippocrates who said many years ago that all disease begins in the gut. I've tried to identify some kind of Ayurvedic or Unani statement along these lines. And so if any of you more knowledgeable than I come across one, please do let me know, because I think I'd like to say that rather than this. In this day and age, we all know that as far as the gut is concerned, it's important for digestion and fluid and ion secretion in order to be able to keep the bolus of food moving down the intestine. One of the things that's quite amazing is that the gut is a rapidly proliferating tissue. And it would surprise us to know that we renew the entire intestinal epithelium once in 4 days. What this implies is that rapidity of proliferation and regulation of proliferation must be very tightly controlled in the gut. And I'll touch upon that a little later. Nowadays, everything and anything is attributed to the microbiome, especially the gut microbiome. We're well aware about how important the bugs in our gut communicate with the host and elicit very profound reactions, even in terms of influencing brain function. But what I'm going to focus upon is disease of the gut, and that is basically diarrheal disease. Now, if you think about diarrheal disease, almost 1 in 10 child deaths around the world result from diarrheal disease, especially during the early years of life. There are almost 1 million fatalities worldwide annually, and most of them, of course, do occur in sub-Saharan Africa and countries including the Indian subcontinent. These kinds of numbers puts in some perspective the reaction that the world has had to something like COVID, and perhaps that kind of attention, if it could be paid to some of these diseases, may bring about profound changes in the lives of many individuals across the world. There are 5 pathogens that have been majorly associated with diarrheal disease. These include rotavirus, Shigella, certain strains of enterotoxigenic E. coli, and other organisms. I'll be focusing on the enterotoxigenic E. coli. Now, these strains of E. coli colonize the gut and harbor plasmids which encode toxins, which are called the heat-stable enterotoxins. Now, shown here are the peptide sequences of the mature toxins produced by various strains of bacteria. And what is characteristic about these peptides, as you can see, is the presence of 6 cysteine residues, which when I show you here are all disulfide bonded. This is what contributes to the heat stability. In other words, heating these peptides at 95 degrees for prolonged periods of time does not inactivate them. Of course, if you add a reducing agent, you will inactivate these toxins. Very early on, almost more than 40 years ago, it was shown that the action, the second messenger that mediates the actions of these toxins is cyclic GMP. Obviously, this toxin and its receptor was not present in the gut purely to be responsive to bacterial toxins. But it took almost 25 years before endogenous gastrointestinal peptides called guanylin and uraguanylin were identified in the gut. You can see that these peptides have 4 cysteine residues as opposed to the 6 found in bacterial toxins. Interestingly, all of these peptides are produced as pre-pro proteins. Very unusual in the case of bacteria to have this kind of pre-pro toxin produced. Of course, it's quite common in mammalian systems. We know a lot about these toxins. We have crystal structures of these toxins. We have NMR structures of these toxins. And just as an aside, I'd like to mention that recent FDA-approved drugs called linaclotide and plecanatide are used for the treatment of constipation. And these peptides represent nothing but the core sequence of the bacterial enterotoxin. These toxins, because they induce diarrhea and fluid secretion, are also efficacious in relieving symptoms of constipation. Now, what about the receptor for these peptide toxins? It took, again, almost 20 years after the identity of these toxins to characterize the receptor. And unlike the peptide toxins, this is about the highest resolution structure we have of this receptor. Absolutely no domain in this particular receptor has been crystallized, or do we have any kind of structural information on. As you'd expect, the receptor has an extracellular ligand binding domain, which interacts with the toxins as well as uroguanylin and guanylin peptides. It's a single transmembrane spanning domain protein, and the intracellular domain is composed of two regions. One, a region which has some similarity to protein kinases and is called the protein kinase homology domain. This is linked to the guanyl cyclase domain, which is a member of the class 3 family of nucleotide cyclases. And so there's a lot of resemblance between the guanyl cyclase domain and adenyl cyclases, which make cyclic AMP. Now, when I first joined the Institute, we were fascinated about this kind of protein where you had profound allostery happening when the ligand binds to the extracellular domain and transmits its signal to the guanyl cyclase domain to increase production of cyclic GMP. Therefore, for many years, we spent our time making mutations along the length of this receptor to try to understand how it functions and what are the critical determinants of ligand binding and allosteric control. The extracellular domain is heavily glycosylated, and we characterize the glycosylation of this receptor by making individual, combinatorial, and all-inclusive mutations of glycosylation sites. We were able to show that glycosylation was not a prerequisite for ligand binding, but was essential for mediating the conformational change that resulted in cyclic GMP production. Now, the kinase homology domain was also a focus of our studies for quite some time. This region has similarity in structure to classical protein kinases, and therefore, working along with Professor N. Srinivasan in molecular biophysics, we were able to model this domain, identify residues that were likely to bind ATP, and mutated those residues to understand the role of this domain in mediating the allosteric conformational change. This region is extremely important because any mutations brought about in this domain, especially those that regulate ATP binding, completely abolish ligand-mediated cyclic GMP production. Now, another region of the receptor that caught our attention was this little region which we call the linker region. This is found between the kinase homology domain and the guanyl cyclase domain and is predicted to have an alpha helical structure. We therefore systematically mutated every residue in this linker region to a proline residue and monitored what would happen to the receptor activity with these single point mutations. And what you can see here is that some of these single mutations have exceptionally profound effects. Mutations in this region of the receptor, as you see, completely elevated basal activity of the receptor so that you had very high cyclic GMP production, even in the absence of ligand. Some of these mutations no longer increased ligand-mediated cyclic GMP production. Please note that the y-axis is a logarithmic scale. So some of these mutations elicited a cyclic GMP response even higher than the wild-type response. Now, I'd like you to remember this as I go on in my talk. This will come back in terms of its importance. Now, where is GCC expressed in the cells of the intestine? We mined the information coming out of single-cell RNA sequence analysis of the intestinal epithelium, published in Nature a couple of years ago. Now, the intestinal epithelium is very complex. It has a variety of different types of cells, each with their own functions. And from single-cell RNA-seq analysis, we could see that GCC was expressed in almost every cell type of the intestine. This included stem cells and their progenitors. Now, the intestine at its base, which is called the crypt, harbors very few stem cells, which are the ones that continuously proliferate. As they proliferate, they move up the villus of the intestine and undergo a process of differentiation, resulting in the formation of goblet cells, enterocytes, enteroendocrine cells, etc. So GCC is also expressed in the stem cells and in the Paneth cells, which are the cells which produce antimicrobial peptides during infection. Now, what about the signaling mechanisms mediated by this receptor? Ligand binding to this receptor elicits cyclic GMP within the cell. Cyclic GMP now activates a cyclic GMP dependent protein kinase, or PKG2, which now phosphorylates various molecules which you would have heard of. One is cystic fibrosis transmembrane conductance regulator, or CFTR. Phosphorylation of CFTR results in increased chloride efflux from the intestinal cell. PKG2 also phosphorylates the sodium hydrogen exchanger and inhibits it. Therefore, there is an accumulation of sodium in the lumen of the intestine. It is this increase in sodium concentration in the lumen and chloride secretion out of the intestinal epithelial cell that generates an osmolarity gradient by which water now effluxes from the intestinal cell. And this is the characteristic diarrhea that is associated following activation of GCC by peptides produced by enterotoxigenic E. coli. It is believed that the endogenous hormones, guanylin and uroguanylin, elicit just the right amount of cyclic GMP to generate enough fluid in the intestinal lumen to allow free passage of the food. Therefore, these stable toxins can be considered superagonists of the receptor. Now, we and others have identified a number of other signaling outputs from this receptor, including its crosstalk with CSARC tyrosine kinase and calcium levels, but I'll not have time to go into that in this talk. Now, what about GCC knockout mice? When these mice were generated more than 20 years ago, they're fertile, live a normal lifespan, and they're resistant to ST-induced intestinal diarrhea, implying that as far as ST peptides are concerned, GCC is the sole receptor. Later on, people showed that these mice had attention deficit disorders because GCC is also expressed in the brain. These mice also show abnormalities in feeding behavior and generally show signs of obesity. They also show intestinal barrier dysfunction and are more susceptible to colitis-inducing agents. We have GCC knockout mice in our colony, and apparently we do not see either of these two phenotypes. We do not see any increase in obesity in these animals, nor do they appear to have any behavioral defects. These studies, one was published from China and one was published from America, so maybe the Indian environment is more conducive to these mice. The fact that GCC knockout mice had no major phenotype led to the thought by the late David Garbers, who was actually the father of receptor guanylyl cyclases, to suggest that GCC likely provides a protective effect against stressors that are not yet tested. And this is what we and others set out to do. And I'll just summarize very briefly some of our findings. Working along with Professor Deepankar Nandi here in the Department of Biochemistry, we could show that GCC knockout mice were more susceptible to infection with Salmonella typhimurium. GCC activity also was required to prevent the formation of aberrant crypt foci in the presence of carcinogens. This was work from a former student in my laboratory. In fact, GCC agonists now are believed to be able to serve as chemopreventive strategies for colorectal cancer in humans. And clinical trials, I'm sure, are ongoing in the West. Interestingly, activation of GCC has also been shown to reduce visceral pain. And so there are a number of pleiotropic effects of this particular receptor that go beyond purely its diarrhegenic role that it seemed to be emerging in the recent past. Now I'll focus on what has caught our attention in terms of translation over the last 10 years or so. And that is the role of GCC in human disease. As I mentioned, it was clear that this receptor was present in almost all eukaryotes, right from fishes onwards. What was it doing and what made it so critical? And why were there no reports of any mutations in this receptor associated with human disease? Around a decade ago, based on the work that we've been publishing related to mutations in this receptor, I was approached by two clinical geneticists from the University of Bergen in Norway. They told me of a family that they had identified, comprising 32 members, where for many generations, all of these individuals, or many of these individuals, reported childhood constitutive diarrhea. You can see, based on this particular genetic profile, that this was an autosomal dominant mutation that they were dealing with. It was not sex-linked. What were the phenotypes of these patients? They passed multiple stools per day. In some cases, some individuals passed as many as 20 stools per day. Stools were of loose consistency, and there was severe abdominal pain in many of these patients. The disease was so severe that many of them had to undergo surgery, which included ileal inflammation or corrections to ileal inflammation, twisting of the gut, adhesions, etc. And there were also anatomical variants found in the ileal-cecal junction. And many of these patients, before we reported our work, were found, or at least to have symptoms of Crohn's disease, and were treated for Crohn's disease. When these geneticists mapped where the mutation lay, they found that it lay on chromosome 12, and the particular region that the mutation lay harbored a number of genes. And when they inspected the genes at this locus, they found the GCC gene. This is when they wrote to me, after sequencing the entire gene and identifying a single point mutation, which was found only in patients, in members of the family that were affected, in a heterozygous state, and this mutation converted a serine in the guanylyl cyclase domain to an isoleucine residue. They asked if we would be interested in characterizing this mutation, which of course we did. And what we found was, by introduction of this isoleucine residue at this position in the receptor was hyperactive. Hyperactive to the extent that ligand-mediated cyclic GMP was markedly increased. Please note that there was no change in basal activity of the receptor, and that is receptor in the absence of ligand. All of these experiments were performed in heterologous expressed cell lines. Interestingly, the mutant receptor also had a leftward shift in terms of its EC50, suggesting that it was going to be a little bit more sensitive to the endogenous ligands guanylin and neuroguanylin. Presumably, what was causing diarrhea in the Norwegian population was not the bacterial enterotoxin, but a response to the endogenous ligands in the gut. Now, this was the first report of a mutation in GCC associated with human disease, and therefore it was published in a reasonably good journal and caught a lot of people's attention to look at their patients who had unexplained incidences of childhood diarrhea. A couple of years after this paper was published, I again received communication, this time from clinicians from a pediatric hospital in the University of Innsbruck in Austria. They had characterized 4 young children, unrelated, across Europe, who presented with extremely severe congenital secretory diarrhea. This is a picture of the child that has just been born, and you can see that her abdomen is completely swollen and filled with fluid. And immediately after delivery, this fluid has to be drained in order for the child to be able to breathe, because there was so much pressure put on the lungs. 3 other children also had less severe symptoms. And when the clinicians mapped the mutations, they found 4 additional mutations that lay along the GCC receptor. As you can see, 1 receptor lay in the kinase homology domain, 1 in the linker region, and a couple more in the guanylyl cyclase domain. This is the mutation of this child. And what we saw here is an exceptionally high basal activity. In other words, this receptor was producing elevated levels of cyclic GMP even in the absence of ligand. And this was further stimulated in the presence of ligand. What was most intriguing is when they reported to us that they found a mutation in this position at 11775. And this mutation was a leucine to a proline. This was the exact same mutation that we had made almost a decade ago when we were studying the linker region. And therefore we knew what this mutation would do. It led to elevated basal activity. We characterized a number of these other mutations, and you can see that these were also hyperactivating in comparison to the wild type receptor. So therefore, these hyperactivating mutations of GCC lead to congenital secretory diarrhea. A couple of years later, again, a father wrote to us from Australia, informing us about his child. And he said he was very happy if we spoke about this work in any of our talks. This child also presented with these kinds of severe symptoms, and the mutation again lay in the linker region. And again, we had made exactly the same mutation 1 decade earlier. What this goes to show is that elevated levels of cyclic GMP cause severe gastrointestinal pathologies. And this intrigued us a great deal. And therefore, we thought that we should now try to generate preclinical models where we would be able to study more closely the roles of hyperactive GCC in the gut. In order to do this, we decided to create a transgenic mouse, mimicking the Norwegian mutation that had been identified, that we had characterized earlier. We ensured that bringing in this mutation in mouse GCC had similar effects as the human GCC. The position in the mouse is S839I because there is one residue that is missing in the linker in mouse GCC. And you can see that bringing in this mutation in the mouse receptor also resulted in increased cyclic GMP production in response to ligand. Moreover, we also saw a leftward shift in the sensitivity of this receptor to the ligand. We generated these transgenic mice with Taconic in Denmark, and the funding was obtained from the Norwegian Research Council, as well as from the Department of Biotechnology. Now, what about the phenotypes of these mice? How closely do they mimic the phenotypes that were seen in human patients? The first thing we measured was intestinal cyclic GMP levels by preparing intestinal epithelial cells from both the ileum and the colon of these mice. And indeed, as one would expect, intracellular levels of cyclic GMP were higher in the epithelial cells in the gut of these mice. Now, mice don't get diarrhea the way humans get diarrhea, and that is presumably an evolutionary consequence of the kind of diet that they feed on. However, there are a couple of things that you can do with these animals. You can put these animals in new cages and monitor the number of fecal pellets that they pass in 10 minutes. And in our transgenic mice, you can see that the fecal pellet number was significantly increased, somewhat similar to the kind of repeated episodes of diarrhea that the patients experience. You can collect fecal pellets and you can lyophilize them and calculate the percentage water content in the feces of these mice. And here again, you could see that the fecal water content in these mice was also increased, again, resembling the watery stool seen in patients. Finally, as I mentioned to you when I spoke to you of the signaling, there would be an accumulation of sodium in the lumen of the gut. And this sodium would now move its way down the gut to be found in the feces. And indeed, the children with congenital secretory diarrhea show elevated sodium levels in their feces. Our mice as well, the transgenic mice as well, showed a significant higher level of sodium in the feces, again mimicking the human condition. Now, we went on to explore a few more parameters in these mice. The first thing we looked at was the fecal microbiome. You can see a clear separation in the species that are identifiable in the wild type and S8 mice. We analyzed this far more closely and actually identified that a number of changes that are observed in these mice mimic those changes that are seen in patients with familial guanine cyclase diarrheal syndrome, as well as in patients affected by Crohn's and IBD. And so this again emphasizes the close link between the microbiome and gastrointestinal disorders such as Crohn's and IBD. We then went on to try to understand a little bit more about the molecular changes that happen in these mice. And therefore, we analyzed by RNA-seq the transcriptome in the colon of these mice. This is the analysis that we have. A very large number of genes were misregulated, most of them downregulated. And using ingenuity pathway analysis, we were able to identify that one of the significant signatures that came out was an upregulation of interferon-stimulated genes, or ISGs. Now, what I'm showing you here is the interferon regulated pathway. You can see that interferons bind to their receptor, activate STATs, which now move into the nucleus and induce the expression of a number of genes. The numbers that you see here in red are the fold changes seen in our transgenic mice. And most of these genes were validated, I'm not showing you the data, in independent mice in our colony. Therefore, what we see in our mice is an activation of the interferon signaling pathway, which could account for the increased inflammation that is reported in patients. We looked at this inflammatory signature a little more closely and compared the genes regulated in our mice with the genes that have been shown to be regulated in a mouse model of colitis, that is where you feed mice with dextrams sodium sulfate. You can see that a significant number of genes are similarly regulated in the basal state in transgenic mice, as is seen in DSS-treated mice. What Ingenuity allows you to do is to predict what it calls upstream regulators. These are usually transcription factors that actually regulate the pattern of gene expression that you see in these mice. And you can see here that, again, we see a good correlation between upstream regulators in our mice and upstream regulators that are upregulated following DSS treatment. And finally, in ulcerative colitis patients, when we made this comparison of gene expression that we've observed, we see that here again, a number of genes that are similarly, that a number of upstream regulators that are activated or inhibited in colitis patients are similarly regulated in, or are doing similar regulatory aspects in our transgenic mice. So what have we found in summary so far? Several monogenic intestinal epithelial defects contributing to pediatric inflammatory bowel disease have been described, and mutations in GCC are responsible for some of them. These diseases are infrequent, so they could be considered rare diseases and are not readily amenable to current treatment regimens. We've shown you that mutations in GCC predispose to pediatric IBD. And a very recent paper that was published from the Children's Hospital in Toronto, where they analyzed almost 1,000 cases of pediatric disease and looked for mutations by whole exome sequencing analysis, found that almost 6% of patients harbored mutations in GCC. And when we looked at some of these mutations, we find that in fact, they have not been characterized earlier before. So this looks to be a challenge that we'll come back to doing the biochemistry that I so love doing. Our transgenic model that we have generated, and we have two others that we are actually characterizing in the laboratory. These are again mutations in the linker region in one case, and another humanized mouse where we've actually introduced the human R792S mutation in mice. These are both under characterization in the laboratory. And we believe that these mouse models can be used as preclinical models to test for drugs, etc., and therapies. What I've shown you are perhaps more fundamental findings on the role of cyclic GMP in regulating gut physiology and biology. And we've now deciphered, or at least think we're close to being able to decipher a molecular link between cyclic GMP and inflammation in the gut. Now, just before I leave you, recently we've surveyed all the mutations that have been identified in the receptor and which cause human disease. As you see, many of these mutations are located in different domains of the receptor. There are different types of variants. They could be stop codons, which lead to truncation mutations. There could be missense. Many of these mutations, we really do not know what effect they may have on receptor activity. Opening up the possibility that mutations in GCC and consequences in terms of gut physiology could be far broader than what has been appreciated to date. Now, with the recent release of the sequence of around 1,000 odd Indian genomes, we of course immediately mined this for any mutations that have been found in our population in GCC. And you'll see that a number of heterozygous mutations have been identified. Some of them don't have any RSIDs right now, which means that they are unique and novel mutations. And you can see that some of these mutations are predicted to be deleterious. And so therefore, this opens up the possibility of looking into these mutants in terms of more subtle phenotypes that they may show in individuals that harbor them. I'd like to close by acknowledging the amazing group of people who've worked with me on this over the years, and without whose efforts, none of this would have been possible. These are the students or former students, all underlined, who have worked on GCC over many years. People underlined are those who have left the laboratory. Many of them have obtained their PhD. Dr. Anakshi Barman was the vet who did a lot of the work related to our mouse model. In blue here are current students, and the ones in black are those currently in the lab. Shashi is the one who did all the microbiome analysis, and Sangeeta and Avipsa are those who have done the RNA-seq analysis in the transgenic mouse model. As far as funding is concerned, I of course acknowledge the Government of India funding that I received for the better part of my career, and then have been extremely fortunate to have had international funding. The Bill and Melinda Gates Foundation funded us to generate a new mouse model, which is being characterized in the lab. I've had funding from the Royal Society. It's a collaborative grant for research professors where I work along with Professor Gad Frankel at Imperial College in London. The Research Council of Norway has funded us along to generate a vaccine for the stable toxin peptide. I've not spoken about that at all. And for that, I'm grateful to Dr. Halvor Sommerfeld in the University of Bergen, who's provided and been a motivator for this kind of research. Currently, I have a CEFIPRA grant with Dr. Laurence Moulade at the Institut Pasteur in Paris, where we're trying to generate peptides which are fluorescently labeled to be able to do in vivo imaging of GCC receptor in whole animals. And of course, last but not least, I thank the Wellcome Trust EBT India Alliance for providing me a Margdarshi Fellowship for the last 5 years in work not related to what I've spoken to you about, but which hopes to interface very closely with researchers in the Institute, a clinical association, largely through biosystem science and engineering. Thank you very much for your attention, and I'd be happy to take questions.
Thank you, Sandhya, for the lucid presentation from signal transduction to human genetics. You have gone through a long way. Thank you very much. Because we have run out of time, I wonder if we can take 1 or 2 questions, but I don't see many people, anybody
maybe
shaking.
Maybe I can start, ma'am. Sandhya, that was fantastic as usual. I had a quick question about your link to inflammation. Is this a cell autonomous link to inflammation, or do you need non-intestinal cells to induce inflammation? So if you were to activate GCC cell autonomously, would you induce inflammation just in that cell without help of any other cell?
Yes, through the innate immune system, through STAT induction. I haven't shown you this data, but it appears by just applying cyclic GMP to murine small intestinal cells, we're able to activate STAT. And so therefore, there seems to be a hidden signaling that's going on here, which we're going to try to elucidate in the years to come. However, the more pronounced inflammation that you see in the gut must necessitate the immune repertoire. For example, you have intestinal associated lymphocytes, ILC3s, which produce IL-22, which now feeds back to regulate intestinal epithelial proliferation. And I think we hypothesize that this whole signaling machinery is somehow regulating the population of intestinal lymphocytes in the gut, which could also be contributing to the enhanced inflammation that we see.
Sandhya, okay, thank you.
Ullas?
Yes, hi. Sandhya again, a nice talk. I'm just going back to the non-mutation case, right? Because that's what really interests me. So have you looked at the homeostasis of the receptor and whether you have unliganded internalized receptors being active or inactive? Because I'm just curious, because, you know, as much as, diarrhea is a problem, constipation is also a problem. I'm just wondering, have you looked at the cell biology of this receptor beyond biology?
Yes, and I just didn't have time to speak about it, but one of the features of the human receptor is that a significant fraction is retained within the ER. Now, we really don't understand why. When we've looked at the ER fraction, it binds ligand, but it's poorly stimulated by ligand. So you need that extra little bit of glycosylation that happens during transit through the Golgi. What that ER localized fraction of the receptor is doing, we don't know, but it could be maintaining a very low level of cyclic GMP in the cell, which could have other roles which one would like to be able to think about and ask questions about later. But yes, that's a very good question. Yes.
Okay. And lastly, there should be a nutritional impact on this, given that you have so much fluid. I'm just surprised. Probably you should go back and challenge some of these mice to see whether that actually can do something.
Again, not had a chance to talk about it, but when you open up our transgenic mice, we see a lot of abdominal fat deposit. So it seems as if there is some sort of misregulation at the lipid level, which again is something that we're going to hopefully be able to look at. We're going to be trying to challenge these mice, obviously, with high-fat diets, maybe low-fat diets. We should definitely alter the nutritional status, because something perhaps I didn't mention is that the Norwegian patients would periodically report severity of disease, which was linked to what they felt, something that they ate. So it could have been fruit, it could have been nuts, it could have been anything. But they repeatedly said that whenever they had that food item, their diarrhea became more severe. Yeah. So it's a holistic action that's happening here. Everything isn't just being done by GCC, but other stuff that must be going on, and including the microbiome. And I think that's likely to play a very important role.
Okay, thanks, Sandhya.
We are already running short of time. Maybe I will allow one question, last one from Mitali Singh.
Okay. Hello, Sandhya. Very nice talk. I have a very small curiosity-driven question. Is there any known ligand by which or method by which you can reverse this overactivation of the receptor?
No, I think, again, I didn't emphasize it, but there are no known antagonists for this receptor at all. All that we can do with this receptor in vitro is biochemically perturb it. So, for example, zinc is a good inhibitor in guanylyl cyclase assays. And zinc therapy, interestingly, is actually used for the treatment of diarrhea in young children, especially in countries like ours. But one of the efforts that I think would be definitely worth pursuing in future is trying to develop antagonists for this receptor. Precisely. These could bind to the extracellular domain. They could bind to the kinase homology domain, because as you know, kinases have been very druggable targets over many years. Mm-hmm. So I think there's a whole scope for identifying regulators of this receptor. But as of now, none are available, except agonists, except linaclotide, plecanatide, but they're all like the agonists of this receptor, and they won't help in this particular case.
Yeah, I agree. Thank you.
Thank you.
Thank you, Sandhya, for that excellent presentation once again. It was a pleasure listening to you. And now I will move on to the next speaker in this session. That is Dr. Professor R.P. Roy. He is obtained his PhD with Professor Vidyathil in 1989. I have a special, both Roy and my student Surya started on protein sequencing together, and I know how hard and they worked and what are the hurdles they had to cross before they could get their sequences. So I know Roy from the time he started his PhD, and from here in the department, he moved to Albert Einstein College of Medicine for his postdoctoral fellowship. And then he is currently at NII Delhi, and his current research interests are that he is engaged in the research at the interface of chemistry and biology, with interest in developing chemoenzymatic strategies for semi-synthesis of proteins and bioconjugates by combining enzymatic peptide ligation and chemical peptide synthesis, organic synthesis, and recombinant protein expression. So these are his research interests. I am sure we will have a wonderful talk from him. So I don't want to take more time. So I request Roy to begin his talk.
Thank you, Professor Savitri. And yes, I remember so many times we discussed things from you, and I got a lot of help on sequencing the xylanase. And in fact, at that time, these two proteins, one that you solved and the or sequenced, and the other in our lab, one was a protein, the other was an enzyme, and these were the first sequences from India. And of course, you were the first, and I was the second. With this, I would like to thank the organizers, Professor Rangarajan and Professor D.N. Rao, for inviting me. And it's always a pleasure to be associated with the biochemistry department and meet old friends. And it is actually a privilege to be associated with the Indian Institute of Science and the biochemistry department. Now, let me share the screen first. Okay, can you see the screen? Okay, fine. So, actually I have spent about 27 or more years at the National Institute of Immunology, and now moved to a new institute a month back, which is the Regional Center for Biotechnology in the NCR Biocluster. And the work that I'm going to present is done at the National Institute of Immunology, mainly for these many years. And the title actually is The Art of Breaking and Making Peptide Bonds. But the original title that you must have seen, the peptide word is missing, and I did not notice this unless a friend told me that, are you going to talk about social bonds? No, these are not social bonds. These are actually peptide bonds. The breaking of the peptide bonds, I learned in my PhD in Professor Vidyathil and Professor Krishnamurthy's lab, wherein, as Savitri said, I was going to sequence the endoxylanase from a fungus called Thermoeschus aurantiacus. And in the process, what you need to do is to first break the peptide bonds of the protein with proteases, and then reassemble the sequence. Actually, this protein sequence required a lot of purification protocols. And yesterday, if you remember, Utpal mentioned about the work of Professor K.B. Gire, who discovered circular paper chromatography. And in fact, Professor Gire developed many methods of separation of peptides those days in early 1950s. And he published at least 5 papers in Nature dealing with the paper chromatography and agar electrophoresis. Now, if you remember those days and compare that with the present times, if somebody today gets 5 papers in Nature, I don't know, we can't imagine how he would look like, and you can't just imagine the kind of arrogance and status that person would assume. But maybe those days the Nature was a journal in black and white and not as colored and hyped as it is today. So first 2 years, I was actually mastering the art of separating peptides by paper electrophoresis and paper chromatography. So first dimension, paper chromatography, and in the second dimension, high voltage electrophoresis on paper. So this was in 1984, and it continued till 1986. And in fact, the sequencing that we used to do was identifying, going through the routine process and identifying the amino acids on TLC. And in fact, I was lucky that in 1986, we got the first HPLC in the department. No, not the first, the second one. The first one was with Professor Mahadevan's lab. And they used to use that HPLC specifically for quantifying the hormones, the plant hormones. And it was his personal, nobody has access to that. The department got one in 1986. And that's where we started using HPLC for purification of peptides. In fact, Surya Narayana in Professor Savitri's lab did the entire sequencing using TLC. TLC and paper chromatography were the most popular analytical methods of those days. Anyway, so moving from here, one important point that I would like to make before I move from here is that after I sequenced the xylanase, Professor Ram Kumar of the physics department in IISc solved the structure, and most importantly, he also, from the electron density maps, corrected some of the errors that I had made in my sequence. And the collaboration that started then has continued, and a couple of years before he retired, we solved two structures on an enzyme called sortase, which I'll just talk about. So now this was about breaking the peptide bonds, but from there I moved on to making the peptide bonds. And the question is, can we make peptide bonds using proteases? Now in early 1950s, the idea that proteases may be important and may be implicated or may be participating in the protein biosynthesis based on the principle of microscopic reversibility of reactions. However, in 1953, Henry Borsuk wrote a treatise in JBC, a 42 pages treatise, where he said, the history of experimental attack on the problem of enzymatic peptide synthesis of proteins is one of the periodic outbursts of activity stimulated by new methods and ideas. Then there is a quiet period when the leads appeared exhausted. We are in the active phase now. So he sincerely believed that the proteases, in fact, would be participating in the biosynthesis of protein. But then the central dogma came very soon and the whole idea got demolished. But some interesting, bizarre examples kept cropping up. And one of them is the biosynthesis of concanavalin A. Now this concanavalin A, generally referred to as con A, is synthesized as a pre-pro-pro protein processed to get a pro protein in which some blue portions that you see here in the middle is excised out by, of course, proteolytic action. Subsequently, from pro-pro, you get a pro sequence in which there is further trimming and the blue portion goes out. And then in the end, by a bizarre sequence of events, you see what you get is that the C-terminal end portion, which was originally made as pre-pro-pro, becomes the N-terminal portion becomes the C-terminal, and the C-terminal becomes the N-terminus. And in fact, this was shown that this joining eventually is catalyzed by an endoproteinase sparaginase. So proteases do bizarre things. Now, the second example was noted in 2004 when in immune recognition of a cancer antigen, it was found that the MHC class 1 peptide generated from this antigen composed of 9 residues in which the 5 residues came from the N terminus and the 4 residues from the C terminus. So apparently there was some event that joined these fragments. They were non-contiguous fragments, and the thinking at that time was that there is processing into large NNC fragments, then that goes into the proteosomes and further cleaved and then presented to MHC. So the joining actually happens before it enters the proteosomes. But it turns out that nature has been doing this for many, many years, and a large number of class 1 ligands that are generated in the proteasome are actually non-contiguous peptides. And in fact, 30% of all ligands presented to MHC1 is constituted by non-contiguous peptides of this kind, coming from splicing of different regions of proteins. And this actually is done in the proteasome itself by a transpeptidation reaction catalyzed by the proteasome. So proteasome indeed is also a peptide ligase. Now, today I'll talk about transpeptidase, which is called sortase, and give you examples of how this enzyme can be used to generate bioconjugates and engineer proteins. Now, this sortase is a surface protein found in all gram-positive organisms, first detected by Sneewind in 1999. This is a membrane-anchored protein. What it does is it recognizes LPX-TG containing surface proteins on the bacterial surface, cleaves at the TG peptide bond of the pentapeptide motif, and then transfers the threonine of the cleaved protein to the amine group of the pentaglycine cross bridge of the peptidoglycan, again forming a TG peptide bond. So it cleaves a threonine glycine peptide bond and it reforms a threonine glycine peptide bonds. Now, so it becomes obvious that it is actually possible in vitro that if you express a protein with LPX-TG sequence at the C terminus and another protein, for example, with glycine at the N terminus, you should be able to fuse them together using sortase. You can also take a synthetic molecule with glycine derivatized with all kinds of labels. You can put on the glycine a fluorophore spectroscopic probe, a radioactive label, a non-standard amino acid, or any orthogonal functionality. So, for example, in our lab, what we did was that we labeled recombinant proteins nested with LPX-TG sequence with azide and alkyne to make fusion proteins. So azide and alkyne, they very rapidly react in the presence of ascorbate and copper sulfate and produce a linkage that is a triazole kind of a linkage. So if you label these proteins with either alkyne, you can have a complementary protein with azide. You can actually click them together. So you can, this way, you can actually make large assemblies. For example, you can make a tetravalent protein. You can make, if you use a cyclodextrin kind of a scaffold, you can make a heptavalent protein. Mind you, these proteins will be highly chemically defined and there won't be any non-specific conjugations to worry about. The beauty of this system is that in the process of labeling by sortase-mediated ligation, you are expressing the protein with a histidine tag, the tag gets removed because it cleaves at TG, and T will go into, T will be derivatized with the label, and then it will be finally conjugated with the counterpart orthogonal functionality. So today I'll give you some example, two important examples where we have used sortase-mediated ligation to engineer sumo conjugates and to engineer semisynthetic histones with defined epigenetic marks for interrogation of eraser specificity. Now, first I'll talk about the sumo conjugates. Sumo biosynthetically, biosynthesis of sumo conjugates involve a complicated, complex process in which at least it involves 3 enzymes, E1, E2, and E3. So sumo actually is synthesized as a precursor where you have extensions here. For example, in sumo 1, you have a HSTV extension, then VY in sumo 4, VY in sumo 2, and slightly longer extension in sumo 3. Now, these are processed by the proteases, which are sumo-specific proteases called centrins, SNPs. So after processing by SNPs, this gly-gly sequence in the terminal, which is supposed to form a isopeptide linkage with the target proteins, gets exposed, and then it is activated by the E1 enzyme, which forms a thioester enzyme linkage, and through the intermediary of E2, the E3 forms a peptide linkage with the target protein, which is again cleaved by the SNPs, and the cycle continues. Now, this is actually multiple steps are involved, and if you really want to make these in vitro, it's quite unwieldy. But since the sumo proteins are about 100 residues long, they are amenable to chemical synthesis. But in chemical synthesis, you see, it is possible to do, and chemists have actually done it. You see, this is not for you to read it, but just to get a sense how complicated it gets if you indulge yourself in chemical synthesis of sumo conjugates. So this is just to give you a sense of the complexity of chemical synthesis. Now come back to sortase, how you can use sortase here. So you actually engineer a LPXTG, the pentapeptide motif, in the sumo sequence. And you see the sumo natural sequence is QEQTGG. So you already have this TG peptide bond. What you need to introduce is just a couple of sequence changes here. So you can introduce this QQ can go to LP, and then you have a substrate ready for sortase. Express it with a histidine tagged protein, and then you have a target peptide which is prefabricated with a gly-gly attached to the lysine. So this is already isopeptide linked, and then you mix the two through sortase. You can actually now this GG will go and attach here. This will come off. So sortase will cleave here and form a TG peptide bond with this one. So you have a sumo conjugate and this conjugate, you get it in simple one step. So here is an illustration of this. So you have, for example, engineered sumo 2, and this sumo 2, engineered sumo 2, actually is made with if you take this, the first step is that you test whether the SNPs are recognizing this or not. So the SNPs are recognizing. What it means is that the LP is not really interfering with anything. And then you see that, it can form this engineered SUMO2 with 2 residue substitutions here behaves in much the same way as the wild type SUMO behaves. It can form a conjugate with RAN gap 1. So after seeing this, now we took a peptide which is derivatized with gly-gly isopeptide linked from a P53 sequence representing residue 383 to 389, and this K is the target there in P53. You can make the conjugate very nicely in one step. So you get this conjugate here, and this is pure in one step. And then you check whether the conjugate is processed by the SNPs. Yes, it does. The SNPs cleave here in the conjugates in all the 3, SUMO1, SUMO2, and SUMO3. But if you take a peptide from SUMO, which is conjugated with the regions which is here, not the entire protein, that is not recognized. What it means is that the full-length engineered SUMO conjugate is processed by the simulation machinery and not of the short peptide, which tells you that the SUMO fold is actually important, and a short peptide sequences derived from SUMO are not going to substitute for that. So this is the summary of the process. And to summarize, what we did was we have generated a one-step assembly of bona fide SUMO conjugates, and SUMO conjugates serve as bona fide substrate for desomulating enzymes SNP1 and SNP2. Now, mind you, if you do not have conjugates of this kind with full-length SUMO proteins, you don't have actually a genuine substrate for these two desomulating enzymes. Now, following up on this, our work involved only the conjugation of a small substrate to the entire SUMO protein. And this concept was extended by Catherine Lang's group based on our work, and she actually added genetic coded expansion of the proteins and was able to put a large protein sequences to SUMO, which were generated with a LPX-TG sequence, which was meant for sortase-mediated ligations. So in other words, she actually coupled the sortase-mediated ligation with genetic expansion and another chemical step of Staudinger ligation. Now I'll move to the semi-synthesis of histones using quickly. I hope I have some time. So in here, everybody in this audience must be familiar that histones undergo a large number of PTMs, and which are epigenetic modifications brought about by writers, readers, and erasers. So writers, the enzymes which put the modifications, the readers are scaffolding proteins, and the erasers are the enzymes which remove them. So there are acetylation, for example, is a critical modifications which plays an important role in gene regulation. And it is removed by the HDACs. There are 18 HDACs, human HDACs, and the specificity of these HDACs is largely not known or not well defined. And what is required is that you need precisely defined chemical post-translational modifications on specific sites on histones. So the challenges are that HDACs exist in multi-protein complexes, and they may be active on hetero or homo oligomers, and the substrate specificity may be context dependent. So if you have histone which is modified on 50 sites, 60 sites, whether you pick up the right eraser for a given site is not really the way to go about it. So let us see how sorties can be helpful here. So this is an example of H2B. How can we introduce a specific modification in H2B and specific acetyl residue, for example? So here, what you can do is you can divide this H2B sequence into two complementary fragments. One can be obtained synthetically, and the other you can express it by, you can make it in a recombinant fashion. And then you need to introduce the sortase recognition motif. So sortase recognition motif, here you can introduce just two sequence changes, L and T here, and get the sortase pentapeptide recognition motif. And then in one step, actually you can introduce the acetyl group. Specifically here, in this case, we have chosen lysine 5 in H2B. So here is the synthesis, H2KB5 cis, K5AC can be separated, and it is recognized specifically by the K5AC, H2B K5AC antibody, suggesting that actually the mutations have not created any deleterious effect. Then this is you can purify it by HPLC, and then the masses are perfect, so it is well defined. Now the next you see if it is recognized by HDACs. Large number of not large numbers. I think some HDACs exist in complexes in cells. So it is imperative that you assay these deacetylation activities in which the HDACs are actually overexpressed in cell lines. So what we did was Shagun in my lab overexpressed these HDACs, and including sirtuins, all of them individually in HEK293 cells, prepared the cell lysates, and then tested the ability of the individual lysates to deacetylate the H2BK5AC. Now in here, clearly what emerged out was that HDAC1 was preferentially deacetylating the K5AC in H2B. And this deacetylation was inhibited by pyroxamide, which is a specific inhibitor of HDAC1. And further, when you use recombinant HDAC1, you can see time-dependent increase in deacetylation. And this process, again, the recombinant HDAC1 mediated deacetylation is inhibited by pyroxamide, suggesting that the overexpressed as well as recombinant HDAC1 preferentially deacetylates H2BK5AC. Now, what happens in nucleosomes? So, what Shagun did was that she prepared the nucleosomes nested with H2BK5AC, in which she first made the octamer, histone octamer, and then put the 146 base pair DNA vidome sequence and prepared the nucleosomes. And then on this nucleosomes, she tested the deacetylation by HDAC1. So this is the scheme for the preparation of the nucleosomes. So you can see that the prepared nucleosome is picked up by the H2BK5AC antibodies, and also by H2B, suggesting that the nucleosome is incorporated with the semi-synthetic acetylated H2B. Now, sorry. Okay, some slides are missing. But anyway, so now in what she also showed was that uh, the H2BK5AC encapsulated nucleosomes undergo deacetylation by HDAC1 specifically, both HDAC1 prepared from lysates and HDAC1 recombinant HDAC1. And she also used a couple of other recombinant HDACs to show that it is specific. And she also used peroxamide to show that the process is specific for this. So in summary, sortase-mediated semisynthesis is a viable strategy for engineering defined epigenetic marks in histones. And HDAC1 is the specific eraser of K5AC and H2B. And now what one can do is that one can prepare modified nucleosomes with now defined multiple synthetic H2B or H3 or H4, any of these, and study the multivalent interactions in nucleosomes, and obviously if you go further into the chromatin. Now with this, I would like to end and thank my PhD students whose work I presented today. Bulk of the work on sumo was done by Vijay Kumar Pawale, and on histones with Shagun Sukla. And the other work that I mentioned was done by Sharmista Saini, Kanchan, and Tora. And none of them are presently in the lab. I'll have occasion probably to present work of others who have not been mentioned here some other time. And of course, the collaboration of Ram Kumar is still, we are engaged in solving structures of new sortases with new specificities to expand the synthetic ambit of sortases. And I was here in IISc from 84 to 89, the wonderful years, the most memorable of my career in life. And then for 27 years in NII, and now at the Regional Center of Biotechnology. Thank you very much.
Thank you, Roy, for
Professor Savitri, you're muted.
if there are one or two questions pressing, we could take. Are there any questions? If we don't have any questions, I thank Roy for that excellent, clear presentation of the work carried out for over several years. I thank him very much for this. Thank you.
Professor Savitri, Patrick has a question.
Yes, please.
Patrick.
Yes. So my question is, RP, is that sortase require the specificity with the GGK sequence all the time? Does it takes other than GGK, I mean target protein? I'm not.
Okay. Actually, I had all that. I missed completely. I actually put a wrong PowerPoint presentation. But anyway, doesn't matter. So yes, what we actually, our first paper was not about GG, but about, the 6 amino sugars, only 6 amino, not 2 amino like glucosamine, but 6 amino like 6 amino deoxy sugars, such as those present in aminoglycoside antibiotics, kanamycin, tobramycin, and so on and so forth. They are very, very good substrates, replacements for GG. Now, the reason is at the 6th position, you have NH2, and before that, you have CH2NH2, right? CH2NH2. And that CH2NH2, and then followed by the oxygen in the ring, I think some way that mimics the CH2NHT CO at the C terminus of glycine. So that was the first paper wherein we suggested that you can use this technology to make neoglyco conjugates. And we made several of those neoglyco conjugates and published in JACS in 2008.
Yeah, okay, thanks.
You're muted again.
Any more questions? If there are none, I would like to thank Roy once again for a wonderful presentation.
Thank you, Savitri.
It was nice seeing you. Yes.
Thank you so much.
Yes. So we now move on to the third speaker of the session, Dr. Mohan Babu. He obtained his PhD in 2004 from MRC Laboratory, Molecular Biology and Trinity College, University of Cambridge. And then he was a postdoctoral fellow at NCBI, Maryland, USA. Now he is currently a faculty in the St. Jude Children's Research Hospital at Memphis. And he's also the director of the Center of Excellence for Data-Driven Discovery, endowed chair in biological data science. His lab focuses on data science approaches to make biological discoveries and reveal fundamental principles of biological systems by employing and developing computational and experimental methods to study biological systems in different levels, molecules, processes, and genomes, with the overall idea of understanding cellular regulation in relation to human diseases. And I would like to, he would like to understand the complexity at from systems at atomic level to the network of interaction between molecules and those pertaining to the level of entire population. So here again, we will have a wonderful talk and very current talk, research. So I invite Madan Mohan to present his talk.
Thank you very much for the kind and generous introduction, Professor Savitri, and good evening, everyone. I just wanted to make sure that the presentation view is on screen. Could someone confirm, please? Yeah, thank you.
Yes, it is there.
Yeah, yes. Excellent. Yeah, I'd like to take this opportunity to thank Professors Nagasuma Chandra, Dr. D.N. Rao, and P.N. Rangarajan for the kind invitation to present our work on this special occasion. And personally, for me too, it's a very special event to give this talk, as I owe a lot to the Indian Institute of Science for the unique opportunities and for nurturing my interest in science, especially to Professor Balram, Vijay, and Srinivasan at MBU, and Professor G. Padmanabhan and Nagasuma Chandra in the biochemistry department, among several other mentors at IISc. Now, as Professor Savitri mentioned in her introduction to the session, I'm one of the many who has benefited from being trained at the IISc, and also distinctly remember attending her talk and several others that I attended as an IISc young fellow in the early 90s. All right, so what I'd like to do in the next 25 minutes or so is talk to you about some of the recent work that we've been doing on GPCR signaling, particularly using data science approaches to understand GPCR signaling, as well as discuss the implications for drug discovery and development. Now, technological advancements over the last several decades have allowed us to generate huge amounts of information describing biological entities and processes at different scales and resolution. Now, this presents us with unique opportunities to use computational methods to gain fundamental insights and discover new scientific principles. Now, the current estimates, according to the National Institute of Health and the European Bioinformatics Institute, is that we have access to over 150 petabytes of biological data. Now, given this sea of information, what drives us scientifically as a team is to find out how we could discover new biological insights and knowledge from large volumes of data. So the mission of my group is to pioneer data science approaches to make fundamental discoveries and reveal principles of living systems, with the emphasis on understanding human biology. So one of our primary research interests, as Professor Savitri mentioned in the introduction, is to understand human biology and diseases through data-driven approaches. And for this, we investigate biological systems at 3 distinct levels of complexity, the atomic level, the cellular level, and the population level. And we bridge these different levels by investigating the impact of mutations across all these 3 different levels. And to this end, my team develops interdisciplinary computational and experimental approaches that tries to generate, integrate, mine, and analyze disparate data sets. And these include data describing sequences, structures, expression, networks of interactions, and many more. So in my team, there are 2 major scientific directions. So one is broadly centered around understanding structure-function relationship. And here we have been focusing on G-protein coupled receptors. And the second broad theme is to understand what's called the disorder function paradigm or relationship, where proteins can adopt diverse conformations, and each one of these conformations can be relevant for function. But in today's presentation, I'm going to focus more on our work on GPCR signaling. So what are GPCRs? So GPCRs, or G-protein coupled receptors, are the largest family of membrane protein receptors in the human genome. There are more than 800 different genes that encode for GPCRs, and they have a 7-transmembrane architecture, and they sit on the membrane where they end up binding the ligand on the extracellular side and triggering an intracellular signaling cascade. Now, each one of these 800 receptors participate in diverse physiological processes, ranging from sensory responses such as vision, taste, and smell, to those regulating mood and behavior, the immune system, the cardiac system, and so on. So in this manner, GPCRs govern virtually every aspect of human physiology. Now, given their role in modulating physiological processes, it is actually not surprising that they serve as attractive drug targets. So currently, over 1/3 of all FDA-approved drugs target about 108 different GPCRs. And more than 300 different agents that target an additional 66 receptors are under clinical trial to treat various diseases and disorders such as obesity, diabetes, asthma, among others. Now, in the last few years, there's been enormous progress in GPCR structure determination. So we have over 500 structures of 91 different GPCRs, thanks to development in structural biology methods, as well as protein engineering methods that allow us to get structures of these important protein complexes. Now, when we have 2 structures, it is very easy to compare what the similarities and differences are. But then when we have a large number of structures, it becomes very difficult to visually compare what the similarities and differences are. So in my team, we developed some network-based approaches that now allows us to compare these receptors and receptor complexes to pin down what are the common principles and differences. Now, this was an idea that was introduced to me by Professor Saraswati Vishweswaraya at the MBU when I was a summer student at the Institute. And this is something that I've developed an interest in and pursued further in my career. So these network-based approaches are very important simply because it now allows us to apply graph theory principles to be able to pin down what the similarities and differences are. Now, using these approaches and through the work of one PhD student at the time, AJ Venkatakrishnan, who was also trained in Suma's lab, we systematically analyzed all the different protein receptor complexes and proteins in complex with the different ligands to understand some common and general principles. And what we were able to do was to discover what's called the ligand binding cradle, which are spatially equivalent positions that are always making contacts with the ligand. So even though they're chemically different, spatially it's always the same position on the receptor that contacts the ligand in the different GPCRs. And by comparing the structures of the active state and the inactive state, we were also able to discover common structural rearrangements, specifically individual residues that always are rearranged in order to be able to interact with the G protein. Now, through the work of another very talented PhD student, Tilman Flock, at the time in the group, we were also able to look at how GPCRs selectively regulate to couple to a G protein. So there are 16 different G proteins, and the precise interaction is really important because different second messenger molecules get triggered, and they can have important consequences, as we heard from Sandhya's talk earlier this session. So what we were able to do was to use computational approaches that integrated sequence data and structure data and comparative genomic data to discover what's called the selectivity barcode of how a receptor couples to a particular G protein. And then again, through structure comparisons, we were able to understand the allosteric pathway that can explain why binding of a receptor can activate a G protein. So through systematic investigation and integration of sequence, structure, and many other related data sets, we were able to describe general principles about ligand binding, and how ligand binding eventually results in receptor activation, and how activation of the receptor is now coupled to the selective binding of the G protein, and how the binding of the G protein to the receptor then allosterically triggers the release of the nucleotide. Now, in another study from our group, we also integrated data on receptor structures with information about population variation from over 60,000 individuals in the human population at the time. And now this allowed us to demonstrate that many of the GPCRs that are targeted by FDA-approved drugs are actually highly variable at the drug binding pocket in the human population, and highlighted the implications for adverse drug response and personalized medicine. So I won't be talking about this today, but I'm happy to engage in a discussion if anyone's interested on this problem. In today's talk, what I'd like to do in the remaining time is to focus on one particular study that was published recently, where we wanted to understand what is called as tissue-specific differences in GPCR signaling by investigating transcriptome data from over 30 different human tissues. So this is a work that's done by a very talented Marie Curie fellow in my group, Dr. Maria Marti. So this was the problem that we became interested in. So given that there are over 50 different organs that are made up of over 200 different cell types and 37 trillion cells in humans, do all cells and tissue respond the same way to a given ligand? And what are the mechanisms that can diversify signaling in the different tissues, and how prevalent are these mechanisms? So we started to focus on alternative splicing as one of the mechanisms that can generate this diversity. So as we all know, alternative splicing is a molecular mechanism where the same gene product can give rise to different mRNA sequences through alternative splicing. And in this manner, you can give rise to different protein sequences with slightly altered amino acid sequence, and thus you can alter the function of the polypeptides that emerge from these different splice variants. Now, transcriptomic data has really revealed that over 90% of the human genes undergo alternative splicing, and a large fraction of the isoforms tend to have tissue-specific distribution. So what was still unknown is that how exactly can this influence GPCR signaling on a systems level? So the three questions that we started to address were the following. How many of these GPCRs have more than one isoform? And how exactly do they differ in terms of their structure and function? How does their expression contribute to the diversification of the signaling response? And more importantly, what are the implications for drug response and drug development of this isoform diversity that may be occurring in the human tissues? So to address this question, what we did was to systematically integrate multiple large-scale data sets. The first one is called the GTEx data, which has transcriptome information for over 30 different tissues from 700 different individuals with the GPCR-DB. And through developing a computational pipeline, we were able to identify all the different isoforms that are expressed in the different human tissues. And what we were able to find was that over 150 of these receptors have 2 or more isoforms expressed in at least one of these tissues in multiple individuals in the human population. So what exactly are the changes that happen in the isoform? Are they simply changes that happen in the 5' and the 3' UTR, or do they actually affect the protein sequence? Now, to address this, we again developed a computational method that then systematically classified through creation of multiple sequence alignments and also analyzing sequences and relating them with respect to the different structural segments. I told you about the 7 transmembrane regions. So we systematically mapped all these sequences and asked where exactly the changes happen. And this allowed us to classify the isoforms into 2 broad categories. So the first one is called topologically preserved. They have the same 7 transmembrane architecture, but only change their N-terminus and C-terminus or intracellular loops. And these are important functional elements, and I'll try to explain to you in a minute. And then there are others where they had a completely different transmembrane sequence or had truncations. To also characterize and quantify what these changes are over the entire superfamily, we developed what's called as a isoform fingerprint, where we started to represent the presence of a region with a black dot and absence of the region with a gray dot. And this allowed us to systematically ask where changes happen in all these different isoforms that we detected in the human tissues. And when we do this, it's not surprising that the reference isoform is the one that is most dominant. But then right after that, you find changes that happen in the N-terminus and changes that happen in the C-terminus as the two most prevalent mechanisms by which GPCR isoforms get generated. You also have systematic truncations that are always detected for some receptors, and then a number of different fingerprints, which I won't be going into in today's presentation. So mining the literature at the time also allowed us to identify what people have done to characterize isoforms in terms of some of these receptors. So there have been individual studies that showed that isoforms that alter the N-terminus can affect how ligands can bind, and isoforms that affect the C-terminus can influence how the receptors can bind to G protein or arrestin. And then you can also affect the post-translation modification sites. And what this means is that you can affect the recycling rate and trafficking rate, and therefore cell surface expression differences on the membrane. And you could also affect how the isoforms can interact with other GPCRs, because some receptors are known to form heterodimers. And then when you have truncated versions, you can have dominant negative effects. So these are all some of the reported effects of isoforms on individual receptors that were available in the literature at the time when we performed this study. So what we then did was to map all the known post-translation modification sites that have been detected using large-scale mass spec studies by many researchers for many different studies. And when we mapped these mass spec data, which had peptide evidence and phosphorylation evidences, they systematically mapped many of these isoform sequences that are alternatively spliced in one tissue but not in the other, again suggesting that many of these can have implications for downstream signaling and can actually bias the signaling state of the receptor in these tissues. Now, the receptors in black here, I mean, it's not easy to see, but in the paper, it's more clearly described, are the ones for which we had literature evidence. But through the computational analysis, we were able to identify many other receptors that have been uncharacterized yet that have isoforms with the same structural fingerprint, suggesting likely functional implication and mechanism for some of these isoforms. So in summary to the first part, what I'd like to highlight is that most of the non-reference isoforms can affect functional hotspots such as ligand binding or receptor coupling, and that characterization of these structural fingerprints suggests the most likely effect of the functional impact of these unstudied isoforms. So the key message is that isoform structural variation can diversify the receptor functional effects. So how can GPCR isoform expression influence signaling states in the different tissues? So this was the next question that we focused on. And here, we again looked at the expression level of these isoforms in the different tissues across the different individuals in the human population. And we could kind of characterize two ends of the spectrum. So in one case, one can imagine that all the isoforms are expressed in all the tissues, in which case there is going to be no variation across the different tissues. Or different isoforms may be selectively expressed in different tissues. And what this means is that for the same ligand, you can generate tissue-specific differences in signaling states. So we developed a computational method that tries to quantify this mechanism, and we call this tissue expression signature. So I'll just walk you through this one example of the cannabinoid 1 receptor, where you have 3 isoforms, but you can see that in the different tissues, different combinations are expressed. However, CD97 also has 3 isoforms, but all isoforms are expressed in all these tissues. So one would expect that for the cannabinoid receptor, for the same ligand, we could expect tissue-specific responses because of the complex expression pattern in the different tissues. However, for the CD97, the same ligand is likely to generate similar responses, at least at the receptor ligand engagement level. You can find differences in G protein signaling, but that's something that I won't talk about today. So when we systematically characterize these differences, we find that most receptors display a unique combination of these isoform expression pattern. And what this suggests is that the number of different tissue expression signatures can likely determine how complex the signaling responses can be in the different tissues. Now, I'm just going to focus on one such receptor, as I mentioned, the cannabinoid receptor, where you have differences in the N-terminus. So there is variation in the sequence of the N-terminus. And then they are also differentially expressed in the different tissues in different combinations. And therefore, the first question that one can ask is, do combinatorial expression and exposure of this combinatorial expression to the same concentration of the ligand result in differences in downstream signaling? So we collaborated with Manoj Puzanvidu and Stephanie Crilly from University of Michigan, who were able to look at different combinations of isoform expression in cell lines based on the expression pattern that we identified through the computational analysis to show that you can alter the downstream signaling state to the same ligand if you have a combinatorial expression of an isoform or not. Now, this is interesting because it has implications for drug development, where when you have a cell line and you express a protein of interest and characterize ligand binding through cell-based assays, knowledge of whether an isoform of this protein is expressed or not is important, because otherwise what the measurements actually do point to is a collective response of all the isoforms in the cell rather than the response of the protein of interest that is heterologously expressed. Now, we did a similar follow-up on one of the C-terminal variations where clearly some of the phosphorylation sites have been altered because a new sequence of the C-terminus was introduced. And here again, through collaboration with Graham Latz and Alison Pierce and Matthew Harris in Graham's group, we were able to show that the co-expression of one of the GIPR receptors results in what is called as bias signaling. So the same ligand can bias downstream signaling response towards cyclic AMP or towards beta-arrestin recruitment. So to summarize this part of the talk, what I wanted to highlight is that all tissues can express non-reference isoforms in combination. Different receptors have different receptor expression signatures, meaning they're expressed in various combinations across the tissues, and that receptor isoform combination can influence downstream signaling response. So the key point that I wanted to highlight is that such isoform combinations can diversify signaling response and may drive what is called as physiological signaling bias. So the same ligand can result in tissue-specific responses in a context-specific manner. All right, so far what I've tried to show you is that there is data suggesting that GPCRs can undergo alternative splicing, and that you could influence the structure and therefore the function of these isoforms, and that these isoforms are expressed in various combinations in different tissues, and thus you could have tissue-specific differences in signaling responses to the same ligand. And I also mentioned to you that GPCRs are one of the major drug targets. So putting all these observations in question, in context, an obvious question emerges. What does this actually mean for GPCR drugs and drug development? So to address this, we focused on just the subset of GPCRs which are known targets of FDA-approved drugs. And we find that 43% of these drug targets have 2 or more isoforms detected. And we thought this could either complicate signaling, or this may present an opportunity for what is called isoform level drug selectivity. So through systematic analysis of where the sequence and structural changes are likely to occur, and also by looking at which tissues express the isoform together or the isoform uniquely in terms of drug and the relevant tissue in which drug action is expected to require, we were able to narrow down a list of about 42 receptors and 64 isoforms that may present interesting new isoform level drug targets. I'm just going to walk you through 1 or 2 such examples. So here is the example of CCR3, which is a chemokine receptor. There are 2 versions of the CCR receptor that is expressed, one which has a shorter N-terminus and the one which has a longer N-terminus. And this is exactly the region that also wraps around the chemokine ligand in order to make the receptor ligand complex. And clearly, the different isoforms are expressed in different tissues, especially in the blood. One isoform is expressed, whereas the other isoform is never expressed in some of these tissues. And therefore, thinking about targeting these regions might prevent off-target effects in some of the other tissues, which might express a different isoform that may not be the relevant tissue where the drug is expected to act. So this is another example of CRF receptor. Again, the N-terminus is different, which can bind to the ligand, and they do show complex expression pattern, suggesting a potential opportunity for isoform level drug selectivity. Now, so far, I've showed you only data on transcriptome levels. The real question is, are they really made into proteins? So we then went ahead and mined public data sets describing peptides. And the key difference here is that we reinterpreted many of these peptide spectra. Because in a typical proteomics pipeline, you need a reference database. And if you don't have a reference database, you cannot match the fragment to a particular peptide. So what we did was to reanalyze and re-engineer the pipeline to introduce and reinterpret many of these spectra by having these isoforms, and that allowed us to detect the expression of these isoforms at the protein level. And we also analyzed individual cells, because all these transcriptome experiments are done in entire tissues. So you don't know whether the same cell type expresses both these isoforms together. So we looked at individual cell lines. And in the manuscript, we also discuss results from single cell data analysis, which shows that the same cell does express multiple isoforms. So this combinatorial expression is indeed a relevant phenomena. And most importantly, we can also ask, well, is it simply an artifact, or can it lead to some sort of a genetic variation and phenotypic differences in the human population? So for this, we made use of what's called the UK Biobank, which essentially provides information about single nucleotide polymorphisms, as well as phenotypic associations for about 500,000 people in the UK population. And by mining this data, we were able to find several polymorphisms that map uniquely to the isoform segment only, but not to the common parts of the different isoforms. And what this essentially suggests is that there is an underappreciated potential for isoform-specific therapeutic indications. So like I was mentioning, this CXCR3 is another receptor where you have the isoform, and you have polymorphisms that show association with certain disease phenotypes, but not the non-reference isoform. So in summary to this part, what I'd like to highlight is that around half of the GPCR targets for approved drugs do have more than one isoform. There is genetic evidence that links receptor isoforms with specific disease phenotypes in the human population. More importantly, these non-reference isoforms, as much as they can complicate signaling, could potentially provide an opportunity by conferring tissue-specific selectivity and may represent novel, previously underappreciated drug targets. So the larger idea is that one could think about rationally exploiting isoform level drug selectivity for developing new drug candidates that may minimize off-target effects and also minimize potential side effects. So all the information that we have compiled has been made publicly available through what's called the G protein coupled receptor database, which is the database for GPCRs that is maintained by our collaborator, David Gloriam at Copenhagen University. And in summary, what I wanted to therefore conclude is that through a systematic integration of multiple large-scale data sets, ranging from atomic level information on structures to expression level information, as well as sequence level changes on the entire human population, we could gain a first insight into the prevalence, the functional impact, and the likely signaling complexity due to the expression of different GPCR isoforms. So the general idea then is that all these observations suggest a need to move from a canonical view of GPCR signaling, where we have a single receptor that can couple to a G protein or arrestin, to what is called a context-specific view of GPCR signaling, where in different tissues, you can have different combinations of isoforms that are expressed. And those combinations may have alterations that may be unique, which can bias the signaling of the system in response to the same ligand. And thus, the varying receptor isoform repertoires can result in system-specific changes in the signaling output to the same ligand. So in summary, I've tried to show you that GPCR isoforms affect functional hotspots and can diversify receptor function, and that unique combinations of isoforms are expressed, and this can result in unique signaling states and can contribute to physiological signaling bias in different tissues, and that such isoforms could explain complex pharmacological profiles of current drugs and may also offer new therapeutic options. So this could be one reason why drugs that work in cell level ends up failing for multiple reasons when one takes it to tissues as well as animal models. So there are a number of implications of this study, and I'm personally excited about a couple of them. So the multi-scale data integration framework that we have developed to study isoforms can be adapted to study other important drug targets. And we are pursuing this for investigating ion channels, kinases, and nuclear receptors. And it could also be used to understand discrepancies in drug response in different cell lines, because the knowledge of the expression of an isoform, if it is not sufficiently documented, this could lead to cell line specific differences, even though one may be studying the same receptor. And it also points to the importance of identifying the right model system for screening drug-like compounds. If some of these isoforms are not expressed in a mouse model, then this can lead to model-specific differences that may not be unaccounted for in these development strategies. As I also mentioned, it can also allow for discovery of novel isoforms for drug development, and could possibly explain how organisms can mount different responses to the same ligand at the tissue level, leading to coordinated but homeostatic response at the organism level. So this is how differences can come together to create a unified response at the level of a whole organism. So in conclusion, I hope I've tried to give a flavor of the kind of studies that we do in my group and how existing data sets as well as new data could allow us to gain new biochemical as well as mechanistic insights, and how the knowledge of these new biological processes and new contexts could potentially be exploited for interpreting the effect of mutations as well as in understanding diseases and possibly for drug development. So with this, I'd like to conclude my presentation and acknowledge a number of co-workers. I think I've mentioned many of the key people involved in the project as we went along in my presentation. I'd also like to take this opportunity to thank St. Jude Children's Research Hospital and the Medical Research Council, LMB, for long-term funding for many of the projects that I presented in today's talk. Thank you very much all for your attention, and I'll be happy to answer any questions.
Thank you, Dr. Mohan Babu, for such a wonderful, clear presentation of data analysis and using it for finding newer insights into the GPCR functioning. Thank you very much. Now, if there are any questions, I would be happy you can ask any questions.
Could I ask a quick question? I'm not able to raise my hand. Suma here.
Yes.
That was a fantastic talk, Madan, a real treat. So a quick question is, you've looked at a lot of transcriptomes, tissue transcriptomes. Do you think the context-specific signaling could be identified just by looking at transcriptomes, assuming there is efficiency in expression?
Yeah, no, that's a great question. So one aspect that we are looking at is regulation of these isoforms through microRNAs. Because not just on the receptor level, but also on the G protein and the arrestin level, because what we're able to see is that every tissue may also have a unique signaling state. The balance between the G protein levels as well as arrestin levels could further influence the signaling bias that I was mentioning to you. I think it requires quantitative modeling, which will have to be done on a system by system basis rather than on a whole family basis. So if one focuses on individual receptors and a relevant downstream signaling pathway, one could think about modeling them in different tissues. So we are looking at cancer states and healthy states. So that provides a very straightforward mechanism by looking at the Cancer Genome Atlas data set, because for every individual, you have matched data on normal tissues as well as the mutated tissue. And hence you could do a more cleaner comparison, but across individuals, this becomes a little bit more tricky. But focus studies, knowing that, here is a receptor system where there is isoform, I think that's the knowledge that studies like this provide. And hopefully, it'll serve as a starting point for those interested in those individual receptors to go in greater detail.
Thank you.
Saraswati, Sandhya Vishweshwarya has a question. Sandhya?
That was really great, Madan. I
Thank
have one question on each of the two parts. The first relates to your analysis of different structures of GPCRs. Now, many of these structures have been obtained by doing all sorts of strange fusions, adding various epitopes, conjugate, crystallize in the presence of single chain antibodies. Right. How close do you really think they are? And what about conformational plasticity in terms of,
question, yes. And you had a second question, is it right?
The second question I can ask now or I can ask later.
Sure,
it's related to
right
your
now.
second,
No, this is a great point, because I think that's why there's been a lot of interest on performing NMR experiments on these receptors. So people have been labeling individual residues either through hydrophobic residues or through fluorine labeling. to be able to understand the conformational plasticity. There are also semi-low resolution information like FRET-based studies, which have looked at movements of individual helices. And I think these are really important points. And that's why many of the crystal structures are really an exaggeration of one of the stable substates. And the use of antibodies is really important because it just allows you to get a stable state, exactly. Otherwise, these are dynamic molecules. So from that perspective, the active states have been characterized extensively because structure function studies have been done to show that the states that have been identified as active and therefore residues that are important when you mutate, you lose functional activity. So it isn't that these are less relevant structures, but these are going to be amplified to be able to capture them in otherwise not easy to capture states for structural studies.
Sure. Yeah. And the second question relates to the second part of your talk, where you talked about isoform expression in different cell types. How confident are you that you actually can correlate that with absolute levels of these isoforms?
Yeah, no, that's a really
So I think mass spec data.
Absolutely. So this is a very, very important question, which is why we had to ask this question ourselves. So all these are transcriptomic studies. Do we even know that these are made into proteins? And this, again, takes us to a very important point, because absence of information doesn't mean that it doesn't happen. So what happens in these mass spec studies also is that you have a reference database. I was just trying to very briefly gloss over it. And if the reference database never has an isoform, you'll never detect it in your mass spec experiment. So what we had to do was to reprocess all the data that was thrown away because they were not mappable. And when we looked at these data that were not mappable, but using a new reference database saying that, these are the new sequences or new junctions that we should detect if these isoforms are real, we could start to map them. Now, this essentially means
can it be quantitative? Is it quantitative?
That is a separate question. So one thing is we can say that, these are expressed, and this serves as a starting point for let's say someone interested in this receptor system to then study in the right cell type or the tissue type. It's really to even get a first view of, yes, there's an isoform here, it is expressed in this tissue, and now is where the starting point happens to be able to go and study the functional relevance in a more systematic manner.
Thank you so much. Thank you.
You're very welcome.
Thank you. We are already running late, so I think we should stop the discussion at this stage. Thank you very much for that wonderful presentation. Excellent piece of work. Thank you very much.
Thank you.
Yeah, we now move on to the last speaker, Dr. Joman Joseph. He obtained his PhD from the Department of Biochemistry in the year 2000. He was, I was the mentor for him, and he started with characterizing an economically important plant virus. But then soon after he got his PhD, he moved for postdoctoral fellowship in NIH, where he developed interest in cell biology and process of cell polarization. So after returning in the year 2005 at National Center for Cell Science, he has been engaged in the understanding of the process of cell polarization, how different cellular functions and signaling pathways are coordinated to achieve polarization is the major focus of his lab, and he's deciphering some unexpected roles of nucleoporins in this process. I now request Dr. Joman Joseph to give his presentation.
Thank you, Professor Savitri, for the wonderful introduction and nice words. And I feel privileged and thank all the organizers, Professor Rangarajan and B.N. Rao, for allowing me to give a presentation of one of these interesting stories that is shaping up in my lab. And actually, I will be more often referring to two words, that is relationship and contacts. And to start with, the first relationship I should mention is that today, as we celebrate Centenary, celebration of the Department of Biochemistry, it happens to be my wedding anniversary, and I'm very happy that my wife is there supporting the scientist madness the idiosyncrasies and excitement and frustrations. And coming to the next relationship is the Department of Biochemistry, wherein I had done my PhD under Professor Savitri, as she has already mentioned. And Professor Savitri's lab is not standing alone, because we had a very able hands supported by Professor Appaji Rao, Professor M.R. Murthy, and Angelica Kande. We used to have a very extensive and interactive kind of sessions, whether it be lab sessions or scientific discussions, or even outing, whether it be birthday parties, etc. So I think this was a very dynamic situation wherein I was fortunate to be there, and then I have learned a lot of things in how to do science, the ABCD of doing science. And from there, looking at the structure-function relationship of proteins, whether viral proteins or human proteins or proteins from other sources, once I set up my lab at NCCS, we started looking at structure-function relationship of cellular organelles. Now, how we ended up in this particular area is something actually I would like to mention here. And also in this particular slide, I would just highlight or give an overview of what exactly I'm going to talk today. Now, we know that the endoplasmic reticulum is an already known structure in the cytoplasm, which makes extensive interconnections. And one subdomain of endoplasmic reticulum is called annulate lamellae. Now, annulate lamellae is a structure wherein a subset of nucleoporins are present. Now, nucleoporins are already known to be present on the nuclear membrane, but a subset of nucleoporins are also present in the cytoplasm as a part of the endoplasmic reticulum. Now, this has been known for quite some time. Maybe in 1950s, it has been known that such a structure existed when people have looked at different cellular structures at the EM level. But the functions are still unclear. People think these are the structures where the nucleoporins are deposited, and as and when required, they will be given to the nuclear membrane during the nuclear pore complex biogenesis, which is true in the early embryogenesis intracellular. People have found it. But even in non-embryonic or non-germ cells, in somatic cells also you find these kind of structures. So we thought we could be having additional functions. And a few years back, actually, we could show that these nucleoporin-containing structures associate with the mRNA containing structures in the cytoplasm, such as P bodies and stress granules. And they do some interesting function, that is the NUP350 at one of the nucleoporins, which is present at the annulate lamellae, is involved in regulating microRNA function. But today, what I'm going to talk to you is about, we were interested in looking at this structure further and then look at if any other unidentified or functions are, we could identify. I felt that, this is a structure which has been neglected for so long time. Now, if you look at the other structure, so nowadays, I think there are multiple organelles that are present in the cell, as we all know, mitochondria being one of them. Interestingly, mitochondria and endoplasmic reticulum make physical contacts. That's where I'm coming to the next contacts. That is, ER and mitochondria, they make physical contacts. Now, people know mitochondria plays a role, ER plays a role separately, but then, in coordination with each other, the contact sites are important for modulating many functions, which has come in the recent past. Now, I'm just giving a brief summary of what I'm going to tell about. It is already known that one of the growth factor mediated signaling, that is mTORC2 AKT kinase, actually stabilizes ER mitochondrial contact sites. But what I'm going to tell today is that the annulate lamellae or NUP358 containing annulate lamellae, which is a part of the ER, also is somewhere in close proximity to the ER mitochondrial contact sites. And interestingly, what we find is that the NUP358 as a part of the annulate lamellae also regulates the mTORC2 mediated stabilization of the ER contact site. Actually, annulate lamellae or the NUP358 in annulate lamellae is playing a negative role. That is destabilizing the ER mitochondrial contact sites by negatively regulating the mTORC2 kinase. Now, before I forget, and if the time is running out, I should show the important slide, that is coming to the next relationship, that is the mentor-mentee relationship. Again, the work which I'm presenting is a small story which is developing in the lab. It's been mainly done by a very talented PhD student in my lab, Nisha, and also supported in the initial stages by Pallavi, who was a DPTI. And also, I would like to thank all the financial support from different sources. Now, interestingly, ER and mitochondria, the contacts have been known for quite some time. Endoplasmic reticulum has been identified in 1945 or labeled or named. Carl Benda in 1898 coined the term mitochondria, and they started looking at the functions of these organelles. And at somewhere in 1977, by Jamshed Tata, they showed that ER and mitochondria can co-fractionate, even in fractionating it. The ER can exist separately, as well as ER with heavy fraction of ER, which contains mitochondria along with it during fractionation. Now, after that, a lot of functions have been attributed. People also found that in the EM level, the mitochondria can make contacts with ER. And another, 2013, which I'll be referring to is, as I've already mentioned, mTORC2 pathway is shown to be stabilizing the ER mitochondrial contact site, which is also called MAP for mitochondria-associated membrane. Now, coming to the an interesting connection between ER mitochondrial contacts and biochemistry department. In 1982, I was just going back to papers, and then I found interestingly that Professor G. Padmanabhan had a paper published in 1982, wherein he shows that the cytochrome C oxidase is actually preferentially synthesized in the rough endoplasmic reticulum mitochondrial complex in rat liver. So his studies actually led them to actually show that or conclude that the specific activity of the cytochrome oxidase was actually more in the endoplasmic reticulum mitochondrial complex than even in the mitochondrial fraction. So that made them think that there could be something exchange between the ER and mitochondria happening at that level. Now, things have taken into detailed directions, and with the advent of more imaging techniques and then functional characterization of many things. Now, the ER mitochondrial contact sites have been shown to be important for calcium homeostasis, lipid synthesis and then transfer, and autophagy signaling hub, everything. Now, given the importance of these contact sites in regulating different aspects of cellular process, it's not surprising to say that many of these processes are misregulated during diseases which are metabolic or neurological disorders. Now, so what this ER mitochondrial contact sites is made of? They come into physical proximity, and that is mediated through subset of complex proteins which are present on the ER membrane and also on the mitochondria, and they do interact with each other. This is what is making them come close to each other, and that is what is important for maintaining the distance of around 30 nanometers between them. Now, if you look at the ER mitochondrial contact sites in a fluorescent microscope, we label the endoplasmic reticulum and mitochondria in green and ER in red. You could see some kind of a physical proximity like this. And if you look at the EM's electron microscopy, you would see that the mitochondria is very close to the ER structure. And these are the contact sites which has been shown as ER mitochondrial contact sites. Now, what led us to the question, and then wherever we stand right now, is that the NUP358, which is one of the nucleoproteins present on the nuclear envelope, as is already seen, is also present in the cytoplasm, as you would see as puncta. Now, as I already mentioned, it's already known some of the nucleoproteins can be present in the cytoplasm as a part of the endoplasmic reticulum and the annulate lamellae. So we co-stained it with the endoplasmic reticulum. If you have a closer view, you can see that this red puncta, which are the NUP358 structure, is actually associated with ER, consistent with the idea that the annulate lamellae is a part of the endoplasmic reticulum. Now, if you look at the EM, which has been done, I think 1960s or 70s, that the annulate lamellae actually makes stacked endoplasmic reticulum structure with some pore-like structure, which they at that moment or at that time they thought it's something similar to that of the nuclear pore complex on the nuclear envelope. But the structure has been known for quite some time, but the functions is not known. Now, starting from the question is that endoplasmic reticulum subdomains are annulate lamellae. Endoplasmic reticulum makes contacts with the ER mitochondria, making a DR mitochondrial contact site. The simple question is that whether the annulate lamellae, what we are talking about, is away from the ER mitochondrial contact sites, or could be proximal to the ER mitochondrial contact sites. Now, to see that, we did a co-staining with triple staining. That is with ER in green, what you would see, mitochondria in blue, and the NUP358 as a part of the annulate lamellae or as a marker for annulate lamellae. Now, if you see a little closer, this is the ER and NUP258, as you would expect. The red puncta is associated with ER. And if you look at the ER and mitochondria, which is also known that the ER makes extensive contacts with mitochondria, you would see blue mitochondria is in proximity with the ER. And if you look at the NUP358 and mitochondria separately, you would see that the NUP358 structure or the annulate lamellae seems to be kind of associated or quite close to the mitochondria. And that being already the ER subdomains, it appears that the NUP358 positive annulate lamellae is somewhere at the interface between ER and mitochondrial contact sites in many places. Not all the annulate lamellae will be a part of the ER mitochondrial contact sites, but a subset of them are sure. Now the question is that it's simple. Now you have annulate lamellae, which is a part of the ER, is somehow in close proximity with the ER mitochondrial contact sites. So the hypothesis was very simple. NUP358 as a part of the annulate lamellae could be regulating ER mitochondrial dynamics or its functions. Now, to start with, we first asked whether NUP358 is important for maintenance of the ER mitochondrial contact sites, or in other words, if you remove NUP358 from the cell, what would happen to the ER mitochondrial contact sites? So one way of looking at it is that we have labeled the ER with RFP and mitochondria with GFP. And we can always look at the overlap of pixel, that is intensity of both green and red in the same pixel, which can be estimated by the software. And then this will give an idea about the kind of overlap between the ER and mitochondria. And if you quantitate, you could see that in the control saRNA, as compared to that, in NUP358 depleted condition, you see an increase in the ER mitochondrial contact sites. Now, this is a microscopic technique, and then we thought we'll further validate this by other approaches. The next approach, what we thought would be to see, there are one of the complexes is PTPIP is on the ER, the mitochondrial membrane, and WAP is on the ER membrane. So we thought we'll just immunoprecipitate PTPIP and look at the extent of WAP that is coming in the immunoprecipitate as a indication of what is the extent of interaction between these two organelles. Now, we did the immunoprecipitation. You can see the input here in the control IP. There is no non-specific band. You can see the PTPIP getting immunoprecipitated, and this is control and NUP358 condition. You can see an increased WAP-B interaction. Consistent with that, in the absence of NUP358, maybe the PTPIP and then WAP-B making better interaction. Or there's an increase in the ER mitochondrial contact sites when NUP358 is not present. Now, another approach is something called proximity ligation assay, wherein if you have two different antibodies for two proteins, and these antibodies bind to the respective target, and the secondary antibody can bind to them, and the secondary antibodies, if they are already tagged with an oligonucleotide, both of them, and you add another set of single-stranded DNA, which would have partial complementarity with this, you can always ligate them, and once they ligate it, it depends on the proximity, then only they will get ligated. And because of the 3' end label and is available, you can add labeled nucleotides, which will actually amplify this region further, and you will see them as fluorescent dots. So what it means is that if they are close enough, they can be annealed, they can be ligated, and the product can be seen. So the puncta formation after this entire reaction is an indication of the in situ protein interaction inside the cell. So if you look at the number of puncta, for example, in the control saRNA, if you do it for WAP-B and PTPIP, these are two proteins of ER mitochondrial contact site complex, you can see again there's an increase in the number of puncta, indicating that ER mitochondrial contact is increased when NUP358 is depleted. Now, the question is that, yes, if NUP358 depletion is causing an increase in ER mitochondrial contact, how does it happen? So, see, when we went back into the literature, and it was shown that, during growth factor signaling, mTOR complex gets activated. There are two complexes. The second complex, which is the mTOR complex 2 and AKT, this signaling actually is important for the ER mitochondrial contact sites stability. That's what was the study about. So what they showed was actually mTOR C2, this complex localizes to the ER mitochondrial contact sites. They also phosphorylate. The AKT can phosphorylate IP3R and other proteins which are involved in ER mitochondrial contact sites, and that's how they stabilize ER mitochondria. So the question is that in the absence of NUP358, if there is an increase in stability or increase in ER mitochondrial contact sites, whether it is through activation of mTORC2? That was the question we wanted to address. Now, so to look at that, in other words, if you deplete NUC258, is there an increase in the mTORC2 activation? So interestingly, that's what we found. If you deplete NUC258, in the depletion, you can see it in the siRNA cell, you can see there's an increase in the AKT473 phosphorylation, which is an indication or is an indicator of the mTORC2 activation that is increased in NUC258 depleted condition. And we also used a CRISPR knockout line, which was made by Rimpi, a talented PhD student in the lab. And in that knockout cell line also, you could see that there is an increase in the phosphorylation of AKT, indicating that in the absence of NUC258, there is a hyperactivation of mTORC2 AKT signaling. Now, so if that is the case, one interesting thing is that, that study is about mTORC2 is actually activated, is known on a growth factor dependent manner. But in the field, it is quite puzzling where exactly the mTORC2 AKT is activated, because the mTORC2 has been found to be present in multiple regions within the cell. For example, it is shown to be present on plasma membrane, it's on endosomes, it has been found to be present on mitochondria. So these fractionation studies suggested that they are present in multiple structures within the cell. Now, where exactly is activated? So the paper, what I refer to in PNAs, they have shown that mTORC2 can localize to the ER mitochondrial contact sites. Now, the question is that whether the localization of the mTORC2 AKT to the ER mitochondrial contact sites is important for it to get activated, or in other words, whether ER MCS in general is important, or any proteins which are present in the ER mitochondrial contact sites contribute to the activation of mTORC2 during the growth factor signaling. So the approach is simple. We have different complexes which actually are important for the ER mitochondrial contact site stability. Now we tried depleting each components and then tried looking at what happens to the mTORC2 activation during growth factor signaling. Here we have used insulin. In the absence and presence of insulin, you can see that the phosphorylation of AKT occurs in the control condition and in different depleted conditions. That is, there are depletion of different proteins of ER mitochondrial contact sites. Interestingly, what you would find that, particularly the VAP-PTPIP complex, if you deplete, it appears to be affecting the AKT activation considerably and remarkably. So somehow indicating that it's not the ER mitochondrial contact sites per se, but probably the VAP-PTPIP complex is specifically important for activation of mTORC2 complex. Now, if that is the case, we were thinking, how does it happen? And it's also not known how mTORC2 complex actually localizes to the ER mitochondrial contact sites. Now, we thought because VAB and PDPAP, they are involved in the activation of mTORC2, we thought whether there's a physical interaction between VAB, PDPAP, and then the mTOR complex. And in the co-immunoprecipitation, that's what we find. In the control IgG, you can see this particular specific band is not there, which you can be seen very nicely. The PTPAP and the VAB immunoprecipitated with mTOR complex. This was again validated by PLA in situ protein-protein interaction assays using mTOR and VAB. You can see the punctures are present in conditions where we have used both the antibodies. These are single antibodies are used as control, where you don't see any kind of puncta, indicating the specificity of it. Now, what it means is that, yes, VAB PDPAP somehow seems to be important for recruiting possibly and activating the mTORC2 in the ER mitochondrial contact sites. Now, whether, as we already know that, NUC258 is having a negative role in this case, right? That is inactivation of mTORC2, as well as probably ER mitochondrial contact sites. So we wanted to ask, what is the connection between NUC258 and VAB PTPAP? So, and whether NUC258 mediated activation of mTORC2 or AKT, is it dependent on VAB and PTPAP? So we co-depleted VAB and PTPAP independently, and you can see that the hyperactivation of mTORC2 indicated by AKT phosphorylation is actually compromised in the absence of VAB or PTPAP, indicating that these are important for the the activation of mTORC2 under NUC250 depleted condition. And we also found that in co-immunoprecipitation that NUC258 also interacts with these two proteins in the ER mitochondrial contact sites, again validated by protein ligation assay, proximity ligation assays, indicating that the annulate lamellae associated NUC258 is also interacting with the PTPAP and VAB. So what does it essentially mean? It means that there is a complex in the ER mitochondrial contact site, PTPIP from mitochondrial side, VAB from the ER side. It appears that both mTORC2 as well as NUP58 can interact with VAB and PTPIP. And our studies also already showed that NUP58 negatively regulates mTORC2. Now, it appears that there is a tug of war or kind of a wrestling going on for the interaction between NUP58 and then mTORC2, possibly because one is negatively regulating the other. So, what scenario we would be able to look at this? We thought during growth factor signaling, mTORC2 is activated, it might get recruited to the ER mitochondrial contact sites, increasing the AKT phosphorylation and increasing the ER mitochondrial contact sites. Maybe when the growth factor is not there, that is when NUP58 would be playing a role in negatively regulating mTORC2 at the ER mitochondrial contact sites and reducing the AKT activation as well as the ER mitochondrial contact sites. So to validate this, what we did was actually to look at insulin signaling, we thought. So what we predicted that in the absence of insulin, we would expect the NUP58 to be having better interaction with VAB, indicating this interaction favored. And in the presence of insulin, somehow the interaction between VAB and the NUP58 or this complex and NUP58 would be less. So to test that again, we resorted to the PLA technique, wherein we have looked at the interaction between VAB and NUP58. And this interaction, you can see that the number of puncta, which was quantified here, that in the absence of insulin, you would see more of NUP58 and then VAB interaction as compared to when the insulin is added, indicating that there's a kind of a reciprocal relationship between the mTOR activation and the NUP58 interaction with the VAB, PTPIP, the ER mitochondrial contact sites. So that brings us to the working model, wherein what we think is that when there is a growth factor signaling, the mTORC2 complex gets associated with this VAB, PTPIP in the ER mitochondrial contact sites, leading to AKT phosphorylation and stabilization of ER mitochondrial contact sites. And when there is a no growth factor, NUP58 would preferably bind to the VAB, PTPIP, somehow dislodging the mTORC2 from the complex and reducing the ER mitochondrial contact. So in other words, there's a kind of a fine-tuning of the action of NUP58 and then mTORC2 complex at the ER mitochondrial contact site, particularly through VAB, PTPIP, seems to be important for the actual growth factor signaling, and the regulation of ER mitochondrial contact sites. So that's where I would like to stop. It was a very small story, but I thought I'll just, given the time limit, I would just present this. Thank you very much for listening to me. Any questions, I would be happy to answer.
Thank you, Joe, for an excellent presentation of showing the NUP358, which is a nucleoprotein involved in complex ER mitochondrial complex. So anybody has any questions, please.
Jaman.
Yes.
So did you able to physically able to figure out whether the ER and mitochondria is in close chemistry by using any synthetic amino acid for cross-linking other than the co-immunoprecipitation?
So we have used PLA co-immunoprecipitation and also looked at by fluorescent microscopy. We are also doing that by EM. So we would be confirming that, but we have not looked at using the other method what you have mentioned.
Yeah, the second question is because mitochondrial structure is highly dynamic in nature. Is a particular stage of cells growth or some signaling only this kind of happen other than the basal? What is the fraction of mitochondria maybe in the contact site normally?
We have not quantitated it. We have only looked at the relative interaction between mitochondria and ER during the growth factor signaling, and that seems to be dependent on NUP58 at the annulate level. But we have not looked at the other signal, which might have other functions. Thank you very much.
Very colorful talk, German.
Thank you.
So depletion of NUP358 leads to more ER mitochondria. I didn't quite understand, how does it affect the cell physiology? Does it affect mitochondrial function? Does it affect, can you speculate a little bit on that?
Right. So I have not alluded to that because of the lack of time. Actually, what happens is that there's a close contact. It means that, ER mitochondria, they're highly dynamic structures. Here I was trying to depict as a kind of a static structure where it may be having more contacts or less contacts. It's actually, it's quite dynamic. At a given time, I think it's more stabilized or less stabilized is a kind of relative indication that the dynamicity is affected. Now, in the absence of NUP258, we think the mitochondria is having less calcium. And we have also indication to say that calcium is not reaching mitochondria, even in spite of having close interaction between ER and mitochondria. And calcium gets leaked into the cytoplasm. And that's another story which I have not told here. But it appears that it's also regulating cytoplasmic calcium and thereby other processes such as autophagy. Now, we are still trying to get into the details of it because it was more interesting for us to first of all verify and then make sure that it is really happening. Yes.
Okay. If there are no more questions, I would like to thank Dr. Joman Joseph for an excellent, fantastic presentation. Wish you all the best.
Thank you. Thank you so much. Thank you so much.
So that brings us to the end of this session. I'm sorry it got delayed because of a little bit of time gap, but all the 4 talks were excellent and very informative. I thank all the 4 speakers for their excellent presentation. Thank you.
I'd like to thank all the speakers of this session for the very interesting talks. And I'd also like to extend my thanks to Professor Savitri for chairing this session. Now I'd like to welcome Professor Nagasuma Chandra, who will be chairing the Centenary lecture today. Professor Chandra is a professor in our department and Associate Dean of the undergraduate program in IISc. She is well known for her studies on computational biology and structural bioinformatics. We welcome you, ma'am.
Thank you very much, Kshitija, and thank Professor D.N. Rao and Rangarajan for asking me to chair this session. It's a great honor indeed for me to chair this session where Professor Tom Blundell has kindly agreed to deliver a centenary talk. And personally, it's a great privilege to welcome Tom, who's been a mentor and a great inspiration to me. Professor Sir Tom Blundell, who's led the Department of Biochemistry at the University of Cambridge and has served as a Sir William Dunn Professor of Biochemistry, has been a long-term friend and well-wisher of our institute and very well known to many people in the audience. He's made tremendous contributions to structural biology, solving 3-dimensional structures of a number of proteins and complexes. And he was one of the few people who saw it very early, the tremendous value of computational biology and bioinformatics, and has been championing algorithm development and numerous applications to drug discovery. He's co-founded drug discovery companies, which has taken several molecules to clinical trials. And Professor Blundell has received numerous awards, medals, and prizes, which include a knighthood, election to the elected as a Fellow of the Royal Society, a Fellow of the Academy of Medical Sciences, a member of the European Molecular Biology Organization, elected as a foreign member of the Indian National Science Academy, the Chilean Academy, and the World Academy of Sciences, and many, many more. He's led prominent research councils in UK and has also been highly committed to equality and diversity. And his laboratory is a great example of that, which has been truly global. He's been a close friend and a long-term associate of Professor Vijayan, which I hear dates back to the time they both spent in Dorothy Hodgkin's laboratory at the University of Oxford. Vijayan has sent his best wishes to Tom, and on this centenary occasion has sent a short message to the biochemistry department. If I can take a minute, Tom, I'll read out Vijayan's message as he's not able to be here personally due to his health. The message from Vijayan reads like this. When I joined the physics department of the institute in 1963, each department was almost a silo. I, however, socially knew fellow students like G. Padmanabhan, who was a couple of years senior to me. In 1971, when I rejoined the institute after a postdoctoral stint with Dorothy Hodgkin, interdepartmental interaction had become the norm, and my earliest interaction with the biochemistry department was in connection with its Golden Jubilee celebrations. At the instance of T.M. Jacob and Joseph Cheryl, I made a model of DNA for the exhibition, which was the first 3-dimensional model of a biomolecule made in Bangalore. I then made a 3-dimensional model of T4 bacteriophage for Joseph Padiati, and then of several proteins, and in a way, together with the Department of Biochemistry, brought 3-dimensionality to Institute Biology. From these early efforts, my interactions with the department grew manyfold over the years, especially in my roles as a divisional chairman and associate director. The achievements of the Department of Biochemistry have been legendary, before which I bow with humility and reverence my very best wishes for the success of the centenary celebrations. May the department grow from strength to strength. Ladies and gentlemen, that was the message from Professor Vijayan, who could not join us in this meeting. He's been a very close associate of Tom. May I now invite Professor Tom Blundell to take over the stage. Professor Tom Blundell, please.
Yes, I have to get it full screen.
Yeah, and I think the share screen button will help you share the presentation. The green button below, yeah.
This one?
Yes,
yeah.
yes. Yeah, perfect.
Okay. I don't seem to be able to get back, but this is not working, I'm afraid. Okay. How do I get back? Yes, okay. Right. Apologies for that, but many congratulations to all of you in Bangalore, especially in the Department of Biochemistry, on your centenary. It's a fantastic achievement. I should say, of course, that I only can celebrate directly 50 years, so half a century since I first came to Bangalore and found that it was going to be one of my favorite places in India. And as you can see from this slide, I've had many contacts from Siv Ramaseshan, Vijayan, Saudamini, Srinivasan, and of course Suma, yourself. This has been a fantastic time to have an international interaction. So what I'm going to discuss today is a personal history, but it's going to range from human genomes and some mention of cancer to mycobacterial genomes, which I've studied quite extensively. And I will talk about structural biology, of course, but I'll focus quite a lot on the resolution revolution that's come with cryo-EM, and especially its value in multi-protein targets. What you see in this picture, and I'll talk about it more later, is in fact a multi-component system which is involved in DNA repair. It would have been very difficult to get this crystallized. It has 6 or so different components, and it's a pseudodimer, and it's absolutely critical to the way that DNA is recruited when it has broken ends to be repaired through non-homologous end joining. So I talk about the resolution revolution in cryo-EM, but that's a recent revolution. I'll also mention the work that we've been doing using all the techniques that we've used on both human genomes and microbacterial ones to study the SARS-CoV-2 proteome. And we published just one paper on the database there using the variety of techniques, computational, that we've developed over the years. And then I'll move to our approach to drug candidates and say a little bit not only about how my company, which is no longer my company, we sold it for 886 million several years ago, but they still keep me on the board and I'm still chairing the Science Advisory Board. I'll tell how we've used this information over now more than 20 years using small fragments to make drugs. And as Suma said, we've have 2 drugs on the market, one selling for $500 million a year for breast cancer. So I'll also just mention a little bit how the revolutionary effects of cryo-EM are paralleled in the computational side by AI, ML. So machine learning has taken over my computational lab, and almost everything we've done using statistical methods, we're now using AI, ML. So this is about 2 concurrent revolutions that have affected our work and most people's work in these areas. The cryo-EM taking over from X-rays, and AI, ML taking over from statistical analyses. I should say I've not given up X-ray and statistical analyses, but most of the emphasis, even on looking at emerging resistance and so on, are in AI, ML, although the statistical programs are still used widely. So that's the background, but my problem of being in the game for over 50 years is that I tend to look at it historically. So let me go on this date in the beginning of 2021, back quite a few years. I'll be mentioning the work of all these Indian scientists later, but go back more than the time that I've been involved in it, to the key work of J.D. Bernal and his amazing science that attracted Dorothy Hodgkin when she was just finishing her first degree, to move from Oxford to Cambridge. I made the same change eventually, and to work with Bernal to discover that you needed to keep crystals wet, and then you could get a diffraction pattern, and then to write to Nature in 1932, just to say that it's now clear that we can find the structure of all crystalline proteins. What we all know, of course, was that was in 1934, and it was going to be more or less another 2 decades before the first structure came, and another 3 decades before they started to come in large numbers. But that was an inspirational development, one that I was aware of because I'd read J.D. Bernal on his historical work as well as his scientific. And I became aware of Dorothy Hodgkin and her studies on insulin crystals. And I rushed when I was an undergraduate, not in 1934, but in 1964, so 30, well, just before, I think it was 62 actually, but nearly 30 years later, into Dorothy's laboratory just to do an extra course, and discovered her and her amazing group, and decided that when I would finish my undergraduate years in 1964, I would join her. Now, the amazing thing about Dorothy's group, which I realized in the early 60s, and particularly when I joined in 1964, that it was totally multidisciplinary. It was multi-cultural, people from all over the world. And there you see Siv Ramaseshan, who was in the lab in 1964, I think it was 64, when I joined. And I really made contact with him. He was my first contact with India, and a huge inspiration in the year that he spent with Dorothy Hodgkin. But also we had people from other parts of the world. And this, no doubt, was one of the features that attracted Vijayan to the laboratory of Dorothy Hodgkin. He arrived in 1968, I'd actually taken a few years off to do my PhD in basic crystallography and computing, so I knew about the techniques. And I joined the insulin group in 1967 when Guy Dodson and Eleanor Dodson were really pushing it forward, and Margaret Adams was just finishing her PhD. So Vijayan and I really began to contribute in the key time before we solved the structure of insulin. Another person was Ted Baker, who joined us in 1968. So this was part of the community, and you can see Vijayan sitting at the front here with Thomas Hodgkin, Dorothy's husband, and a person who traveled the world widely. And there is Dorothy Hodgkin talking to Tony North. And of course, Dorothy Hodgkin features here in the middle. So Dorothy was the inspiration of us all. And we soon realized that what we were doing on the insulin project had many features. First, it had beauty. And I was brought up in a family that focused on art and music. And I'd done a pre-university course in art. And I immediately saw that science was beautiful, that the 12 apostles in the Chartres Cathedral window had things in common with the 6 subunits of insulin. I also not only thought of beauty, but I thought about function. And the crystals of insulin that we were working on, they are rhombohedral, and they've been used in the pharmaceutical industry to treat diabetics for many years, in fact, since 1934 or around that time. So even 30 years before this was going on. And what we could see was that the work that was evolving on the granules of insulin, which are actually in the pancreas, you could see that in fact, the crystals that we were using in the lab were related to the storage form, the granules of insulin, which are very important. And these very tiny crystals were important on keeping the insulin in circulation. And of course, everybody mimicked that when they came to use insulin in treatment of diabetics. They used minor crystals. And here are the people, which is the third aspect of science for me: beauty, function, and people. You see on the top myself looking a little bit more beautiful than I do now, talking to Dorothy Hodgkin. But below you see a very excited Guy Dodson. And I'm not quite sure what he's doing to Vijayan, but seems to be coaching him in a paper at a meeting. And you see behind the advertisement, and I think that's Kalyani at in the background. So this work on insulin was inspirational to somebody who had not intended to be a scientist. I thought I was going to be involved in politics and other aspects to think about going forward. But I did get involved in politics. I was in Oxford, and what I suddenly realized was that there was a plan to build a motorway following those red arrows through the middle of Oxford, through Christchurch Meadow, the beautiful meadow next to the river that goes through Oxford, the Thames River, and then arriving right next to my house. And I became motivated. Why did they need to build a motorway through one of the most beautiful cities in the world? And I became politically motivated. I became active in the city And as a result of my activities, and I was elected eventually in 1970 to the Oxford City Council. It took another couple of years before we got complete control of Oxford, and I could begin to think about saving some of the very beautiful houses and other things. So in 1972, the Labour Party that I was a member of took over the city of Oxford, and I took over being chair of the planning committee. And the first thing that I did was to stop the motorway. So that was amazing to be able to do that in parallel to doing science where we had the structure of insulin. So I was in a real dilemma. What should I do with my life? This is a personal history. I stopped the motorway. I pedestrianised the centre of the city of Oxford. I made North Oxford a conservation area against everybody's advice, because there were houses like the one on the top left, which were Victorian and not hundreds and hundreds of years old, like many of the university buildings below. And I introduced bus lanes. So I changed, and these things have never been reversed in the city of Oxford. But nevertheless, I felt I was really interested in science. So what should I do? There was a huge problem, and I decided just to travel and to find out about the world before I made the decision. Should I become a politician, or should I be a scientist, or could I be both? And I set off to Moscow, where I'd already been in 1966 for the International Congress of Crystallography with Dorothy Hodgkin. And I then got the train from Moscow, and for 7 days, I traveled across the Trans-Siberian from Moscow to Khabarovsk on the Trans-Siberian Railway. When I got there, having passed through amazing tunnels and incredible countryside, 7 days on the train, a very long journey, I then had the problem is, how did I get from that part of Russia in Siberia to Japan? And amazing thing happened. I met a very incredible Japanese person called Reiko Sakama, and she had a plane which was going to leave the eastern Siberia, where she'd been trying to make amends for the Japanese Russian war earlier on. And I went with her to Japan. And then I traveled down from Japan to Hong Kong, where I have friends. And then, of course, experienced music. I was a jazz musician, but interested in multiple forms of music. And then I listened to Chinese opera, and then I traveled on to India and eventually down to Bangalore. And that was the way, not the straightest way, the most direct way, that I first arrived in Bangalore in, I think, 1972. And I was aware that Dorothy Hodgkin, of course, had contacts in Bangalore. So though I've been to Calcutta and to Delhi and other cities in India, Bangalore was where I wanted to go. And of course, Vijayan and Kalyani had already returned. Siv Ramaseshan and his wife, Kausani, pictured here, were there. And so that was a great incentive to go. And so this was my first visit to Bangalore in 1972, not quite 50 years ago. And that's myself and Vijayan just as I was leaving by train to travel around India. So my first connections. But the other connection with India in that period in 1972 was the music. While I was in India in 1972, I listened to the Veena. I learnt about Carnatic music, and I started to study the Veena. And I met a man called Krishna Raghavendra eventually, who'd been in your area. And I had a few lessons on the Veena. I then carried it all the way back to England and started taking lessons. So these were my first connections with India. And it was through people, it was through culture, it was through music. Wonderful. But I had to get back uh, to um, to Oxford at the time, and then I wondered, what shall I do? Shall I continue in science? If so, where shall I go? Or shall I go into music? Or should I do something traveling the world? And I decided that I'd stay in science. I went um, on Dorothy's advice and left Oxford to set up my own group. And this is Sussex. This is the Sussex Downs where I had been brought up, a very beautiful countryside. And I was given a position in 1973 in Sussex University. And I just gave up politics. I gave up running a modern jazz group, and I focused on the science. It was the most productive period in my life. I first had a paper in Nature on glucagon. I had a paper in Nature on the insulin function and interactions, which is doing a collaboration with chemists to work out exactly what was the area that interacted with the receptor. And I also began to think about evolution under the influence of John Maynard Smith, who is an incredible scientist interested in evolution and a great influence on me. And so I started thinking about insulins. Were the changes that we saw in insulin between different species, were they Darwinian? In other words, were they selective or were they neutral? And I think this was a very lucky position because with insulin, we had the structure, and unlike in most proteins, we had lots of sequences. And so I was able to begin to think about the work of Moto Kimura, who've been thinking about evolution in the context of insulin. And I wrote this paper in Nature about selective and neutral mutations, and focusing on Darwinian aspects, but also emphasizing that not all changes in proteins are functional. And that was published in Nature as well. So I was very lucky. In 3 years, I had 3 main articles in Nature, and I was offered the chair of the Department of Crystallography, which had 100 people in Birkbeck College. I'd also written with the help of Louise Johnson and very much influenced by Eleanor Dodson's work on insulin, which hadn't really been published, to write a book on protein crystallography. So that's the other thing I really did in Sussex before moving to Birkbeck. And Birkbeck was of interest to me because it was the place where Dorothy's mentor had moved when he left Cambridge in 1938. And it was a multicultural place where people like Erin Klug moved, Rosalind Franklin was there, and very amazing places with music, both classical and jazz around. And there I began to think of what I could do. So I thought about insulin. I had lots of connections with industry through the insulin because, as Vijayan will no doubt have told you, we interacted with the companies to get our insulin samples. So I wondered what I could do which was useful and basic science. I have my papers in Nature. What can I also do? And I thought about lowering blood pressure because that was related to an enzyme called renin, where I defined the structure. That was another Nature paper a little bit later. And I began to think about that. So the first thing was to understand the renin family of aspartic proteinases. And I looked at the binding pockets. I looked at the hydrogen bond systems to understand functionality. And then I started looking at the pepsin itself. And I thought, this is strange. The two halves of the pepsin are almost identical in fold. Nobody had noticed that, even though we've been looking at the structures for some time. And I predicted that there was an ancestor like this in 1978 in a paper in Nature. And this, everybody, in fact, Max Prutz told me I really shouldn't write any more papers like that. They were pure speculation and I should do experiments. But I kept looking for 6 years and I eventually found this molecule, or one that was very similar to my predicted ancestor. So it was published. That was the evidence that I had. But in 1984, I realized that I could find that protein in the HIV genome. And if I could find it in the HIV genome, I could predict its structure. And we had the HIV epidemic. So if I could predict its structure, I could start thinking about targeting it and making drugs against HIV, which is what I did. But I didn't publish my speculations. I did the experiments. It took me a few years to get the crystal structure. Alex Vladova, an old friend, even we've been talking today on the email many years later, did the same thing. And we were able to predict how drugs would bind. And this did quite a lot, though it was done in companies, to get the first drugs against HIV. So I realized that drug discovery could be aided by structure guiding, but I also realized that you might get resistance. So this was the basis. Now it's 1989, 30 years ago, and I wondered what I should do. Well, first, I obviously needed to think more about going from genes to proteins to make predictions. And I also ought to think about going from proteins to drugs. And so both of these I began to focus on. I should say I got a bit distracted because Margaret Thatcher, even though I was a left-wing Labour councillor, invited me to advise her in number 10 Downing Street. But I still worked on this and also the problem of chemical space screening of chemical libraries. So the first thing is we had to use structural biology to define the biological space. And so let me just give you one area where I've continued to do that. So this is really building on the structural work that we've done much earlier, but it's focusing on DNA repair. And I knew that it was an area which I could find targets for cancer. And so I looked at this system, which is called non-homologous end joining, and I started working on it just over 20 years. It has three main steps. One is the synapses, bringing the two ends together, then end processing, and then end ligation. And these processing with an enzyme called Artemis, and then here is the ligation. So I spent around 20 years looking at this system, looking at different components, using small angle X-ray scattering, doing computational predictions. but also doing classical X-ray work, which Lynn Savander and Dima Chigatsa helped in. And that was published in Nature and then in Science as we got higher resolution. But I realized that cryo-EM was going to be useful, and that was because of the work of Helen Sable that I'd recruited to Birkbeck, and also, of course, Richard Henderson. And so we did the cryo-EM of this protein, and of course, lots of other nanospray mass spec and other methods. I just want to show you what's happened over the last 2 or 3 years on the cryo-EM, which has really been amazing. So the person who over the last 2 years has done most of the driving of this is Amanda Chaplin, who's a postdoc in my lab for the last 3 years. And she, with the help of Steve Hardwick, has got a very high resolution. So the highest resolution I got of the DNA PK CS, which has 4,000 amino acids, was 4.3 angstrom. And using cryo-EM, we've been able to get 2.8 angstrom. I never believed, really, when we started that the resolution revolution in cryo-EM would allow me to get better resolution than I could with X-rays. But here's the evidence. This huge molecule with 4,000 amino acids interacting with 5 other components. We've defined the structure of it, published partly in Nature Structural Biology, but now we have sent to Nature some higher order structures as the DNA and the other components interact with this structure, all defined by cryo-EM, and also forming dimers and higher complexes that we've been able to explain the roles of in DNA repair. So this is a multi-component system, 10 or so components I've got in this structure that we've just defined now, which we call a super complex. All of these components, XRCC4, ligase 4, with the DNA PK. And this is the work that we have that I mentioned in the introduction. We've just submitted this to Nature. It's a huge complex, which we couldn't have got with X-rays, but it tells you how the components like XRCC4 and XLF assemble with COO, which recognizes the DNA, and the DNA PK, which is the general center of the DNA repair mechanism, but is a kinase and activates other components. So here we have this now at 2.8 angstrom resolution, and we can look at some amazing consequences of this complexity. So this is where, for me, the structural biology has gone. With the help of all these great scientists I have in my lab, we still have to think about changes in time and not space. So I've done quite a lot of work using magnetic beads and magnetic traps to look at these processes over time. But these are important. But how, with 20,000 genes in the human genome and 4,000 in pathogens like Mycobacterium tuberculosis, do we actually get the structures? Well, we have to do something computational. And so I realized that the software that I developed in the early 80s, late 70s, some of it, could be used then to generate proteins for the whole of the genome, the whole of the human proteome, the whole of the proteome of pathogens. And this was going to be useful in selecting drug targets. So we wrote some software, quite a lot of it published around the 1990 period. The most important software we wrote was in 1991. It didn't get published until 1993, but with this genius of a student called André Chaly, who was in my lab. This single paper has actually it's 12,400 citations now, and some other software developed to do sequence structure homology recognition has got over 1,400 citations. So we use these two software with thousands of other people using it to develop a database, not only for the human proteome, but for Mycobacterium. And I just note here that Saudamini and Srinivasan were thinking along the same lines, and they involved us in their paper on the database of the interactome of Mycobacterium tuberculosis. So my Indian collaboration was continuing. And then we developed software for other mycobacteria. This is the one that's involved in cystic fibrosis. And we also, with another Indian, got involved in leprosy. So Sandeep Chaitanya, you see his picture here, he was in fact head of the, well, he's still head of research in the leprosy mission, but he came from India and joined my laboratory about 3 years ago. And we're now the center of leprosy research. So we've done the same thing with the proteome of leprosy, which is much smaller and much of it's not used, as the leprosy or the Mycobacterium leprae is dependent on the human proteome as well, or genome. And then, of course, we've done all the similar things in the last year on the SARS-CoV-2. We've modeled all the components. We've analyzed the oligomeric structures, which I think most people didn't do. We've looked at binding prediction of small molecules. We've done mutation analysis and docked in various candidate drugs. So we've used this with a big database, which is publicly available and published, to understand the proteome of SARS-CoV-2. And if you've done it on the human, you've done it on mycobacteria, it was very straightforward to get it published pretty quickly when we found about CoV-2. And of course, we found cryo-EM on CoV-2 components. This is just a beautiful 2.4 angstrom cryo-EM structure of the SARS-CoV-2 NSP15 protein. And at the same time, we've been using the other revolutionary thing, which is AI, and writing lots of software using machine learning. I haven't time to go through that, but that's been led by a brilliant computer scientist called Aryan Jamas, who started off as a biologist, and we've had some amazing developments now, rewriting everything we've done in statistical form in a machine learning form. So these have been very exciting times. So we've, of course, made a database of the cancer gene census, all the proteins, 700 of them. Only 400 of them have parts of them experimentally defined, and we've modeled that as well. So lots of things going on, all the way from cancer through to mycobacteria to the SARS virus. And we've taken the work that we've developed on structure-guided drug discovery, which we developed in Aztecs, and you see on the top right-hand side here, small fragments. So you only need around 1,000 of those, or even less, to cover the chemical space that you would be with simply billions almost, certainly multiple tens of millions of drug-like molecules. And we formed a company with another Indian. This is Haran Jyoti, who was my student in the 1980s. And he came back having worked in various pharmaceutical companies. And in 1999, we founded this company called Aztecs with a chemist called Chris Abel, who very sadly died very recently. And we set off the company to use this method to define new drugs. We had 3 people in the lab to begin with, and Harren had a room over the local shopping center. But soon we showed that the fragment-based method would work. We raised multiple millions, built a big lab, and over the subsequent years, we got a lot of drugs into clinical trials. We now have 2 on the market, I'll say about that, but we realized that we needed more investment. And so we sold the company for 886 million. I should say it's capitalism. So the founders got almost nothing as we borrowed a lot of money, and the investors took almost all of the 886 million. But our pleasure was in doing something useful. So in 2016, our first drug got FDA filing, and through Novartis collaboration for advanced breast cancer. And that's the one I was mentioning earlier on. And that's the one selling a lot of money, 500 million. And then a second drug in 2019 is now on the market, and others are on their way. So that was just, if you have a good idea, there's not everything you can do in academia. Some of it you have to do in industry. But we could then use those methods proved for cancer in neglected diseases. And that's what I've been doing quite a lot over the last years, taking tuberculosis. Bill Gates actually came to the lab, and I had dinner with him and his wife, Melinda Gates, and we began to collaborate on tuberculosis. And I realized that this was particularly important to us because it was affecting the southern part of Africa, where my family comes, or my wife's family. So here is my wife, who's a famous scientist, Lynn Sibanda. Here are my 2 daughters, who are now, one's a medical doctor on the right, the other one's making a lot of money in finance area. We were doing okay, but back in Zimbabwe, our great grand nephew and niece and others were facing the spread of the virus. So of tuberculosis, sorry. So we started working on tuberculosis, and we did screening. You can see here how much I've been dependent on Indians, such in Surade, Pooja Gupta. Pooja I met actually when she helped me get back from the north of India to Delhi to catch my plane. And then she turned up and has been working in UK very successfully. So we used the biophysics fragment-based discovery using thermal shift, SPR, and other methods to find fragments that bind to TB. And then we use thermal shift and ITC and NMR and X-ray to get the structures and the binding and the kinetics. And all of this attracted no other than Bill Gates, who already was funding us. He telephoned me from the USA and said that he wanted to come and spend a day in the lab, but I wasn't to tell anyone else. But you can see that one photographer found out about it. We just picked him up from a car on his own. That's Chris Abel, myself, either side of him. And we made him put a lab coat on, and he spent the day in the lab. Just an amazing, amazing guy. And he was very impressed by the fact that it was multicultural and so on. And so what I said to everyone in the group, you say 2 things. One, what is your first main discipline, showing our multidisciplinarity. And secondly, say which language was your first language. And in my group, we could do 30 languages, just over 30 languages. And he was very impressed. And we had a great day and some very good collaborations with him. And we managed to do something for him. We got some fragments through two nanomolar binders. But the real problem with TB is getting them on the market. Still a solution that I've not yet found. And so that's where we are. We have been looking a lot at mutations. We've tried to use the software that's been developed in my lab. Again, another Indian here who is Aram Prasad, who I met in India, came worked in my lab, developed with others this software for statistical potential energy functions to predict the effects of mutations. We also, of course, have used machine learning methods, and we have some other software which we started to do 8 years ago. So we were using machine learning a lot before it became really popular, and have a whole suite of software for defining the impacts of mutations on ligand binding and so on. And so all of this comes together to understand not only drug resistance, but to think about designing drugs against resistant species. And there, of course, we've been looking at cryo-EM, and this is just an example of work that's just really been done. It's been accepted for publication on CAT-G, and looking at CAT-G using cryo-EM to look at the impacts of mutations experimentally. So machine learning on the one side and cryo-EM, the two revolutions in our subjects, have really come together to help us move forward. So that's my story. I think I'm more or less in time. You can see our thoughts now are, how do we repurpose old drugs? How do we fragment and regrow the drug-like molecules? Or do we get interfacial stabilizers and so on? So these are the people who've helped a cryo-EM facility team that's been amazing in our group. Lots of very good rotation and part 3, that's 4th year undergraduate students. But this is the main team. And you see there's quite a lot of Indians appearing, not only Sandeep, but Sheikh Arif and others. Asma Munir is actually from Pakistan, Pooja Gupta from India. So it's multidisciplinary, it's international, and I think it's gender balanced. That's the pleasure of working in science. And I'm very lucky to have very active, very interactive people. And thank you very much again for inviting me to give this talk. I hope that you've been tolerant of my personal history. I hope that you agree with me that although X-rays and statistical analyses are still very important, cryo-EM and AI, machine learning are going to be critical. But most of all, the message is that Indians in my laboratory have played a major role in what we've been able to produce. And I've been very impressed by what's been achieved by people who, some of whom have been in the laboratory and others I met in India have achieved. So congratulations to you on the 100 years at the Centenary of Biochemistry in Bangalore. Thank you very much for listening to me. Thank you.
Thank you very much, Tom. That was an engrossing account and taking us through the journey. It was fantastic. Could we have a couple of questions? Would that be okay?
Certainly, as long as they're not too difficult.
Okay, so we can have some questions. I think everybody is just awestruck. So let's, yeah. So if there are no pressing questions, let's thank Tom for a fantastic talk and for agreeing to give this talk, for making time for this. Thank you so very much, Tom, and I hope to see you in Bangalore soon. Hope things will get back to normal.
I'm looking forward to coming to India this summer if possible, because we're doing a tour looking at leprosy and tuberculosis and how we can learn about leprosy research from tuberculosis. And so I look forward to coming to Bangalore if the pandemic has and the problems have gone away a little bit. But thank you very much for inviting me.
Thank you so very much. Yeah, thank you. So I'll now hand it over to the organizers. Thank you.
Can I log out now? May I?
Thank you so much. Yeah.
With that excellent talk, we come to the end of the first session of today. Once again, I'd like to thank Professor Blundell for his talk and Professor Chandra for chairing this session. I'll now hand over to Humaira, who will be coordinating session 4. Over to you, Humaira.
Thank you, Kirtana. Good evening, all. Before we begin with the second session of today, I would like to request to kindly adhere to the timings. Each talk will be of 25 minutes, followed by a 5-minute question and answer session. Attendees may verbally ask the question or post their question in the Zoom chat box or YouTube comment section. Session chairs will coordinate the question and answer by calling the attendee verbally to ask the question or reading out the question themselves. I also request all our speakers to disable the attendee annotation from the More Options tab on the top right corner of your screen after sharing the screen. Now, I would like to welcome Dr. G. Subba Rao, who will chair the second session of today. Dr. G. Subba Rao is a student of Professor T. Ramasarma and graduated in 1975. He is currently CEO of DXSys Inc., which he founded in 1998. His current research activities include point-of-care diagnostics for traumatic brain injury, cardiac markers, and COVID-19. He is closely associated with the IISc Alumni Association of North America, AANA, and has been very helpful with regard to our department's centenary activities. Sir, you are most welcome to start our session 4.
Thank you for the introduction. Can everybody hear me now?
Yes.
Yes.
Okay, yeah. Thanks for the introduction. And I would like to thank Professor D.N. Rao and Professor Rangarajan for giving me the opportunity to chair this last session of the Centenary Symposium. And it happens to be the first day or the beginning of the second century of the Biochemistry Department. I was a student here when we celebrated the Golden Jubilee of the department in 1971. And now I'm here again at the Centenary celebration. Hope we can have another event in the Institute where all of us from this virtual meeting can meet and greet and in the campus and discuss the vision for the next 100 years, or at least for the next 20 or 25 years. Since we are already running late, I think we'll start the session. The first speaker is Dr. Sri Kumar Chalapan. Dr. Chalapan obtained his PhD in biochemistry from the Indian Institute of Science and did his postdoctoral training at Duke University Medical Center in North Carolina. Subsequently, he became an assistant professor in the Department of Pathology at Columbia University in New York. He joined Moffitt Cancer Center as an associate professor in 2001 and was promoted to full professor in 2006. He was recently named as chair of the new Department of Tumor Biology. He has also served on the editorial board of multiple journals, including Molecular Pharmacology and Molecular Cancer. And Dr. Chellappan's research aims to understand the mechanisms by which extracellular signals regulate the cell cycle machinery and how a loss of this regulation leads to oncogenesis. Today, he will be talking on the novel strategies to combat cancer. Now, I invite Dr. Chellappan.
Thank you very much, Dr. Subrao, for the very kind introduction. Let me share my screen. And I hope you can see the screen, I hope. So let me disable the annotation for others and spotlight. So I want to start by thanking the organizers, Dr. Rangarajan, D.N. Rao, Nagraj, Muniappa, all my former friends and colleagues in the department for giving this opportunity to make this presentation. As all of you know, the Department of Biochemistry is very close to my heart for the past, since I walked through the doors 40 years ago. So I had to take 2 minutes to thank some people and talk about how it was before I go into my talk. So as Dr. Subrao mentioned, I've been the chair of this Department of Tumor Biology for the past 10 years, since 2010. But one of my favorite and memorable achievements in the past at IAC was, I would think, I was editor of the BICS letters for at least 2 years when I was there in IAC, and I was also the secretary of the BICS for at least 1 or 2 years, which gave me an amazing opportunity to interact with many of you and with most of you, especially pretty much all my contemporaries, and they have remained friends for life. So it has been a great opportunity, and we had some fun times. So one of the funnest things was, again, to wake up people like Dr. Sheshagiri and even Utpal Tatu and many of the others and make them play cricket for the department. And our annual cricket matches were all so much fun, where Professor GP and Professor Shastri, all of us used to join once in a year occasionally. Those were great times. I mean, I really miss those days. But luckily, among doing all those things, we also managed to do some science, and I was awarded the Giri Memorial Award back in '87 for the best thesis, surprisingly. So all these things wouldn't have happened. Unfortunately, I am unable to go into the presentation mode. Yes, there it goes. So none of the things would have happened. Wouldn't have been giving this talk today, except for the mentoring provided to me by Dr. Professor T.M. Jacob, who was probably the best mentor one could ever have. He was so kind, empathetic, considerate, and extremely intelligent. And I used to tell even my students some time ago, some of the things I learned from him. He used to always say that if you do the same experiment the same way, you get the same results. So many of my former trainees used to come and say, oh, the experiment didn't work, I'm going to repeat it. So I used to tell exactly what Professor Jacob used to say. I also was lucky to work with Dr. John Evans, who was also an extremely nice person, but much more aggressive scientist. But I was so fortunate to have their training in their own ways. I should also thank so many former mentors in the department, Professor GP, Kurup, Charayal, Padati, Dr. Shastri, Ram Sharma. I mean, all of them played some role or the other in my development as a scientist. So thanks to all of you, my current and former colleagues. I should also, so those were the olden days, and things have changed quite a bit now. I am in touch with the department in many aspects. And one of the best things we look forward is interacting with Professor Gopinathan, who has been a mentor and a friend for the past 40 years. And we always have a blast when we see Dr. Gopinathan. I mean, so here are some of the alumni, Namrata Borasingal, Nupa Mahajan, Mohan, Kiran Mahajan, etc., who we all get together once in a while when Professor KPG has visited us many times. And of course, many of the alumni we meet during the AACR meeting. Here we can see Anand Surup, who is, I guess, one of our most illustrious alumni. Than Kalvanaklavu, and Chitra Manohar, and here is Durga Kumari and other people. And of course, other connections with IISc still remains is Namrata and Mohan and many others. The IISc students have come and worked in the lab, and they have done extremely, extremely well. And I'm so extremely thankful to their work as well as whatever the department has done for me for the since 1981, one way or the other. So having said that, let me start with my work. So our work, what I'm going to talk to is one aspect of what we have been doing on lung cancer. We have been working on non-small cell lung cancer, especially in the context of nicotine and tobacco smoke components. So this is an old experiment done by one of my students 15 years ago, for which she got the best thesis award from this university, where we found that if you administer nicotine to animals, the tumors become much larger, they become much more metastatic, and they also show this phenomenon of EMT, that's epithelial mesenchymal transition, which my good friend Centenary described beautifully yesterday with some fantastic images. So if you treat cells with nicotine, you get the cells the flat epithelial cells become very mesenchymal, they get detached, they can now migrate to other sites and form metastasis. So we have been trying to understand the mechanisms of what is happening here, and that's what I'll be talking about and how we can target these things for cancer therapy. So as you know, many of the proteins that drive tumor progression and metastasis or even tumor initiation are not targetable. KRAS is a typical example. Of course, now there are drugs for one specific mutation or mutant of KRAS, the KRAS G12C, that has some very nice drugs which have been recently approved for clinical trials and for certain users in certain cancers. But similarly, there are so many other proteins which have been extremely difficult to target by small molecule inhibitors. So that's what I'll be talking about, how these studies showing these tobacco smoke components. So nicotine is not a carcinogen, it is a tumor promoter. Such elements of tobacco smoke, how they promote cancers and how can you target that? What is the basic biochemistry of that and what can we do for that? So again, this is something, again, Dr. Centenary spoke about yesterday. So there are two different models of cancer initiation and progression. One is the clonal evolution model. So there are all the normal cells in the body, when they are exposed to carcinogens or environmental toxins or radiation, they undergo mutations. And the clonal evolution model essentially says pretty much.
Okay.
Did somebody ask me something? No. Okay. So any of these cells can form a tumor. So they are essentially capable of forming tumors on their own. So pretty much every cell has a potential to form a tumor. That's what the clonal evolution model says. But various studies from several labs in liquid tumors, I mean hematological malignancies, as well as studies on solid tumors from many labs, starting with Max Vicha's lab on breast cancer, showed that only a subpopulation of cells in a cancer can initiate and propagate tumors. And they called it as a cancer stem cells because their properties are stem-like cells. So the cells, they self-renew into a stem-like cell and a non-stem-like cell. So these are the differentiated cells that we hear about. And the cancer stem cells can keep on propagating tumors and keep on propagating. So as Dr. Mani mentioned yesterday, if you eliminate just the differentiated cells, which are the ones that are targeted by most anti-cancer drugs, you cannot prevent the progression and metastasis of cancers because the cancer stem cells still keep dividing and they still keep generating these differentiated cells. So these guys are very difficult to target. The reason is that they are very slow dividing cells. So most of the cell cycle inhibitors are not effective on it because these cells are not dividing very fast anyway. So the cell cycle inhibitors do not work. Another feature is that they have a lot of drug exporters on their membrane. So any of the drugs, what you give, the regular chemotherapy drugs, these stem-like cells can spit it out. So they have a lot of survival proteins. The BCL2 family members are overexpressed. They can metastasize to distal organs, and they are highly, highly drug resistant. They can also stay dormant for a very long time. For example, in breast cancer, as you know, after treatment, people get back the same cancers after 10 years, 15 years, etc., etc. And those cancers recur, have pretty much the same genotype as the original cancer that was completely cured by chemotherapy or radiation or anything like that. So people have characterized these stem-like cells that are from different organs. They can use different cell surface markers, different properties, and you can characterize these stem-like cells. So you have to eliminate essentially the cancer stem-like cells as well as the differentiated cells for complete cancer elimination and for curing cancer and to prevent their recurrence eventually. So showing some of our studies, this was done by Sandeep Singh, who is a faculty member at NABGM, who worked with me 15 years ago, was an outstanding scientist, as well as Namrata Bora Singhal, who was Anjali Karande's student, and she did really, really well in our lab. So what we found was that there is a subpopulation of cells called as site population cells. That's how we isolate them by a specific phenotype. If you take these stem-like cells, what we find is that they have multiple embryonic stem cell transcription factors, the Yamanaka factors, much higher level, OCT4, SOX2, NANOG. This is a cell surface transporter that we use as a marker for this in the stem-like cells in multiple cell lines, as well as in human tumor samples. And when you deplete one of these transcription factors, SOX2 specifically, the self-renewal or the ability of these cells to grow under stem cell conditions where they form the spheres, that is abolished. So the sphere formation is a measure of the stemness of this stem-like cells, of any of the stem-like cells. So they're grown on low adherence plate in very selective media where the normal differentiated cells die. Only stem-like cells can self-renew. And if you deplete SOX2, the cells do not divide anymore, and OCT4 and NANOG are much less involved in the division of this self-renewal of the stem-like cells from non-small cell lung cancer. Of course, non-small cell lung cancer is highly correlated with smoking, and most of the smokers harbor mutations in the KRAS or GOG. So what is the connection with nicotine here? So if you take the stem-like cells and culture them or grow them in the presence of nicotine, what you're finding is that 1 micromolar of nicotine, what is present in the bloodstream of a heavy smoker, that can significantly enhance the self-renewal of the stem-like cells. The differentiated cells do not. This is from a different cell line. It is not just nicotine. Now, e-cigarettes are very popular. At least in the US, they have been used as a nicotine supplement or as a replacement for therapy or for smoking. What we find is that if you take equivalent amount of e-cigarette liquid or e-cigarette content, it can significantly induce the self-renewal of this cancer stem-like cells. And of course, we published a couple of papers on this based on this data. We have studied the actual biochemistry of this, what are the factors contributing to it. And I'll be talking a little bit about that and how to target them. One interesting part of this cancer stem-like cells is that they can differentiate into more differentiated cancer cells and to other types of cells present in a cancer. So cancer cells are, as others mentioned yesterday, are not just a bag of cancer cell, epithelial cells that have been mutated. Cancer cells, the tumors have endothelial cells, blood vessels, cancer-associated fibroblasts, immune cells, macrophages, everything in there. So something like almost 20 years ago, Mary Hendricks and other people showed that when tumors grow, of course, the tumors require oxygen as well as nutrition to grow. When the tumors reach a critical mass of, or critical size of 1 centimeter, they require blood vessels to form, new angiogenesis to happen for them to grow. But what Mary Hendricks found was that before the actual angiogenesis takes place, a subset of the cancer cells can acquire the phenotype of the endothelial cells and start supplying nutrients and oxygen. She called it as vascular mimicry or vasculogenic mimicry. So here what we are finding is that some of the stem-like cells we isolated from the lung cancer cells, so these are pure lung cancer cells, and you put them on Matrigel and give them VEGF, they can form very nice blood vessel-like phenotype. They form this very nice network, whereas the differentiated cells do not. So this feature is called as angiogenic, is called vascular mimicry or vasculogenic mimicry. So this is one way the stem cells, unless they are eliminated, they can keep supporting the growth of tumors. So to make some, cutting back a couple of years of work, we did differential gene expression analysis of the stem-like cells, non-stem-like cells, and we found that one of the major proteins that is altered in the stem-like cells, that is upregulated in the stem-like cells, is this particular protein called YAP1. That's called a S-associated protein 1. So S-associated protein is a transcriptional co-activator. It does not bind to DNA. It does not have any enzymatic domain. It just binds to other transcription factors and enhances the transcriptional activity. Its main transcriptional targets are TEAD family of proteins. It can also bind to proteins like E2F1, which is very close to my heart, as well as HIF1 alpha and other proteins. So this is a protein that is regulated, it is downregulated in a normal cell through the hippo signaling pathway. So this pathway is, when this pathway is active, when the cells are tightly packed, the organizer is the right size, the YAP1 protein is retained in the cytoplasm and it undergoes degradation. So this happens through the LATS kinase and the protein ubiquitination mediated degradation. When this pathway is inactive, when the cells are loosely packed, when they're dividing, YAP or its close family member TAS goes into the nucleus, associates with the TIDA or other transcription factors, and induce the expression of a variety of genes involved in tumor progression and metastasis as well as cell proliferation. So this we thought was very interesting because what we found was that what has been published was that various cancers, pretty much every single cancer in the body have alterations in the YAP levels. Either YAP is amplified, it is overexpressed, or it is enriched in the nucleus in pretty much every single cancer type we examine. So we have been focusing on the non-small cell lung cancer, again, which is correlated with smoking. So we thought this is a very good protein to work on, but it has many challenges. Again, this is to show that when you look at YAP protein in non-small cell lung cancer, it is the normal lung looks like this, the squamous cell carcinoma, the adenocarcinoma, there are higher levels of YAP1 as seen by the intense staining, and higher levels of YAP definitely correlate with poor survival compared to patients with lower levels of YAP1. So this is a pretty good target if you can target this protein. But there are several challenges in targeting YAP1 because it has no enzymatic activity, it does not bind to DNA, and there are no specific pockets one can target with a small molecule. So to summarize some of the work done by Namrata Borasinghal during her first couple of years, what we found was that the YAP1 protein is highly expressed at higher levels in the stem-like cells compared to the non-stem-like cells. This happens at the protein level as well as at the mRNA level. The stem-like cells have much more YAP1 protein. And when you deplete YAP1 protein, what she found was that the self-renewal is completely reduced. And also we find that the SOX2 expression is completely gone. I will talk about that a little bit later in the next couple of slides. And what Namrata found was that the cells that are depleted of YAP1, they do not form tumors in mice or form very small tumors in mice. And if you take these tumors and these cells, and if you complement by overexpressing SOX2 in that, the tumors grow back up, strongly suggesting that the YAP-mediated regulation of SOX2 is contributing to the tumor growth. So when you deplete YAP, the tumor growth goes down, and this YAP null cells, you overexpress SOX2, the tumors come back, again providing pretty good evidence that it is a regulator of YAP1, it's a regulator of SOX2. Switching gears a little bit, coming back to the vascular mimicry and the role of YAP1 in angiogenesis. Again, some very beautiful work by Namrata that is under review in a pretty decent journal. What Namrata found was that, again, we were working on the vascular mimicry, how these cancer cells acquire the phenotype of angiogenic or the endothelial cells. And what she found was that in cancer cells, YAP1 regulates VEGF expression. So VEGF, as you know, the vascular endothelial growth factor is absolutely necessary for the formation of angiogenic vessels. And when she depleted YAP1, there was no angiogenic tubule formation. I'm not showing a huge amount of data what she has generated, But basically, the VEGF expression goes down when YAP levels are downregulated with the siRNA, shRNA, sgRNA, whichever way we looked at. So this was something relatively new. So what she also found was that YAP1 physically interacts with the HIF1 alpha, the hypoxia inducible factor. So this is a proximity ligation assay, which I guess one of the speakers mentioned a little bit earlier. So this is almost like a double immunofluorescence, but you are essentially amplifying the signal by a PCR-like method using fluorescent nucleotides, and every dot you see is a foci of interaction between HIF1 and YAP1. So under normoxic conditions, there is not much YAP1 HIF1 alpha interaction. When the cells are subjected to hypoxia, when there is low oxygen, that is 1% oxygen for 24 hours, we find there's a huge interaction of HIF1 alpha protein as well as YAP1 protein, and they physically interact with each other. And that has been shown by immunoprecipitation Western blots here. And not only that, we can do the proximity ligation assays where we measure the interaction between the proteins into tissue microarrays, not just the cultured cells. And what she found was that, of course, this interaction is elevated in the tumor cells compared to normal cells. These are the lung cancer tissue microarrays, and the results are quantified here. So this is apparently a meaningful interaction that contributes to angiogenesis as well as tumor growth. So how is HIF1 alpha regulated by hypoxia? This is something for which Bill Kalin and others got Nobel Prize 2 years ago. Under normoxic conditions, there is particular protein called prolyl hydroxylase, which hydroxylates proline residues. And under normoxic conditions, when there is sufficient amount of oxygen present, when the HIF1 alpha protein is hydroxylated on the prolines, there is this ubiquitin ligase VHL binds to it and ubiquitinates it, leading to its degradation. Under hypoxic condition, when there is no oxygen present or very little oxygen present, the PSD2 protein does not have oxygen to hydroxylate the HIF1 protein. So the HIF1 alpha levels go up, it is not degraded, it induces multiple genes, including VEGF, and you get transcription of all this protein, of the critical proteins necessary for transcription for angiogenesis. So we found that something identical happens to YAP1 as well. We found that YAP1 is prolyl hydroxylated by immunoprecipitation Western blots. So she identified the domain of YAP1 that is prolyl hydroxylated. She mutated that, and she found that when it is mutated, the hydroxylation is gone. So we know which is the residue that is prolyl hydroxylated. And by in vitro hydroxylation, as she found that it is hydroxylated and it's suppressed here. And a whole amount of experiment to show that under normoxic conditions, when PSD2 is depleted, YAP1 goes up. When VHL is depleted, YAP1 goes up. So it's essentially regulated very much like HIF1 alpha. So you can use this for targeting as well, which is a strategy we are planning to start in the lab. We haven't started that yet. You can do what they call as PROTACs, where you can link YAP1 to VHL, for example, and lead to the degradation. So those are some of the strategies what we are thinking of. So essentially what I've been trying to convince you is that YAP1 is very much important for stemness, for angiogenesis, and this is a regulation what I talked about. Under normoxic cells, PSD2 will hydroxylate YAP1, leads to degradation, whereas in hypoxic cells, VHL does not interact with YAP1. YAP1 goes into the nucleus, induces angiogenesis as well as VEGF receptors as well, promoting angiogenesis and tumor growth. So coming to the title of the talk, targeting YAP1. So some of the information what I mentioned, and YAP1, I hope I convinced you, contributes to tumor growth, contributes to angiogenesis, but it is very difficult to target. But can you develop strategies to target this protein? So that's what I'll be talking about for the next 10 to 12 minutes till my time is up. So what Namrata found was that the SOX2 enhancer and the SOX2 promoter, they have very good OCT4 binding sites. And what she found was that YAP1 can be detected on these particular sites by chip assays. And again, she showed by a whole series of experiments which we published in stem cells, showing that the physical interaction of YAP1 with OCT4 induces a SOX2 protein. And you can find OCT4 and SOX2, OCT4 and YAP1 colocalizing in the cells, colocalizing in tumors. We published all that. And she also showed that it is a WW domain of OCT4 that interacts with the PPX5 motif of of OCT4 of WW1 binding to PPX5 motif of OCT4. So these are some of the critical preliminary experiments what she did. So what she did was that she made peptides corresponding to the WW domain of YAP1. She conjugated that to a carrier peptide called penetratin. She delivered it into cells, and she found that the SOX2 expression goes down. We also found that the self-renewal goes down significantly. Essentially, what she showed was that when you prevent the binding of OCT4 to YAP1, you can prevent stemness, prevent SOX2 expression, etc. So can you use this strategy to develop new drugs? So this is one of our drug discovery project in which both Mohan Kumar Dureraj, who was Kumar Somasundaram student, and Namrata were very much involved. This was funded for 5 years by Celgene and BMS. Now BMS has taken it work in-house. Our strategy was to screen for small molecule inhibitors of the OCT4-YAP1 interaction. So YAP1, you cannot directly target it. So what we have done was that we have an in vitro screening protocol for the small molecules. We screened some libraries. We screened up to 3,000 compounds. We made a lot of mimetics of the PPX5 motif. In the in vitro binding assays, what we get low IC50, those compounds we take for a luciferase assay in a large scale screening, look at suppression of self-renewal, and conduct a whole series of biochemical assays and cell-based assays. So this has been our strategy. We did a lot of SAR studies, lead optimization, etc. So this is some of the compounds we identified just to show a snippet of our results. So this is again a proximity ligation assay. Each dot of interaction you are seeing here is a foci of, each dot is a foci of interaction of OCT4 and YAP1. So this is the control cells. And when you treat with some of our identified drugs, you can find that every single spot is gone. Whereas a control, an inactive compound, it does not disrupt the interaction. So that was for OCT4-YAP1. This is for a control protein TEAD, its main interaction partner, which binds to a different region of YAP1.
Yes. Excuse
me. So here what we are
finding
finding is that these drugs do not disrupt the binding, showing that it is highly selective. We have shown this by immunoprecipitation Western blots, double immunofluorescence. So we have some fairly highly selective compounds we have identified. And they're pretty potent, as I will show you. So this is a combined immunoprecipitation assay, what we have shown here. The compounds can reduce the binding.
Excuse me, Dr. Salapan, you have only a few minutes left. We're exceeding the time. Can you please close?
I have 5 more minutes or 6 more minutes. Yeah, I will finish by then, don't worry.
Okay.
So by chip assays, we are finding that the compounds reduce the binding of OCT4 to YAP1 on the promoters. Many of the YAP1 targets, including SOX2, is downregulated. These compounds can completely eliminate the self-renewal of the stem-like cells. I mean, these are the control compounds, and the cisplatin chemotherapy drug has no effect on the stem-like cells. Our compounds can completely eliminate that. So these are some of the values. So essentially, it can inhibit the binding in vitro, luciferase assay. It can also affect the viability of the cells very well. These are some studies on lung cancer organoids, which we use significantly in the lab for a large number of experiments. We are finding that these compounds can essentially prevent the growth of primary lung cancer organoids. So this is an experiment where we have done DIOC staining. The green organoids are live ones. Treated with our compounds, organoids are much smaller, and whatever that's left over, they are all dead organoids. So this essentially summarizes whatever I said in this context. So these compounds can essentially suppress cell proliferation. They can overcome drug resistance. I didn't show that data. They can suppress the growth of adherent independent growth organoids. They have all the features of a pretty good potential drug. But these are, as you know, in early preclinical development, they don't have very good PK/PD values yet. So we have to improve these compounds significantly. In the next 3 minutes and 30 seconds I'll just show another strategy what we have taken. So one of the proteins what we found to be overexpressed in these lung cancer cells, especially upon exposure to nicotine, was this kinase called tank binding kinase 1, TBK1. TBK1, what we found long ago, this was published in Nature Communications a couple of years ago, was that it is present on centrosomes, promotes mitosis, etc., etc. Surprisingly, we found that TBK1 physically interacts with and phosphorylates YAP1. So this is in tumor in cancer cells, tumor tissues, etc. By immunoprecipitation Western blots, by in vitro kinase assays. And we also found that if you deplete YAP1, so this was work by a talented research scientist in the lab called Bisharup Saha. What he found was that if you deplete TBK1, the YAP1 levels go up significantly in the cancer cells. And this was pretty much independent of the hippo signaling pathway. So as a kinase, TBK1 is very much involved in innate immunity. It regulates NF-kappa B function. But surprisingly, the inhibitors of TBK1 have been very ineffective as anti-cancer agents. So we believe that one of the reasons why TBK1 inhibition is not working as anti-cancer agents is because when you inhibit TBK1, YAP1 levels go up in the cells. So we have been trying to study the mechanisms by which TBK1 suppresses YAP1 and how depletion of TBK1 enhances YAP1 protein levels. Again, it happens at the level of proteins. What we found was that YAP1 is methylated on arginines by this protein called CAR1 and PRMT4. Again, very nice work by Biswarup Saha. If you inhibit this CAR1 kinase, the YAP1 level goes down. This is all happening at the level of protein stabilization, not transcription. CAR1 protein interacts with the TBK1, interacts with YAP1, and this interaction is much elevated when you deplete TBK1. Again, series of biochemical experiments showing that when you deplete these arginine methyl transferases, you can downregulate YAP1, and this interaction is much higher in the cancer cells compared to the normal lung. So this is a pretty good target, and this is a CAR1 inhibitor that is going about to enter clinical trials. So the advantage of this strategy to use CAR1 inhibitor to inhibit YAP1 or downregulate YAP1, the advantage is that there are small molecules that are close to clinical trials, whereas OC4 YAP1 is a very good, challenging, a very promising strategy, but they're still in preclinical trials. So this is a multispectral imaging of human tumor tissue showing that the regions of tumors which has higher levels of CAR1 have higher levels of YAP1 and higher levels of SOX2, suggesting that the pathway that CAR1 stabilizes YAP1 to upregulate SOX2 and promotes tumor growth is indeed valid, and perhaps the CAR1 inhibitor can be used to target that. Again, I think this is my very last data slide. Again, we are showing that when we treat with CAR1 inhibitor, YAP1 protein level goes down, the growth of lung cancer organoids go down, and essentially it's a very good strategy to combat this particular very difficult to target protein called YAP1. So I didn't show this whole bunch of data showing you can strongly inhibit cell viability, can inhibit adherence independent growth, it can prevent the expression of YAP1 target gene. So you can do a lot of things like that with these compounds, and we are taking it further, and the animal studies are going on as we speak. So I will stop here. I guess my alarm went off, so I guess I am right on time. So just want to thank the people who did the work, Mohan, Kumar, and Namrata, IAC alumni, of course. We are all having lunch here, and Biswarup Saha and others in the lab, my collaborators, Nick Lawrence, and a whole bunch of other people. And we were lucky to be funded by NCI, by multiple grants, and by DOD, and also Celgene did a huge amount of support to our ongoing drug discovery studies. I will stop here and take any questions you might have.
Thank you, Dr. Chalapan, for a very nice talk. And we won't have too much time because we already exceeded the 30-minute time. So we'll probably take one question or two questions. We cannot have too many questions because it's getting late. Yeah. Let's go with
I just have a one. I just have a one question.
Sure.
Oh, Dr. Mani, sure.
Wonderful talk. I'm just curious, do you need a HIP1 for your TBK1 and YAP1 to interact? And do you see like such an interaction happens in the hypoxic region rather than a periphery of the tumor?
So we looked at tissue microarrays. So it is very difficult to say. These are essentially punches, as you know, 1 millimeter punches. So it is very difficult to say whether it is the periphery or the middle of the tumor. We haven't done that yet. For that, probably what we should do is look at the mouse tumors, which we can look at in the lab. We haven't done that yet.
Subbarao, can I ask one little question?
Yeah, sure.
Dr. Chalapan, thank you for the very nice talk. I just have a simple question, and that is, would you comment on the homogeneity of your cancer stem cells? It looks like you started with cells which get inhibited by SOX2, but they don't have an impact on OCT. OCT4 does not respond to your siRNA. Then we end up with the OCT4-YAP1 interaction. And so are you working with the same cells all the time?
Pretty much the same cells, but you mentioned the key word there. The OCT4 is downloaded by siRNA. So I don't think that's a complete depletion of OCT4. The reason why I'm saying is that when we try to establish cell lines, which is depleted of OCT4, we just cannot get them to grow. So OCT4 is absolutely critical. So probably those cells have a residual amount of OCT4 still present there. I cannot explain it in that way. We have done that in multiple ways. The moment we try sgRNA to OCT4, the cells just don't grow anymore. So probably that is what is happening.
Thank you.
Thank you, Dr. Chalapan.
Can I ask one question quickly?
Okay.
I want to thank him.
Okay, see you soon.
Yeah, Chalapan, very wonderful talk. The only thing I was trying to figure it out is you were not able to connect with our monies. Maybe you can collaborate with the EMT. Because I'm sure probably the stem cells, everything else, probably going towards EMT. That might answer Suraj's question too. I think that's probably the EMT pathway.
I don't think they are homogeneous cells. So that is in the first place is.
I think that's a wonderful talk.
Yeah.
Oh, thank you. So if I may quickly add, so if you deplete YAP1, the EMT pathway goes down. We published that in the stem cell paper a few years ago as well. So YAP1 really contributes to EMT as well. So those features and those genes, the VEGF receptors, a whole panel of genes go down when you deplete YAP1. So yeah, so that is indeed true. Sure. Thank you so much for the questions, and once again, thanks to everyone for the opportunity to speak here. Thank you, Dr. Subrao.
Thank you, Dr. Chalapan. It was a wonderful talk, and now we will move on from cancer to a different area. The next speaker is Dr. Manzoor Bhatt. Professor Bhatt obtained his PhD from the Institute and Shiga University in Japan in 1992. During his graduate research, he worked on microbial degradation of halogenated aromatic compounds, but after his PhD, he developed more interest in neuroscience. So in order to pursue his passion, he decided to join Dr. Hugo Bellon at Baylor College of Medicine to do his postdoctoral research, where he worked on the role of neurexin in axonal insulation and cellular junctions. He subsequently joined Mount Sinai School of Medicine as assistant professor and then moved to the University of North Carolina as a tenured professor in 2003. For 9 years, when the University of Texas Health Sciences Center at San Antonio offered him the chairmanship to head the Department of Cellular and Integrative Physiology, he could not resist. He moved there. He has been there ever since. At UTHSC, his current research focuses on using genetic and biochemical methods to identify and characterize molecular complexes that are involved at the interface of axons and glial cells in Drosophila and mice, and how loss of these molecules affects conduction of nerve impulses and synaptogenesis. He will be presenting some of these aspects, including translational benefits of the basic research. Now, Dr. Bhatt. Yeah, go ahead, Dr. Bhatt.
Well, thank you. Okay, good. Well, Dr. Subarao, thank you very much for the kind introduction. Good to see you finally. We talked on the phone one time. I would like to first thank the organizers of this meeting and congratulate the current chair of the department and the former chairs as well. I would also like to thank Dr. D.N. Rao for being the key organizer and all the other colleagues who have been engaged with this process. I would like to spend some time acknowledging all the colleagues who came obviously before us and have contributed to make the Department of Biochemistry one of the places of excellence, both for training and science. And if I may, it is through this department that I benefited tremendously, both as a scientist, as a person, and I also met my wife there. So I am much grateful to the institution that has essentially trained me to the level that I have sustained science ever since. I also want to acknowledge my professors who guided me and were instrumental, especially Dr. Vedyanathan, with whom I started my PhD. And after he retired, I worked with Dr. Appaji Rao, and over the years, when I was with Dr. Vedyanathan, Dr. Rao was pretty much involved in our day-to-day affairs. It was through a chance meeting with Dr. Nozaki that I took 2 years to go to Japan and study additional biochemical and molecular methods. And after I completed my PhD, as Dr. Subbarao mentioned, I decided to move to a more genetic model system where I could use the biochemical methods I had learned and apply to the genetic methods, genetic models that I was going to start. And Dr. Hugo Bellin was tremendously a good mentor and a good generous colleague who let me take whatever I had decided to work on. So to also acknowledge colleagues who have contributed to our work over the past 2 decades. These are the current lab members and members here who are in white have worked on the fly system, Drosophila system, and those in the red have worked on the mouse system. Over the years, we have benefited from many colleagues who have collaborated with us. I want to acknowledge their help as well. Much of the work that we have done has been supported by many foundations, the National Institutes of Health over the years, and I want to acknowledge that as well. So to begin with, what is the fast node conduction and why do we need that? You can imagine you are driving a car and your visual system is looking at the road, but your foot is actually pressing the gas and the brake. And your foot does not have eyes to see, but your brain is constantly controlling it. And so essentially the nerve conduction from the brain is moving all the way to the foot, allowing your foot to coordinate what you are trying it to do. So you can imagine that this process must happen so fast that you do not have much time, and essentially occurs in milliseconds. So when a neuron decides to fire an action potential, it'll travel down the axon, and it could be next target could be another neuron, or the next target could be a muscle. So when you finally arrive at the terminal stage, that's where the function will occur. It could be cognitive function, it could be motor function. So you can imagine how this circuit then works. You can decide to move your foot, and your motor neurons will send a signal to your foot muscles, to your foot area, and that same signal will go to cerebellum. Once cerebellum decides how much correction has to be made to make it more fine, it'll send back the information to the motor cortex, and motor cortex will then send that information back to your foot. And this is how we coordinate our motor activity. But there are deficits that can happen because most of these circuits have myelinated axons. So you can imagine if a person is 5 feet 9 inches height, it will take 8 milliseconds for the action potential to go from head to toe. And you can imagine that is traveling roughly around 360 miles per hour in a myelinated axon. But if you had an unmyelinated axon doing that, it would take, it'll be only 2 miles per hour, which would be much more slower. And that would not give us the cognitive ability that we need to function properly. So this is where this talk is going to go. What happens when this nerve conduction declines or is disrupted, and what happens to the subject? So you'll see this is a person who has peripheral neuropathy and is not able to walk properly. He needs help. You can see his feet and his legs have become weaker, and he needs support on the sides to make sure he walks. Eventually, next 5 to 10 years, he will go in the wheelchair and will not be able to walk. And this person's feet and hands will eventually become like this. They will crinkle because there is no strength left in the foot muscles so that this person can walk properly. And you can also have other deficits, which can lead to either deafness or muscle atrophy. This is a beta 4 spectrum gene that mutations in this lead to children with very little muscle tone and also deafness and severe muscle atrophy. And as you can see here, the legs have no strength left. You can also have mutations in genes that I'll be talking about, which is known as contactin-associated protein. These children have all mutations in this gene, and different forms of mutations are different, either a null allele and a hypomorphic allele are both null alleles. And you'll see that some of these novel alleles can cause severe dysfunction, both motor and other neurological functions. Ultimately, these children will die within the first 2 or 3 years of their life. And if they're not maintained on life support, they will not live. So you can also have dysfunction of nerve conduction when people have multiple sclerosis, where our own immune system starts damaging the myelin sheath. And you'll see what happens with these people.
I woke up one day and I didn't have any vision in my one eye. I started with just my left cheek going numb. I awoke with a tingling in the bottom of my right foot. I had burning in my legs.
And so we can now figure out, why do all these people have these deficits? It turns out that their myelinated axons are not functioning properly. And we have been working on this for the last 20 years. And if you look at a myelinated fiber, many of you might have seen this in neuroscience textbooks. There is a neuron. This area is called axon initial segment, where the neuron finally decides to fire an action potential. And this is the myelin sheath. And these gaps are called nodes of runway. Some of you might have heard that. So if you look at this structure, it looks pretty simple. And if you now slice this axon here in this area, you'll see this is the classic picture of a myelinated axon. You have the axon here, and this is the myelin sheath. But what came as a surprise, that if we slice this axon longitudinally, that's around the nodal area and across the nodal area. And this is what we saw, that the electron microscopic picture of this area is, this would be the node of runway. Pay attention, there is no myelin sheath in this area. It has to be myelin bare. And next to that are two areas called paranode. This is when myelin sheath from one side will come and connect with the axon at this paranodal area. But pay attention to this area here. The myelin sheath is very compact. But when it moves towards the paranodal area, it becomes into these loose loops that seal the myelin sheath with the axon. And so this is non-compact area. So the proteins that our lab has identified over the years, and several other labs, are that this is the nodal complex where the sodium channels are, and they are critical for driving nerve conduction. And this is where the contactin-associated protein is that makes the paranodal area. And this is where the potassium channels are, and those are made by CNT and AP2. So our lab has played a major role in identifying these proteins over the years, and have made somewhere between 10 and 15 knockout and transgenic animals that have given us information on these. So you can imagine now, if you take the nerve fibers out from a mouse sciatic nerve and tease them into single fibers, what you find, and they're stained with 3 protein markers. That's the node stained for sodium channel. And this is called the paranode that's stained for CNT and AP1. And this is the juxtaparanode that's stained for potassium channel. And this is where the myelin sheath would be. So how do you organize these complexes to make the nerve conduction run? And so if you look at the more finer picture of this area, this is where the axon is, and this is where CNT and AP1 is. It connects the myelin sheath with the axon. And if you don't seal this here, you will have leaky current and the conduction will fail. So now we can go back and see what are the proteins that are localizing there. These are the proteins that make the paranodal complex, only the key proteins. And all of them have been knocked out in our lab over the years. And if you look at the first knockout, as I showed you here, if you look at the CNT and AP1 knockout, this is the control mouse, and this is the CNT and AP1 knockout. You can see the mouse is paralyzed. It's not able to go, even though it's trying, but its body is not coming because the peripheral and major nerve conduction is not working at all. So the mouse will essentially die within 2 to 3 weeks. So now the question is, what happens at the paranode? If you look at this picture carefully, you see the node, the paranode, the juxtaparanode. But in the knockouts, there's no paranode left. You only have potassium channels and sodium channels next to each other. And also the myelin, I won't show you all the data, the myelin sheath does not seal with the axon. With the result, there is breakdown of nerve conduction. And now the question is, how do we know that the myelin sheath connects with the axon at this site? So we had to identify the protein on the myelin side, that's neurofacin 155. So if you make knockout of that protein, what happens? This mouse also gets paralyzed, as you see here. So now we know that if you disrupt the nerve conduction by making single gene knockouts, you're going to have disruption in nerve conduction. And now coming back to what drives the actual nerve conduction at the node surface, that if you look at the nodal protein areas, and if you look at the complex here, these are the proteins that make the nodal complex. Pay attention to this channel here, that's the sodium channel that drives the conduction. So we have made knockouts of all these genes here, and I'll show you one knockout, that's neurofacin at the node that makes the nodal cluster. you can see this mouse is completely paralyzed, cannot move at all, the one with mouse. So now we have a question that if we make the knockouts of these genes and they die within 2 to 3 weeks, these cannot be used as model systems for human disease, because human diseases take longer time to develop, or if it's an adult disease, which happens in late 40s or 50s, those people have lived enough time, but the disease is now developing later stages. So what we decided to do, that we might be better off doing the inducible knockout that we can do anytime using tamoxifen inducible Cree system. So when we did that, we create the genetics. These animals have absolutely no phenotype. We inject tamoxifen. This time can be chosen whether it's 1 month, 2 months, 6 months, and then we wait for 1 week or 10 days and stop tamoxifen, and then you follow the animal, what happens. And as you'll see here, we're following this cluster here, what happens with this picture. And how long will it take before this cluster breaks down? And if you follow here, this is time 0. You have the nodal area, the paranodal, the juxtaparanodal area. Follow that the paranodal area is going down all the way, and in 90 days, we have no paranode left. And this is the same phenotype that we get if we make a conventional knockout. But now we have almost 3 to 4 months before the animal will start showing signs of the phenotype. And you can actually follow the phenotype. You can see this mouse, its hind legs are getting weaker. This is 1 month after induction, after knockout. And you can do a simple assay, put the mouse on the edge of the cage and ask it, how long does it take to go back in the cage? And when you put a knockout mouse on the edge of the cage, you can see the mouse is not able to go back in. And so we know the nerve conduction is declining, and eventually this mouse will get paralyzed. So now, if you now wanted to know what single neuron knockouts of these genes will do, we followed here Purkinje neurons. That is the unique neuron in the cerebellum. So this is the control mouse. You put them on the edge of the cage, they'll go back quickly. And you put the knockout of one single neuron, Purkinje neuron. This mouse cannot go back in the cage because it's unable to do the fine motor coordination that will allow it to go back in the cage. So now if you wait for this mouse to get old, and you can see how this walks with the left, right, and the symmetry is gone, and it's not able to walk. Eventually, the mouse will be completely paralyzed. So now coming back to this human, why does our work connect with human disease? So when you looked at those children, these children all have mutations in CNT and AP1, and they have a broad spectrum of phenotype. But there are some of them who have very severe phenotypes. So our work over the years had already established that there's a complex of CNT and AP1, contactin, and neurofacin 155. This complex is what makes the paranodal domain. That's shown right here. So if you now look at the picture, I was fascinated by Dr. Blundell's talk, and we only take the picture what's published by other people. And if you put now the CNT and AP1 protein in the ribbon diagram, you can see all these domains that make this protein. And some of the mutations have now been mapped to where they actually are. And you can see here, this is glycine 349 becomes valine, cysteine 323 becomes arginine, arginine 388 becomes cysteine. And so all these point mutations have now been mapped. So what we have now looked at, if we look at the structure of the protein that's shown here, and these are the amino acid changes that have taken place. And I'll show you, we have decided to follow the families who have these mutations. 2 children here, Isabella is from Netherlands, and James is from California. Their families have agreed for us to share the data. Isabella has a mutation that makes glycine 349 to valine, and James has a mutation that makes cysteine 323 to arginine. And you can see they are highly conserved cysteines, highly conserved glycines. And what we're trying to do now is, can we make mouse model systems that we can understand the disease? And if you follow James, very unfortunately, this is his situation because he cannot respond and he is unable to walk. And if he doesn't have the feeding tube, the air tube, he cannot survive. So now coming back to this, what we learned that James' mom carries the hypomorphic mutation. James' dad carries the null allele. In case of Isabella, father carries the null allele and mother carries the hypomorphic allele. So I will quickly show you what we have done. So we created these mutations in CNT and AP1, and then we expressed these proteins. One thing that became clear, that the mutation that James has, the protein does not localize the cell membrane where it's normally supposed to be. I'm going to skip the other 2 because we have more data on this one. We wanted to know, if you express these proteins in cell culture, can they bind to contactin? If you take the mutant CNT and AP1, what happens? It's interaction with contactin. What we learned, that the interaction with contactin, you can see here, we are expressing contactin and Casper together, and then we are doing the IP with either contactin or with Casper, and then we want to follow how much Casper is present there. In a normal situation where you have the wild type condition, Contactin and Casper, you can see how much Casper is pulled down. And if you look at the mutant forms of Casper, very little is coming down. And this information is very helpful if we want to design any targeted strategies for these children. So as I mentioned, we created using CRISPR-Cas9 technology. We have now created all these mutants and have the mice available in the lab. And I will show you at least a couple of them. And you can see here, this is the mouse that is the same mutation as James. This mouse does not thrive. This is the point mutation or null allele. And you can see the protein is not very stable. These mice don't gain much weight. And if you don't feed them separately food, they will die quickly. Fortunately, the other two proteins that Contactin-associated protein works with are not reduced. That's good because if you want to do any gene therapy, you want to make sure the other two proteins are still present and you can rescue them. What's very important is this, that the mutant proteins follow these two stainings here. This is the normal Casper localization of the paranode, but the mutant protein does not go to the paranode. Whichever way we look at, the mutant protein is not able to move out of the paranode. So if what we find that the protein is basically stuck in the ER and doesn't transport out to the cell surface. And you can see here, this is the heterozygous situation where we can see the mutant protein here in the cell body. This is the motor neuron, and in the mutant or minus, the same protein gets stuck in the cell body. So with the result that if the protein is either misfolded or not transported properly, it fails to go to the paranode and establish the axonal junction. And so if you look at these mice, this is a control mouse that's able to cross the beam. So we can put the mutant mouse on the beam and see how severely are they impaired in their motor coordination. And you can see this is the knockout. This mouse is not able to hold on the beam and quickly falls off. So what we have done now, we want to make sure that we have both these children and their mice established. We have now made the mouse that carries this girl's mutation, that's glycine 352 valine. GGT is glycine, and we have now GTT, that's valine. So this mouse is also being characterized now. But now, this is all well and good. How do we move from mouse studies that we have done over the years to human studies? And this is where the challenge lies. We have been able to do in one mouse model that we can rescue the mouse. And if you follow here, this is how the mouse walks. This is control mouse. You can see hind foot and this is front paw, hind paw, and both sides, they're marked. This is called catwalk program that tells us how the mouse is walking. And if you look at the beta 4 spectrum mouse, this mouse is unable to walk. And if you now try to rescue, we can follow the rescue now. This is control. This is beta 4 spectrum mouse that gets paralyzed within 10 months, not able to walk. And if you now re-express the gene, we can make the mouse walk back again. But the timing of this rescue is very important. Earlier the rescue, the better the mouse is. Because as we have no conduction decline, the muscle becomes atrophied and you lose the muscle. In most of these cases, muscle atrophy is what makes people go in the wheelchair. So now coming back to the human studies, what we have done now, we want to first establish that we can rescue the mouse phenotype by doing conditional expression of CNT and AP1. And here what we have established is the lock-stop locks expression for CNT and AP1. It has a stop signal. It will not express the protein unless you cross it to a CRE line. So we have established that now that we can, in fact, if you look at the flag staining in the mouse, which has lock-stop locks, without CRE, there is no expression. That means it has its type. With actin CRE, we can regain the expression of Casper. And this is endogenous Casper. And this is the nodal staining. And now this is very important for us to show that we can actually rescue in a timed manner. There should be no expression without crossing it to CRE. And you can see when we cross it to CRE, we get Casper back. And this is using anti-flag to differentiate the transgenic CNT and AP1 from non-transgenic. So this is all good. What we have to do now is try to express this in the motor area. So we want to know, can we rescue the motor function in these animals? And we use the stereotaxic injection. These are the CRE mice, CRE transgenics that express green fluorescent protein. We deliver this to the motor area and we want to follow, is it making the protein? And we can actually say that the experiments are successful. This is the injection site where the virus is injected on both sides of the motor area. And the virus does spread, expressing by expression of green fluorescent protein. So we now know that we are able to deliver the virus, and we want to follow, can we rescue the knockout? Can we rescue the hypomorph or null? Can we see a benefit in terms of phenotype? And those experiments are ongoing. But one good thing is that we are able to express, and the protein that is exogenous or transgenic and the endogenous protein both do co-localize. That means the transgenic protein is going back to the paranodes, as you see in these dots. This is the myelinated area of the corpus callosum. These are the motor areas or the cortical areas. And we can tell by GFP that the transgene is being made from the virus. So what we are going to do now is we don't want to have direct transgene expression. We want to have a viral expression. So we can directly inject the virus to the people or to the animal so that they can get the benefit. And this is done in a lentivirus vector, not the conventional AAV, because the AAV cannot accept the large CNT and AP1 gene. These experiments are ongoing. The viral expression is being carried out, and hopefully we should have that information in the next few months. The questions that we worry about, as every scientist has to think about, what would be the critical factors that will determine the rescue? One is, can we get enough viral spread to infect enough neurons on myelinated axons? Is there an issue that if we did not catch the developmental myelination to establish these extracellular contacts, will it work after we make the protein once the child is already, or the mouse is already 1 month or 6 months old? Then the issue that we have to deal with is, if the muscle atrophy has already kicked in, can we rescue the animal and be able to walk again? So I will summarize this part, given the interest of time, that we have used genetic and molecular methods to identify the proteins that drive nerve conduction and maintain our motor function. And next, mutations in human CNT and AP1 do impair fast nerve conduction, and they lead to neuropathy and paralysis. And mouse models are being now developed and to design and test potential therapies that can restore nerve function and motor activity. And we are now assessing the efficacy of lentiviruses in CNT and AP1 mutant animals of different genotypes and backgrounds, so that we can then go to the children if this is approved by multiple agencies, so we can get this back to some level and help the children to recover from their deficits. I will stop here and take questions from you.
So, can I ask a question?
Absolutely.
Oh, this is spectacular talk, and I'm just curious to know. There are two problems you presented. One, a mutation-driven paralysis or a neuropathy in young children, and also in adult, an age-driven process. So the approach you are taking is spectacular to fix for these children. Hopefully, I'm sure you must be considering the novel RNA-based kind of the vaccine similar approach where you could probably deliver RNA. I'm sure you must be doing that. But the question I have is for the older people. Have you guys thought about using an epigenetic approach? Because there it seems like aging, it's not going to lead into accumulation of additional mutation. Rather, it would be an epigenetic phenomenon. So have you thought about looking into epigenetics or epigenetic modulators, inhibitors, to see whether you could reverse that?
Well, the issue is that as we age, nerve conduction declines. If you want to use epigenetic methods, they can be used if it's an expansion of a repeat like a polyglutamine. If you want to suppress the expression, the problem we are facing, at least in these children, they have a null copy from father or mother, and they have a single point mutation that makes the protein probably misfold and get stuck in the ER. So, and you have a bigger problem after that. This is not a single cell issue. If it was just a liver cell or just a neuron, it was fine. We could do something. You have to make the protein in the cell body. It has to be transported down the axon. That could be a meter long. And you need to get the protein to the paranode where the actual function is needed. And so no matter what we do, you have to make sure that myelination functions properly. And so whichever way you look at it, if you don't have Casper functional, we cannot restore nerve conduction. So and also you have to cover enough neurons to get a benefit. You can inject 10 neurons, 20 neurons, but you need the whole motor system that must fire, and you need the whole cognitive system that must fire. With us having billions of neurons, how many neurons can we reach? And so that's a challenge for all neurological diseases. If it's a single cell type of cancer, liver cancer, kidney cancer, or breast cancer, you could try. But with neurological problems, you have circuits, you have developmental issues, and then you have muscle atrophy. And those are not fixable. So what we are trying to do is we want to know how much the mouse can inform us first. Nobody will allow us to inject virus into the people. It's impossible. So unless we have all the IRBs and IACUC approvals within our institution, we may take it to the only viral therapy seems to be the only method, because you need these proteins to function at a very specific place, and that's the paranodal region. And without that, these children have no hope. If you don't feed them and you don't put them on the ventilator, they cannot live. Thank you.
Well, Subbarao, can I?
Yes.
Can I ask you a question? Manzoor, this is Suraj Bhatt.
Hi, Suraj.
It's a pleasure to meet you, even if it's only virtually.
Likewise, likewise.
It's such a joy to have you speak here with a terrific talk, a phenomenal talk. I won't speak any Kashmiri at this time, but I would go to science and this expression of CNT NAP in vitro. When you do those studies, do you find expression of other proteins associated with this CNT NAP1?
No, so
it's transferred from ER to the cell surface.
So very true. What happens if you have CNT AP1 mutation, CNT and contactin is stuck there too. They need to move together. So if you don't fold properly, you don't transport properly.
Absolutely.
And so we are looking at ER stress as well, because these proteins will cause ER stress.
So
and the PERK pathway is activated.
You'll possibly find some chaperones there, which may be required to transfer it to the cell surface.
And we looked at BIP, we looked at CHOP, and we're looking at all those proteins, because for us to get the proteins.
Did you look at any crystallines?
We have not looked at crystallines, but we could. I mean, any protein we can look at to benefit some way, it's doable. And the difficulty is that you do not have all neuronal proteins that you need in hex cells. So we are doing co-culture now. We're doing the co-culture in mutant neurons and wild type oligodendrocytes. Then we can, using the knockout mice, and then provide back the point mutations. And that we're doing in co-culture. Yeah, that's the difficulty, because you need both cells. You need the neuron and you need the glial cell.
Absolutely.
But very important point. We're looking at the ER. We are not experts on ER stress, but certainly we want to know what's going on.
But the transport to the surface is a critical, important aspect that you pointed out. And that could be one way of reaching the therapy to correct that particular area. Phenomenal. It's so good to see you. And we will talk. We'll talk. It's a pleasure.
We should chat sometime.
Absolutely.
Now, just quick suggestion to Manzoor. This is Srivatsan, because we have been collaborating with somebody, Stephen Gray. He is in US, the Southwestern. Yeah, he is doing direct injection, because we are doing some of the giant axonal neuropathy, some of those kind of stuff, connection to cancer, because you're talking about cancer and everything else. He is doing direct injection into the neural site. I don't remember the exact site.
I know Steve. Yeah, I know Steve. Yes.
So you've been, because you're talking about very difficult, but he has been doing it. I agree, 1 or 2 children at a time, but I think it is possible.
The difficulties are how to cover. The gene is too long for AAV, so you need lentivirus. And so they did try AAV, but they didn't succeed. And so they had difficulties. But you cannot touch a child unless you have the proof of principle in mouse. You cannot even do anything in human. So we're doing the lentivirus. Yes.
We are doing lentivirus. We are doing lentivirus with Don Cohn. You know Don Cohn. He is there, the gene therapy business. Like there was somebody was saying, just change only the polymorphism. That's what we are trying to do. Just to do one SNP change, and then see with the lentivirus and see injections.
You could, but the problem what we have learned from the structure that I showed the ribbon diagram, that wherever the mutations are in the protein, you cannot truncate it, because the mutations are spread throughout the protein. So you need the entire protein, because the exoglial junction between the myelin sheath and the axon is 19 nanometers. So if these proteins don't fold properly, they'll not make the junction to regain nerve conduction. So there's a lot of biochemistry, a lot of cell biology, but the therapy is where we want to go, using all the knowledge that we have to see if the children can be helped.
Yeah, you're absolutely right. I mean, lentivirus, first thing we are also trying to do is put the whole gene. So you are absolutely right.
Sure.
That's probably the best way to know the proof of principle. Thank you very much. It's wonderful.
Yeah, absolutely.
Thank you, Dr. But I have a small question about this neurofascin and Casper proteins.
Yes.
We know that there are some autoantibodies against the neurofascin and so on in the neuropathies, right?
True.
But do you find the fragments of this protein? For example, in traumatic brain injury, in Alzheimer's disease, some of these, whenever there is a brain damage, you may find some of these proteins are fragments of the proteins. Do you know any information on these two proteins?
Well, so certainly neurofascin is and Casper. Casper is CNTN81 is also called Casper. And neurofascin. There are antibodies in MS patients and targeted to these proteins. And those people also develop neuropathy, both peripheral and central, and essentially get paralyzed. Now, your question in TBI, every time you have myelin damage and myelin sheath disorganization, you will trigger some antibody response. But we have not directly, obviously that's not an area that I work in, whether the demyelination or TBI or any tissue damage in the myelin area will lead to production of antibodies against these proteins is possible. And especially in MS, once you start damaging myelin sheath, these proteins are right there. But one point I can tell you what we learned when we were trying to make antibodies to these proteins, the company said the animals are getting paralyzed. And that means if you inject the mouse protein and you're making antibodies, it may target the animal's proteins itself. And they did become weak, but I do not have enough information to answer the question directly, what would happen in TBI or what would happen in other model systems.
Yeah, I'm more interested in the protein itself instead of the antibodies, because if there is a damage, for example, the glial fibrillary acidic protein, the GFAP,
yes,
that's used as a marker for TBI. And then UCHL1, and there are spectrins, breakdown products, so many myelin basic protein, S100 protein. Many of these are tried for the brain damage. I was wondering whether neurofascin can be used.
This is very unpublished in the lab, so kind of being protective of my students.
Okay.
We think that these proteins may have,
Dr. Bhatt.
Yeah, we think these proteins may have some signal transduction function as well, and they may be triggered under certain conditions. Yeah, but it's
Thank you very much.
Thank you.
Thank you very much. I think we have to move on to the next speaker.
Absolutely. Thank you.
Okay, it was a wonderful talk. We all enjoyed your talk.
Thanks again.
Thank you. Thank you very much.
And we will talk. We will talk.
Yes. We'll go to the next speaker. He is Dr. Murali Krishna Kaja. He received his PhD from the Institute in 1995. His graduate work focused on protective immunity to Japanese encephalitis virus. Following this, he received postdoctoral training at Emory University. During this time, he published some of the most highly cited papers pertaining to the mechanisms of immune memory and its maintenance. Following this, he served as a faculty at the University of Washington for a decade. Then in 2011, Dr. Murali joined Emory University as an associate professor of pediatrics, Division of Infectious Diseases and Emory Vaccine Center, and as the associate director for the joint Emory ICGEB program. It's an international center for genetic engineering and biotechnology program. In this capacity, he spends most time in India overseeing the ICGEB Emory Vaccine Center joint program on translational aspects of human immunology, immunotherapy, and vaccines in India. Today, he'll be talking on the human immunity to dengue virus. Now, Dr. Murali, please go ahead and start.
Yeah, thank you, Dr. Subbarao. Can you see my screen?
Yes.
Okay, great. Let me put it on to the slideshow. Okay, all right. Thank you for the nice introduction, Dr. Subbarao. And good morning, good evening, good afternoon for all of you, where depending upon wherever you are. Um, and I also want to thank Professor D.N. Rao, Professor Rangarajan, and Dipankar Nandi, and all the Biochemical Society organizers and the symposium organizers. It's a wonderful feeling and opportunity for me to have the chance to present our work in this 100th year of my beloved department. Um, and um, many of you are mentors, and some of you are actually mentors, and some of you are friends and colleagues and seniors, and some new students. Thank you all. I also want to acknowledge my mentor, Dr. R. Manjunath, who actually initiated me into scientific career in this department. Um, when Dr. D.N. Rao asked me to give a talk, I was kind of thinking, one of my actually most beloved memories of this department was the old seminar room, which was actually right here in this building. There is always so much of excitement when seminars happens, and the physical the interaction with the speakers and one-on-one interactions. And unfortunately, now we are distant. We are talking through cloud. Hats off to the technology, but it kind of gives a strange feeling that we are talking through cloud, but that's okay. Um, and when when Professor D.N. Rao asked me to give a talk here, I was like thinking, should I give all my summary of my research, my life journey from IISc till now and so on. But then I thought that maybe I should actually talk some of the key findings, which are actually not yet published, but they are evolving um, as a result of studies that we started in India about 10 years back. And I decided to talk about them in this next 20 minutes or so. And I decided to talk about them simply because I feel that these findings are very important and timely and relevant for India, both for dengue vaccine development and implementation and policy design. So that's my intention in this next 20 minutes or so. So many of you obviously know, you have seen dengue in India, and there's not so much to detail to go about the introduction about dengue, but I want to bring out 3 or 4 salient points, to bring you up to date and set stage for my talk. So, so one point is that India actually has the highest dengue burden worldwide. Um, it was not the case, like 4 or 5 decades ago. Actually, dengue was very high in some of the Southeast Asian countries like Thailand and so on for many centuries, many hundred, probably around 100 years also. India actually happened to have a major dengue outbreak only around couple of decades back. But within these 2 decades, dengue has exponentially increased in India, and now the current estimates are that we actually see around close to about 30 million human infections each year. And that's a huge exponential increase. And the recent estimates suggest that if you look at all these parts of the world where dengue is predominant, which is not green color, India remains epicenter, and and nearly 1/3 of the global dengue disease burden in the world that happens in India. And the second point that I want to bring out is that some of the dengue cases can be very severe. So if you look at this nice cartoon that was published in Nature Reviews in Immunology, if you take the total dengue infections of humans, majority of them actually are, could be very mild or not even symptomatic. But on the top of this pyramid, you will see these are symptomatic cases, which means that they would actually need to go into the clinic. But even within these symptomatic infections, at the top of this pyramid, these red cases are the ones which have the most severe dengue, which can be defined as dengue hemorrhage fever or dengue shock syndrome, and some deaths happen here, especially in children. Now, although it sounds like this small red portion is so small compared to this, now if you take the total number of people that are usually get infected every year, and then out of them about a quarter of them come to the clinical cases, and considering that India actually has nearly a third of global dengue infections. So in a typical dengue season in India, there is a huge stress and strain on healthcare systems, and this is what you see normally. And basically, health systems are clogged, and people are, distressed. And also, currently, there are no vaccines, at least in India, in dengue, and there's no licensed therapeutics. So only supportive clinical care is the only thing that is available. So, and the third point that I want to bring out is that dengue is considered to be a baffling human disease. And I'm saying it is a baffling human disease, not because a majority of the, only a fraction of the infections become symptomatic or severe cases, but because it is, there was some very interesting concepts that have been developed over the last 60 years. Actually, this concept developed by Dr. Scott Halstead, and Scott Halstead was, actually he was born in Lucknow in India. And then, of course, when he was doing the studies in Thailand, he found that majority of the severe disease cases actually happen to have IgG and or neutralizing antibodies or hemagglutinin antibodies, which means that they probably were previously infected with dengue and again got infected, and then so that led to development of this idea that primary dengue infections usually are unlikely to be dangerous. Primary means first time infections. But if a person who was infected with one dengue serotype, I forgot to tell you, dengue has 4 different serotypes. If a person is infected with one dengue serotype in the first time, and maybe they don't really develop a dangerous disease, but if the same person gets infected to a second time, that's called secondary dengue infections. And that with a heterologous serotype, then it is more likely to be dangerous. And this is a baffling, for me, it appears that this is a baffling concept, not simply because of anything, but if you really look at, what all we know about generally in the infections or infectious disease areas, in most cases where there are multiple serotypes, if one serotype infects and then afterwards a second serotype comes in there, you expect either they are protected, or if in worst case scenario, they would be as severe as in the first time infection. But a second time infection becoming a lot more dangerous than the primary infection, for me, is very baffling. So there must be something very interesting going on. And Scott Halstead put this idea around 1960s, probably around the time when I was born. And in these last 60 years or so, huge efforts have taken place all over the world, and then trying to understand the mechanisms of this, why this happens. And there are many theories. The most prominent is that first time infection happens and people develop antibodies. And second time, as different serotype infection happens, and these pre-existing antibodies would cause something called antibody-dependent enhancement of infection, which would lead to this severe dengue disease. That's what has happened. And now it's very important to understand that this concept has enormous implications on development of dengue vaccines and or therapeutics and also on implementation of the vaccines. And so several dengue vaccine efforts are obviously are underway. Even in India, actually, ICGV, where we are working, has put a major emphasis on developing dengue vaccine by my colleague, Dr. Naveen Khanna. And in the world, there are some vaccines that have gone through the clinical trials, and probably the Sanofi vaccine is the one which has gone through the most advanced clinical trials. And a good part is that it worked well in people who are already immune to dengue. And but the not so good part is that some of the vaccinated naive individuals actually developed clinical dengue when exposed to the natural infection. That was kind of like unexpected, and it's not a good thing. And according to Sanofi Pasteur, basically what they think based on this idea that I introduced from that the secondary infections are likely to be more severe, they think that this vaccine partially mimics primary infection and thereby increasing the risk of severe dengue during the subsequent infection. So this is where things were around the time when we started the work on dengue in India. Um, so when we started the work in India, basically we wanted to ask a question on what kind of immunological differences exist in these patients with and without severe disease. If you analyze the patients who come to the clinic, our idea was based on the introduction that I gave, that considering that the secondary infections are likely to be the most severe disease cases, so we thought that we should see if you recruit the clinical patients from India and then analyze, it is very likely that people who have the most severe disease symptoms should be the secondary dengue infections. Whereas these people who have milder dengue fever should potentially likely to have a mixture of primary and secondary dengue infections. And if we understand what are the immunological differences between these two groups of patients, then we would actually understand the potential mechanisms and which would help us to design better vaccines and therapeutic strategies. So we started looking at T cell responses, B cell responses, and also we started isolating the B cells from these different kinds of patients and then try to understand. We started making human monoclonals so that eventually they would lead to improved therapeutics in India. But what I'm going to do now in the next 10 minutes or so, I'm not really talking about all those immunological differences and making monoclonal antibodies and so on, although they are very interesting. I want to take a very key finding from these and basically I'll tell you what is coming at the end of my talk. That way there won't be any surprises. Our studies in India actually are not aligning well with this idea that is developed over the past 60 years, suggesting that secondary infections should be the most severe cases, whereas the primary infections not. So let me now get into data without much delay. So basically, this is our study design. We recruited children, the ages can be as low as 2 months to 18 years. And we recruited them from 3 different clinical sites distributed in different parts of India: Delhi, Bangalore, and Vellore. From Delhi, it is All India Institute of Medical Sciences. From Bangalore, it's St. John's Institute for Medical Sciences. And in Vellore, it's Christian Medical College. And that recruitment happened from 2013 to 2018. And from this patient cohort, we basically identified 619 clearly dengue confirmed febrile children, and based on the well-established methods MS1 and or IgM and or PCR. So our goal was to characterize disease severity and then perform immunological assays. And the disease severity was characterized based on the World Health Organization guidelines. So essentially, those patients who have severe plasma leakage or hemorrhage and organ impairment are called severe dengue, and which I would call SD in my future slides. And whereas those patients who have either, these kind of milder symptoms or some kind of warning signs that the patient might develop into this kind of category are called dengue infection or dengue warning signs. So I would call these categories in my future slides. So the very first thing that we wanted to ask is that how many of those patients, the 619 patients that we recruited from these different parts of India, are primary and secondary infections. Our hypothesis, based on the introduction that I gave you, is that most of the secondary infection should be exhibiting the severe disease, whereas the primary infection should be having these milder dengue. So first, we ask just what happens in the total patient population. But to ask that, one important point that you want to understand is that these are natural infection models, right? So you don't know whether this person was exposed to dengue in the past or not. So it's not easy to understand whether it's a primary or secondary infection. But there are well-established procedures and assays, and that's based on this simple concept that if a person is exposed first time, which is called primary infection, usually during the fever time, they would induce a lot of IgM, a little bit of IgG, which comes later. But if the person is immune and then get to a second time infection with a different heterologous serotype because they have memory B cells, they would use very fast responses of IgG and low levels of IgM. And historically, this IgM-IgG ratios would differ between primary and secondary infections. And based on very well calibrated ELISAs to classify the primary and secondary infections, World Health Organization recommended a particular set of ratios and to delineate these two categories. So we did that, and the first thing that we find is that in our total patient cohort of the 619 patients, nearly half were primary infections and half were secondary infections, which is expected. But what then we would expect based on the results that are, the theories and I mean the ideas that are evolved over the last 50, 60 years, most of these secondaries should be having severe disease, whereas this primary should be mostly milder infections. But we did not find that in our India studies. That's the bottom line of my talk, and which was surprising, and I'll show you the data here. So when we look at, here we are looking at the total patients or the patients who are primary dengue infection, patients who are secondary dengue infection. And now we are looking at the percent of each of these categories that have either severe dengue disease or dengue with warning signs or dengue infection. Essentially the dengue warning or dengue infection can be considered as milder dengue, but this is the most, or at least non-severe. And this red portion of the pie diagrams are the severe dengue. So to our surprise, we found that it did not really matter whether we look at all cases or primary patients or secondary dengue patients. In all of them, there are very similar proportion of severe dengue cases. So essentially, what this is, the bottom line of this, what this is telling is that our 6 years of work in India is basically not aligning well with the 60 years of the concept that has evolved in the dengue field, suggesting that secondary infections would be much more severe. At least they should be associated with severe cases. And that was very surprising to us. And obviously that concept was developed by studies conducted in other parts of India, because in India, there are very few studies on these aspects. And now when your 6 years of work does not align well with the 60-year concept that has been established in the field, then obviously you need to do a lot more work to convince yourself first, and of course also to convince the others. So we were wondered, there were some unfortunate fatalities in this patient cohort, and we wondered whether the fatalities happened in primary infections or secondary infections. So here what we see, out of the 7 fatalities, actually 5 were primary dengue infections, which also kind of tells that it's not very well aligning with the concept that is out there in the field all over the world. So now we took a step back and asked, are we really distinguishing these primary and secondary dengue infections properly? The methods that we used are the standard methods that are actually being used all over the world. And these are very well calibrated and highly evaluated, and World Health Organization recommends them to use. But one of the things that came is that if somebody is truly primary infection, they should be inducing a much lower levels of neutralizing antibodies to the virus. But if somebody is secondary dengue infection, they should be inducing much higher levels of neutralizing antibodies. So we went back and looked at a subset of our patients and asked what are the neutralizing antibodies against each of the dengue serotypes. We looked at the dengue 1, dengue 2, and dengue 3, and dengue 4. And what we are looking at here is that this set shows the primary dengue infections, and these are the patients that were characterized as secondary infections based on our capture ELISAs that I described till now. And you see that if the primary infections, most of them were either neutralizing antibodies cannot be detected, or if they are detected, they are much lower than the secondary infections. And that happens whether we look at 1, 2, 3, or 4 dengue serotypes, or look at the dengue serotype that is infecting. So this actually strengthens that our observation is correct here. And then we asked, let's go to a little bit more deeper step and ask if whether the methods that we use to classify primary, secondary dengue status correctly classify infants as primary dengue. And here is the logic is that if you look at the infants, obviously the infants are born just now or a few months back, and there is no way they could have got one time dengue and then afterwards getting again a second dengue. So infants should theoretically be only primary infections, or at least a majority of them should be the primary infections. So we wanted to know if we use the same methods that we use it to classify the primary versus secondary dengue infection in all the patients that I described till now. Do they correctly classify these infants as the primary dengue? So we looked at it, and here I'm showing infants and children, which means that these are children 1 to 18 years, and these are infants who are basically less than 1 year old. And the methods that we used basically tells almost all the infants are primary infections, and whereas in children, it's half and half, primary and secondary infections. So this tells you that the methods that we use are correct. The methods are classifying all the infants as expected are the primary infections. And we also wondered what happens to the disease severity. Here they are all primary infections, and here there is a mix of primary infections and secondary infections. And what portion of these infants who came to the hospital had severe disease? So So what we actually find is that these infants who are all primary infections actually had much higher levels of severe disease compared to the children, and regardless of whether those children were secondary infections or primary infections. So there is some level of age-related issues that are going on, in the sense that if you are younger, very young, probably that you can have much higher disease. And I wanted to bring out the fact that in the field, historically, there, it has been recognized in the past that infants do get severe disease. It's just that, I think the interesting thing that we are finding is a lot more infants actually have severe disease. But infants do get severe disease, and it was kind of explained that infants may have the mother antibodies, and which may cause severe disease through the antibody-dependent enhancement of infection, and which need to be examined in our Indian studies. So that's basically where we are. And in summary, basically we examined 619 dengue confirmed febrile children from 3 hospitals in different regions of India. And what we find is that primary infections, dengue infections, accounted for more than half of the total clinical cases, severe disease cases, as well as in fatalities. And we also find that the younger children were more likely to have primary infection and more likely to exhibit severe disease. So now, to put these things in the perspective, going back to the introduction that I gave, so basically these findings question this currently prevailing notion in the field that severe disease in dengue is predominantly associated with secondary dengue. And additionally, obviously, because now there are a lot of primary infections and they do have severe disease, younger infants and children. Basically, and considering that currently the dengue vaccine development, evaluation, implementation, and policy vaccine implementation policies are driven by this notion that severe dengue is predominantly associated with secondary dengue, we need to, at least in India, take a step back and reevaluate our strategies. And basically they are telling us that it is very important for us to develop vaccines or other potentially therapeutic treatments that can protect naive individuals, especially children and young children. And with that, I want to acknowledge this is actually truly a team effort. There is a unique partnership between Emory Vaccine Center, as Dr. Subbarao said in his introduction, and which I direct. And without this partnership, actually, this kind of huge task of studying these children would not have been possible. And we have excellent clinical collaborators from all over these different parts of India, AIIMS Delhi and St. John's Hospital from Bangalore, and CMC from Vellore. And these are, fantastic clinicians and who really worked very well with us and in recruiting the patients and providing the samples and providing the clinical characteristics. And National Institute of Virology is involved in understanding the dengue infecting serotypes. Translational Health Science Institute, Guru Prasad Medigesi has been a long-term collaborator. Excuse me. And then there are a lot of people involved from our laboratory, our joint laboratory at ICGB in New Delhi, as well as from Emory University. Anmol Chandali is an assistant professor at, and who heads our group at the ICGB Emory Vaccine Center lab at ICGB in New Delhi. Many of the students and both from our ICGB Emory Vaccine Center lab at Delhi, and students and postdocs, and as well as from Emory University have made a huge contribution to this work. And finally, I also want to acknowledge funding. This funding is actually largely supported by two initiatives. One is National Institute of Health, ICIDA program. It's called International Collaboration in Infectious Disease Research Program, to increase actually the immunology and infectious disease capacity in developing countries. And we were fortunate to have this funding when we started this work. And the Department of Biotechnology, Government of India has been a huge supporter in these works, and also in association with Indo-US Vaccine Action Program and Human Immunophenotyping Consortium. So I don't know why my screen looks okay. So I want to end this talk. This picture always amazes me. I mean, these are basically the tree climbing goats from Morocco. Every time I look at this, and every time I look at the way the science progresses, and the way at least my science progresses, tells me that science is an evolving concept, and if we keep trying, we will reach there. And with that, I want to thank all of you. And if there are any questions, I would be happy to address.
Thank you, Dr. Kaja. Yeah, we'll take the questions.
Bro, can I ask a question?
Yeah, sure.
Dr. Krishna, Dr. Krishna, thank you very much for a very lucid talk, very informative talk. It's obvious that the IgM and IgG ratios may not be the perfect phenomenon to catalog primary and secondary infections and catalog severe, particularly severe and non-severe infections.
Right.
I would like you to comment on where is the bottleneck? Have all the antigens been identified? Where is the problem in identifying the antigens? And where is the problem in making a vaccine? What are this? This is something by far the most important talk, which has direct relevance to what's going on in the pathological area of Indian clinical phenomenon.
Right.
So would you comment on that? Where are the bottlenecks?
Right. Yeah, I mean, it's a very relevant and important question. I think where are the bottlenecks? I think the bottlenecks are coming from, at least from my personal perspective, there are three sides. One is that, dengue has 4 different serotypes, and obviously you need to develop a vaccine to all 4 serotypes at the same time. And there is substantial progress has been made. NIH vaccine has all 4 serotypes, and Sanofi vaccine, which I mentioned, also has all 4 serotypes, although the methodology could be different. And they might work. I mean, the Sanofi vaccine actually worked very well in the people who are already immune to dengue. And it's just that it did not work in the naive people. And there are these perplexing results that the naive people who are vaccinated actually went to clinic with clinical symptoms, which baffled the people. And then it is explained that it is because of this antibody-dependent enhancement and all that thing. And now our India results are saying that no, if you take the primary people, you take 619 patients coming from different parts of India, and there is a huge number of primary infections who are actually developing severe disease. Now, if you go back and look at these Sanofi data, I mean, should they be? I mean, in those countries where the Sanofi vaccine is tried, why did they first expect that primary people should not be developing the severe disease? I mean, that is because of this concept that has been there in the last 60 years. So,
isn't there a dedicated effort in India where in a few centers are putting their efforts, their academics and their clinical prowess into getting to the bottom of this, so that this problem could be addressed both at the academic level as well as at the clinical level? Is there an effort by the government or by ICMR, by anybody, rather than depending on foreign organizations to look into dengue? The example is the response to SARS-CoV, the way the country has gone into making antibodies and things like that. Is there an effort like that in that country?
Absolutely, there is a lot of effort. In fact, I mean, the Indian government Indo-US Vaccine Action Program has, and with ICGB, where we have this partnership, put a huge emphasis in the last many years to develop a tetravalent dengue vaccine, which is actually now transferred to a biotech company, and then they are trying to do the GLP production. And of course, the other dengue vaccines are also, being considered in India for clinical trials. So the point that I'm making is that all these policy decisions are driven by this concept that severe dengue is, is the most dangerous thing for causing the severe disease. And what we are finding in this last 6 years of our work from India is that our findings are not aligning with that concept. So now we actually have to develop our own strategies of how to either develop the dengue vaccines and therapeutics, and more importantly, how to implement them. Because, the concept came from Sanofi vaccine. The vaccine can be given only for the people who are more than 9 years old, because, it's very likely that they would have been already exposed, whereas the children before that time could be naive, and then they either don't need the vaccine because naive infections are unlikely to be severe, or the vaccine might be dangerous to them. So that's one part. And second party, the Department of Biotechnology, ICMR in India, are making huge efforts. Actually, they have supported our program. And, in the recent past, we actually developed the COVID monoclonal antibodies from India, and many of them are neutralizing. And we also developed dengue antibodies. Many of them are neutralizing. I think we will reach to some therapeutic aspects eventually, but it takes time. I think you need to do the work in a more systematic manner rather than looking at the fruit right there the next day.
Okay, sorry to interrupt. Sorry to interrupt. We have two other questions on the chat. Let me read those questions, and we have to be quick in the answers also.
Absolutely.
The first question is from Dr. Padmanabhan. There's a nice talk.
Murali, I can ask this question, and a very nice talk, Murali, very useful. I just want to know, people say ADE is not an issue in COVID. What is your opinion? I'm digressing, but just want to know your.
Sir, it's, when you do ADE experiments in vitro, obviously it works with almost every virus infection. It doesn't have to be dengue, it doesn't have to be COVID, it doesn't have to be anything. You take JV, you take West Nile virus, most of the things work in vitro on cell lines. Whether the same ADE phenomenon happens in vivo, currently the field is not at a stage to really say it with confidence. In COVID, we are at a too much early stage. And in dengue, the field has advanced so much with the idea that the infant should be developing severe dengue because of the ADE. But then the infants could be developing antibodies, some low level of IgG, just like children develop the low levels of IgG, and we don't know yet. So I think the people who are looking at the policy decisions and taking bigger steps in trying to look at the direction should look at these things much more carefully. So
we have one last question.
Yes.
From Santa Ramakrishnan. Is he going to ask? Or I can read that question. Hi, Murali, great to hear your talk. Is the 60-year hypothesis based on adult data or children data or both?
Nice to hear from you, Santa. So the 60-year hypothesis is basically based, it's basically based on both children and adults, so see, you just don't take me wrong. We are not at a stage where all the hypothesis, that has been generated in all over the world is wrong. At least in India, that doesn't apply. That much we are confident. Whether in the world, whether that happens or not, there could be some issues, and there are some people who disputed that in the past. Actually, Leon Rosen, who in his American presidential address in the American Society for Microbiology spent a whole hour trying to tell people that don't believe this hypothesis. That he did it in 1976. At that time I was 11 years old. So, but I only read it in the recent past. So, but then the general thing in the field is that secondary infections are severe ADE causes, and so we should develop a vaccine which doesn't cause ADE, and we should vaccinate the people who are pre-immune to children. So what I'm saying is that in India it doesn't happen. That way it doesn't happen. In the world, I don't know. I mean, Leon Rosen disputed it, but then, I don't know if all these people, how can I say that all these people who worked for the last 50, 60 years, they all said these things based on some data. So I don't know.
Okay, thank you. Yeah, I think we should move on to the next speaker, because it's getting late for people in India. I think some people are leaving already.
Yes.
The last speaker for this session is by Dr. Ganesh Kishore. He is a co-founder and managing partner of Spruce Capital Partners. He has a distinguished track record of accomplishments in biotechnology, research and development, and business both. So after receiving a PhD from the Institute in 1976, Dr. Kishore received postdoctoral training in chemistry and biology at the University of Texas in Austin. He then joined Monsanto, where he made contributions to the discovery of Roundup-ready technology and the synthesis of aspartame. During his tenure at Monsanto, he was named a Distinguished Science Fellow and won the prestigious Queenie Award. Before moving to DuPont, he served as the president of Monsanto's nutrition and consumer sector. He joined DuPont as the Chief Technology Officer of Agriculture and Nutrition. He retired from DuPont as Chief Biotechnology Officer to join Malaysian Life Science Capital Fund as CEO, and later co-founded Spruce Capital Partners. He serves on the board of several companies, academic institutions, and the advisory board of Scientific American. He's a recipient of Bio's Legacy Award, ASPB's Innovation Award. Let's listen to him about RNA biologics and gene editing now. Yes, Kishore.
Thank you, Subbarao. I missed the golden jubilee of the Department of Biochemistry by 1 year. And today I'm only late by about an hour. So I'm certainly heading in the right direction. I realize that I'm holding up people from either their breakfast or their bed. So I will try to be brief, but keep my points fairly crisp. One of the things that is very evident as we have listened to all the presenters today and yesterday, there have been very lucid presentations, as many have commented. It is very clear that science for science's sake is very enlightening. But when you actually translate that science into humanity's services and products, it is gratifying. And when you do them in an outstanding manner, it is invigorating and energizing. It is that theme that has guided me throughout my life. And I do not distinguish between applied science and basic science because they kind of transform each other and allow us to reach new heights. I was really blessed when I joined the Department of Biochemistry. I was interviewed by 17 faculty members on day one before I was even admitted. And one of the people who interviewed me last was Professor Kama. A thorough gentleman he was. He made the observation that I had responded to the questions that every staff member had asked, and he was going to therefore find the limits of my knowledge. And the first question, of course, was, what is euthanasia? My own thought process was, I hope this is not about killing youth, I was one of them at that point of time. Um, uh, but I did say that it must be about something that put something to rest. And he said, yes, it is mercifully putting people to rest. And then he asked the question, what is nanobiology? I said, I have no clue. He said, it is germ-free biology. And of course, little did I know that that germ-free biology would guide us on understanding the importance of germs in a good way as well as a bad way. But today my talk is going to be about keeping the good guys in a good state and making sure that we kill the bad guys to the extent that they affect our own health and our own wellness. We run a venture capital fund, but we are investors in very early stage companies. We are in the business of translating science into technology and technology into products and services. I will not talk about every company that is on this list today. I will focus on two companies, Greenlight Biosciences from an RNA perspective, Amphora from a DNA editing perspective. Needless to say that Amphora's DNA editing technology won the Nobel Prize just this year in chemistry for DNA editing technology. Greenlight Biosciences leverages the principles for which Nobel Prizes have been awarded, and probably for one for which Nobel Prize will be provided or given over the course of the next year or two. So without much ado, let me focus on the topic of biologics at scale powered by the RNA platform of Greenlight Biosciences. One of the foundational developments that occurred in Greenlight Biosciences during the last 4 years was to develop cell-free systems for the production of different types of RNA and to make them very cheaply and to make them with high quality and to make them quickly. Faster, better, cheaper is the lingo of the business world. And indeed, that is essentially what Greenlight Biosciences did. But in addition, they did one other thing. They actually did develop a process for making everything in one pot so that the supply chain, which is so critical for businesses, can be conducted wherever you are, whether you are in the United States or whether you're in India, whether you're in Cameroon or whether you are in Australia, you can do that. So fundamentally, several technologies were developed, technologies for regenerating ATP and other nucleotide triphosphates, technology for taking nucleosides and then converting them to nucleotide triphosphates so that you can start with very cheap sources of nucleosides, and also technologies for capping RNA, technology for adding poly-A tail to RNA, if it is messenger RNA, technology to make double-stranded RNA, if that was the goal of the program. All of these processes were developed over the course of the last 4 years, and we are now applying it to make various products that I will describe to you in the next few minutes. Greenlight's process is so cheap today that we are able to make grams of material at less than about $50 to $60 per gram of mRNA. So if you are injecting a vaccine at 50 micrograms or 5 micrograms or 100 micrograms, that obviously has significant implications for the cost of the vaccine. And that is essentially where Greenlight is focused. Similarly, if you're going to use double-stranded RNA as a pest control agent, you want to make sure that it can compete with products that can be made with chemistry or with biology. And there again, the cost is good enough to compete. And that is what I will show you today during the course of this talk. One of the important facets of biotechnology, especially in industrial biotechnology, is your ability to scale. Many companies have failed because they are unable to translate what works in a shake flask to a fermenter, because the entire process changes when you go from a shake flask to a fermenter. in Greenlight's hands, the technology is readily scalable because everything is carried out in what is called a homogeneous state. In fermentation, unfortunately, that is not the case, but with cell-free bioreactors, you can carry out reactions in a homogeneous state, and that allows for scalability. Greenlight has now focused on making vaccines essentially for COVID and influenza, both very important pests as far as humans are concerned. And I don't have to tell you what COVID has done during the course of 2019 and 2020, from starting from China to other parts of the world. The loss has been estimated to be around $12 trillion in terms of global economy. And many of us have experienced influenza, the seasonal influenza, and this too remains an important disease of consequence. Just a couple of days ago, there was a report that in Russia, bird flu has come from birds, chicken, to humans, and this is a disease that can infect not only animals that we cultivate, but also, of course, humans. That back and forth between these animals and humans is something that we have to pay attention to. And this is where the messenger RNA technology is so pivotal, because you need vaccines which can be developed very quickly and made available for people to use in order to manage and, of course, prevent the disease itself. This is essentially a summary of the very first mRNA vaccine that was just approved by the US FDA for emergency use a couple of months ago. And work on this vaccine began in February of 2020. You can see how quickly the vaccine was developed. And that particular vaccine is an mRNA molecule shown here with a cap at its 5' end, which Dr. Venkat described extremely well yesterday. It has a 5' untranslated region. It has a signal peptide region, which allows for the protein, the spike protein, that is used as the antigen to be transferred from the cytosol to the cell surface so that it is available for immune recognition. The spike protein itself and the 3' untranslated region and the poly A tail, which we heard about yesterday as being important for mRNA stability. Number of different changes were made in making this messenger RNA vaccine. One of the key components is the replacement of uracil with pseudouracil, which you can see here. Pseudouracil obviously is a different molecule than uridine. Chemical structure is shown. Every uracil in the mRNA for spike protein was replaced with pseudouracil. And in addition to that, a prolyl-prolyl bond had to be introduced in order to stabilize the conformation of the spike protein on the cell surface, so that the spike protein actually looks like the viral protein in its conformation, as opposed to collapsing and having a different structure altogether. So this has been a remarkable journey in a matter of few months, and much of this work was done between 2005 and 2020 in terms of gaining the insights. The notion that uracil itself is capable of triggering the immune system and also triggering nucleases which destroy the RNA, that was understood first. That led to the replacement of uracil with pseudouracil. Of course, prolyl-prolyl substitution has been used historically in order to stabilize molecules. And of course, the poly A tail by itself cannot be used in making plasmids, because the DNA becomes unstable if you have too much of repeat elements, especially if you make the DNA before you can make the messenger RNA, and you have to clone that into a plasmid for propagation. So all of that information which had accumulated for the last 15 years promulgated in the development of this mRNA vaccine so quickly. Our company, Greenlight Biosciences, has also been experimenting with various COVID vaccines. They have made both the natural mRNA vaccine, where the uracil is kept intact, as well as the pseudo-uracil containing mRNA. They have their own untranslated region. They have their own modifications in the spike protein, and of course in the 3' poly A tail as well. And in addition to those, they're also in the process of generating mRNA for many of the variants that have been found, especially the 501 strain, which is supposed to be more virulent. And now they're beginning to look at the 357 strain, which also has been found to be more virulent. And in addition to looking at these mRNA vaccines, in the discovery phase, we are looking at not only the mRNA that can be imported by the cells upon injection into the human to make the protein, but we're also looking at self-amplified mRNA, where we can amplify the mRNA in the cell itself, and this amplified mRNA can produce more protein, so you can actually use less of the mRNA in producing the antigen in the human body. One of the important things that I must also point out is that it is not sufficient to simply make the mRNA and administer it to people. It has to be in what is called a lipid nanoparticle formulation. And these lipid nanoparticle formulations typically contain some phospholipids, cholesterol, and some cholesterol derivatives, PEG derivatives as well, PEG 2000, salts like potassium chloride, sodium chloride, and sucrose, all of which help protect the mRNA. And these mRNAs can be kept for at least about 20 days at about minus 20 degrees Celsius without much difficulty. This is some data. I don't want to spend much time on it other than to tell you that these mRNA vaccines, when they are injected into mice as well as hamsters, are capable of producing outstanding neutralizing antibody titers, and at levels as low as 5 micrograms with the 2 doses, the first dose and then a second dose on day 21, we can pretty much mimic what Pfizer is able to get at 30 micrograms, which tells us that this vaccine is more potent than what has already been commercialized by Pfizer. This is a summary of the state of the art as we are able to determine that various mRNA vaccines that are available, we are able to get a much higher neutralizing antibody titer. In terms of the T cell response, there is obviously a Th1 bias for most of these mRNA vaccines, which is great. And we are also seeing, of course, a very positive CD8 T cell response as well for many of these, and all of them have protective activity in the hamster model against the COVID virus. We are now working on the influenza vaccine, and there the goal is to make the quadrivalent influenza vaccine. It is unfortunate that India today still does not have the vaccine, and I must mention to you that I'm working with a company in India. I'm on the board of a company that was spun out of the Indian Institute of Science called MinVax. And Professor Raghavan, who is in the biophysics department, is also, of course, making more thermostable vaccines for the Indian use. But the combination of the mRNA vaccines as well as the protein vaccines should allow us to actually manage influenza in future without having to depend upon the egg yolk, which was used as the production factory for these particular classes of antigens. This molecule, again, these mRNA molecules are capable of eliciting in the animals similar responses as the COVID. And when challenged with the influenza virus, they're very protective. So we are extremely delighted with these findings, and we're in the process of advancing those into clinical trials. I talked about the human application of mRNA. I'm now going to switch gears and talk to you about a pest control application for another class of RNA called the double-stranded RNA. These double-stranded RNA molecules are between 400 to 425 bases. They recognize one of the important proteins typically expressed in the gut epithelial cells, and they inhibit the production of those proteins by killing the mRNA that is responsible for making those proteins in the epithelial tissues. In this particular in this particular instance, we are looking at an insect called the Colorado potato beetle, which is a major pest that is found in North America and Central Asia. It devastates, of course, the production of potato, and farmers have to spray pretty much every week in order to protect their potato crop. If you do not, you can see in the untreated control what happens to that potato plant. So the double-stranded RNA, when it is applied to these plants, it is sprayed as a typical agrochemical is sprayed. We get complete protection of the potato plants against this particular pest. This product now is being advanced through the EPA for commercialization. But in addition to this, we are also making double-stranded RNA to protect bees against mite infestation, and that product is under works. So we have a number of products in agriculture for plant health management, in human health, in the form of not only vaccines, but also, of course, gene therapy that too can be delivered via mRNA. These are the people who are associated with the company. Andre Zarur, who is the CEO of the company, is instrumental in putting together the company and the technologies that I described. In the final section of what I'm going to show you today, I'm going to switch gears and talk to you about plants, a company that we founded about 3 years ago with the express purpose of addressing an important nutritional need of many of the people on this planet. And today in the Western world, we consume a lot of animal proteins. And I'll tell you in a little bit the issue with animal protein production. But suffice it to say that green revolution, which kept people like me when I was a kid fed, and many of us on the phone, because in India, the Green Revolution, which was spearheaded by Swaminathan and of course by Borlaug in Mexico with wheat, they were the principal reasons why we survived and we were able to produce enough food. Those semi-dwarf varieties were very responsive to fertilizers, and those fertilizers caused increased yields of those crops. But when we increased those yields, we did not pay attention to what was happening in the protein in the grain. And one of the unfortunate outcomes of that process was a reduction in the protein. So we were actually getting enough calories, but not enough protein in our food. And the chart here shows the response of crops that we have today, mostly cereals, to the applied nitrogen as a fertilizer. You can see that it declines before starting going up. And one has to apply significant amounts of nitrogen to a point where it is polluting to the environment. And that is not a desirable outcome either. A paper was published in Lancet a couple of years ago saying that we should move away from animal proteins into consuming more plant proteins. In the United States today, you will find companies like Impossible Meat, Memphis Meats. You will find companies like Beyond Meat. All of these companies are focused on bringing plant proteins into meat-like foods so that we can reduce our dependence on animals, not only because they can transmit diseases, but we also have pandemics in animals. Last year, China had a major problem with pig production because 50% of the Chinese pigs developed African swine fever. And on a regular basis, lots of chickens are culled from production systems because of the disease. And of course, they're also not healthy because cardiovascular health has been typically connected to the consumption of red meat, especially barbecued red meat. So because of these reasons, we are extremely interested in increasing the protein content and making sure that this linkage between yield and protein, which is negative, is addressed. At the time we were thinking about this problem, there was a researcher in Iowa State University called Ling Li, who had identified mutants of starch, and these mutants of starch were capable of producing more protein. They were capable of producing more starch and less protein in their leaf. And it turned out that they were defective in a particular gene called the QQS gene. And QQS actually stands for quaquin. Quaquin does not mean anything other than it is Ling Li's daughter's name. And she named this particular gene, which encodes for about 59 amino acids, the QQS gene. When the QQS gene is overexpressed in Arabidopsis, it reduces the starch content of the leaf and increases the protein content of the same leaf, as you can see by iodine stains in the Petri dish that is shown here. Ling Li went on to introduce the QQS gene into crops like rice, soybean, and corn. Every instance, she was able to demonstrate that the protein content can be increased, whether it is the leaf or the seed. In all cases, there was an increase with no reduction in yield, no reduction in maturity. So we felt very positive about taking on this technology and introducing that into commerce. However, we also recognize, because I've spent about 30, 40 years of my life working on GM crops, that if we can deliver this technology without genetic modification, that would be an outstanding accomplishment. So we wanted to understand how the QQS gene worked, and Ling Li demonstrated that there is a transcription factor called NFYC4, which is recognized by QQS, and this NFYC4 gene is hyperexpressed when the QQS gene is present. And NFYC4 is part of a heterotrimeric transcription factor family in plants, and they are responsible for activation of the catch domain that is present at the transcriptional start sites of many of these plant genes. And indeed, this NFYC4 is able to bring about a series of transcriptional a series of expression of genes which are capable of changing the ratio of protein to starch. And indeed, we were interested in figuring out if NFYC4 overexpression could cause exactly the same phenotype as the QQS, and indeed it did. And one of the things that was also recognized in that process was that NFYC4 was auto-regulated. And indeed, there were a number of sites that we were able to identify which were serving as negative regulators of expression of the NFYC4 gene, and they were known as the RAF1 and the ERF motifs. We went on to use the CRISPR-Cas9 to delete those recognition sites, and by simply deleting those, we were able to increase the transcript of the NFYC4, produce more protein, and reduce the starch. I do not have to tell you that rice, which has lesser amount of starch, higher amount of protein, every Indian who wants to eat rice, if we are able to do that on a daily basis, my calculation tells me that is the equivalent of eating one whole egg, which is a significant amount of protein. And indeed, we are now in the process of editing these sites in soybean, in rice, in a number of other crops so that we can bring this to commerce. Indeed, technology has been demonstrated to work across by editing across soybean and rice already. We're in the process of evaluating these products, of course, for not only plant meat production, aqua feed, but also for human consumption. Shown here is a picture of Ling Li, who did most of the work. Mike Lasner, who is the CTO of Amphora, the company we founded, Lloyd Kunimoto, the CEO of the company. So let me terminate my presentation here by acknowledging my colleagues, Dr. Raja Weiss, who is also a plant biologist by training, John Hartnett, who is our partner, who comes from the technology industry. He was the COO of Palm. Greg Young, my partner. It has been a journey that has been wonderful. We focus on learning about the science in a regular manner. People from Indian Institute of Science, other scientists who have spoken, but we also focus on the practical side in terms of the problems that these types of discoveries can deliver and make products that can benefit humanity. Thank you very much, and I appreciate the opportunity that that all the people who are here have given me today, and I hope there is time for one or two quick questions. Subbarao, you're muted.
Oh, thank you, Dr. Kishore. It was a wonderful talk. It was a very nice presentation of the RNA vaccines and the DNA editing strategies. And we can take some questions, and there are a few questions in the chat box also. Can you ask the question, Salim? Yes, Salim Ahmed Khan has a question.
Yes, sure. Thanks, Kishore, for a great presentation. My question is very simple, and that is, I can understand why a variant from South Africa or UK is sort of has increased infectivity because of change in the spike protein. But sort of what's the molecular basis for increased pathogenicity? Is it just increased replication and viral load, or is it that a spike protein has some other role to play in the disease process?
Great question, Salim, and great to hear you as well. The key comment here is that to the extent that we know, it seems to interact, the spike protein seems to interact with its receptor more tightly, at least for some of the variants, and is also able to deliver the virus into the cell a lot faster than is typically encountered in the wild type strain. So these RNA viruses, I did not spend much time talking about the edit function they have. Many of these RNA viruses that are larger, they tend to repair their RNA during replication because fidelity of replication is quite critical. But even then, even the coronavirus makes some mistakes, and those mistakes sometimes can be detrimental. And sometimes, of course, it could be more virulent than the original strain. And that's exactly what happens. The molecular basis for all of this is still, of course, very preliminary based on our in vitro understanding or ex vivo understanding. But none of them, of course, may hold true under in vivo conditions until more experiments are done.
Thank you.
And there was a question from Beena. Is Beena there? No. Is any other question?
I just have one.
Yeah, Money.
Sorry, a wonderful talk. Really, eye opener with respect to the technology. My question is, as you said, now we have in the COVID space, we have a number of vaccines coming up, this mRNA vaccine, but most of the, we are talking about Pfizer vaccine working for some, but not for other variant. Have you guys thought about making a cocktail of mRNA rather than just having a one RNA? So that way, this cocktail should be able to cover not only the original virus, but also the mutant, potential, the mutants. And then that would kind of one vaccine.
Yeah, I got the gist of your question, if I may. So in the interest of time, I kind of glossed over the point. In fact, we are doing two types of studies. One is a polyvalent vaccine, where we mix the mRNA in LNPs, or different LNPs carrying different mRNAs. Both are being done. Give us a few more weeks, and I will tell you whether in hamsters it works, and we can advance that. So the goal is precisely what you described, because coronaviruses are not going to go away. And for those who have studied coronaviruses for the last 20 years, know COVID-1 was here in 2002 to 2004. MERS, there is no vaccine still today, and it is still in the Middle Eastern countries. of course, in other regions of the world as well. And the COVID-2, and the COVID-2 will evolve. And it is routinely encountered in animals. Animals have to be routinely vaccinated. They have been using the adenovirus vaccine for some of those. So the short answer to your question is polyvalent vaccines will be needed. We are advancing that for flu and also for the coronavirus.
Super. Thank you.
Subbarao, one
Suraj.
little
Yeah,
question.
go ahead, Suraj.
Kishore, terrific talk. Thank you. That was tremendous. Oh, I noticed in a fleeting fashion on one of your slides with respect to the mRNA, there was a comparison between Moderna and Greenlight and Pfizer. Pfizer was at 200. I don't know what was the unit for 5 micrograms, and Greenlight was 137,000, which I think translates to the stability of the messenger RNA possibly. Would you comment on what was so special in the messenger RNA in the Greenlight preparation that contributes that stability?
Suraj, I must first of all applaud you for the detail.
I can't hear you.
You can't hear me? Can you hear me?
Yes, we can hear you.
Okay.
Yeah, we can hear you. Suraj, what happened?
Okay. So first of all, I must applaud you, Suraj, for the attention to detail that you paid. Certainly, we find that by making alterations with respect to the RNA stability and translatability, this is where the specific choices of sequences matter. I wish I could tell you exactly what change we made. Unfortunately, my lips are sealed.
Okay.
In that particular direction, being an industrial scientist that I am. But what it goes to show is that it is possible to fix those types of situations so that we can bring the dosage levels of these RNAs to very low levels. And this is why I'm so excited. The other part that I did not mention is just linking it to the dengue dialogue that we had. And there was a very elegant theory. There is no reason why we can't create polyvalent dengue vaccine and test that hypothesis. So all of this is being enabled by the progress that we are making in the discipline of RNA. So it took us 20 years to really develop solutions using RNA. But my belief is because of the way RNA molecules can be made, the simplicity with which it can be made, this is going to revolutionize the field.
And we can start making vaccines.
I think this technology should go to solve the dengue phenomena.
Every year we'll be taking a new.
Yeah, I think Muthukrishnan is trying to ask some question.
They can predict.
Okay, go ahead, Muthukrishnan.
Hi, Kishore. You mentioned in passing that you're amplifying RNA inside the cell. Does that involve RNA-dependent RNA polymerase, or is this generally used?
There are two types of techniques that are being explored today. One is using certain viral recognition sequence, which the host RNA-dependent RNA polymerase can do. And the second is to also deliver the mRNA for the RNA-dependent RNA polymerase. I do not know which of those will end up being commercial, but obviously for human applications, the preference is to not introduce more than we need in terms of RNA-dependent RNA polymerases.
Yeah, that's very clever. Thank you.
Yeah. If there are no more questions, let me ask you a question, Kishore. Yeah. I have a doubt about this RNA vaccine. The RNA vaccine, when you translate into the protein, it covers only a small portion of the spike protein in S1 protein. It doesn't even cover RBD. It doesn't make RBD also.
That's not true.
No, that's not correct.
Vaccine is a full-length spike protein.
The full-length
making full-length, but the sequence you showed in this slide, it doesn't have a full-length sequence. Right?
I did not show the full-length sequence because I just wanted to make a point that it is pseudo-uracil at the beginning. It's on, it makes the full length spike protein.
Makes full length.
Yeah. And we
So
can
then-
also add, but your
Subbarao.
question is right. You can add.
This GP here, I just want to make a point to Ganesh.
Yes, Dr. Padmanabhan, yes.
Yes. You know,
Yeah.
India, I don't know whether you have heard about Genova at Pune. It's making a self-amplifying mRNA vaccine.
Yeah.
It's already there. It's gone into phase 1 trial. Just want to give you the information.
Good.
Thank you.
I'm
That's
delighted to hear that because I know there is the adenovirus vector-mediated vaccine
No,
that
no,
people.
this is a self-amplifying mRNA vaccine.
Yeah. And the principle is very similar to what I described, right?
Yeah. It's making a particle which contains RNA-dependent RNA polymerase, and therefore it amplifies.
So,
The
Dr.
primary-
Padmanabhan, is that for COVID?
It's for COVID.
Is that for COVID, Dr.?
Yeah, it's for COVID. Yeah. It's gotten
Kishore.
Yeah.
This is Jagadishwaran. I was curious about your the protein-rich rice. When you make it, you mentioned that you will get an egg level of a protein, amount of egg. How much of a rice you have to consume in order to really get that, and will it really be toxic after a long time usage?
Okay. First of all, of course, when our grandfathers grew rice, did not have to worry about adding fertilizer and things of that nature. They were actually growing rice with 12 to 15% protein in it. So it was more nutritious rice. So with modern technologies, we were able to increase the yield of rice, but we reduced the protein content. So in that regard, we are not doing anything to change the composition of proteins. We actually increase all proteins in the seed. The second point, of course, is people consume varying amounts, but I'm talking about people who consume about at least in 2 sittings rice as the main center plate of their meal, which is a typical South Indian meal for lunch and for dinner. So if that's the amount that is consumed, then you get the equivalent of 1 egg. If you only have 1 meal with rice, then you would not.
Okay, you get half egg.
Yeah.
Can I ask a question? George Thomas here.
Oh, yes, George.
Yeah. So, Kishore, rice is deficient in certain amino acids. So, if you increase the protein level, will the deficiency of amino acids create a problem?
The deficiency of amino acids are not a problem. First of all, Indians do not get the 23 to 24% protein that is needed on a daily basis. This is one of the reasons why in children, you actually see this dual malady of calorie and protein malnutrition. So that is not an issue. With respect to lysine and methionine, the 2 amino acids that are limiting, and sometimes even threonine and tryptophan, those are not addressed. We have simply addressed the quantity of the protein today, but we can actually replace in the storage proteins, which will be the next phase of the program, where the editing technologies will allow us to introduce small amounts of these amino acids in storage proteins so that you can actually make it balanced protein.
No, but then the amino acid has to be available also in the rice to be incorporated into the codes that you build in.
Yeah, they are. There are actually, it is just like the prolyl modification that I mentioned. There are specific regions which we know about the storage proteins where we can do those. One thing we want to be very careful about is not to take a hypoallergenic product, which rice is, and make it an allergenic product. That is the primary concern.
Yes, lysine introduction can cause that, yes.
Thank you, Ganesh. Great talk.
Thank you.
Are there any more questions? So that we are coming to the conclusion of this session. Yeah.
The concept that you have essentially established using Arabidopsis, how would it be translatable to all other plants?
You know, the beauty is this is why we took it to three other crops: rice, corn, and soybean. One is a legume, one is a C3 cereal, another is a C4 cereal. For those of you who don't study plant biochemistry, C3 plants are the ones that fix carbon primarily through the 3-phosphoglyceric acid route, whereas C4 plants have a secondary pathway where they take those carbon dioxide molecules and put them into PEP to make malate and oxaloacetate, and then recover that carbon dioxide and then put them back into a C3 molecule in the form of phosphoglycerate. But the beauty is it works in every plant, it works in every plant cell, which suggests to us that this is foundational. This NF5C4 regulation is so foundational that it is operating at the interface of carbon flux between protein and carbohydrates.
Good, thank you.
Whether it works in yeast, that is something that we will look into because there are similar, this one, mammalian tissues. I don't have to tell you, if we increase the mammalian cell production of proteins by 20%, it certainly changes downstream steps in terms of efficiency, productivity goes up. So there are also opportunities there, but we are far away from getting to those points.
Okay, thank you, Kishore. And it was a very nice talk. And I would like to thank all the 4 speakers who have done a great job today. And before we conclude this session, I would like to share some of my thoughts. So in 2007, the IAC Alumni Association of North America organized the first IAC Global Conference in San Francisco Bay Area, in which 30 professors, including Professor Balram, Professor Balkidal, attended. The theme, primary theme was how to increase the collaboration between university and industry, that is IAC and industry. But apart from that theme, we had discussed some other visions for the next 100 years of the institute. And I will just list 2 of the visions which Dr. Balram was mentioning. The first one was to start the undergraduate program. And the second one was to start a medical school. As you all know, the undergraduate program was already started, and we hope that the institute will have a medical school also in the future. The biochemistry department will have a great opportunity to do more clinical research if there is a medical school associated with the institute. The symposium was also very thoughtfully organized, with the last session chaired by me. I'm an entrepreneur, and the end with a speaker who spent most of his career in 2 large corporations, Monsanto and DuPont, and who founded an investment company. This shows that all the basic research, when it ends up in commercial applications, the research becomes very meaningful. And that's all I had to say. And even today's talk from Dr. Blundell, you see that he also became an entrepreneur. And so from basic research, how he started the company. So with this, I will conclude, and I will turn over to the organizers for the final conclusion. Thank you all.
A heartfelt thank you to all the speakers of the session for taking time out of their busy schedule and enlightening us with their talks. I would also like to thank Dr. G. Subbarao for chairing this session. Now, I welcome Professor D.N. Rao for the concluding remarks. Over to you, sir.
For your participation yesterday and today's, all the speakers and chairs for this wonderful 2-day session that we planned, and it was very successful according to many of us. Now, this is one of the things that we planned, that we will repeat these kind of 2-day conferences or symposia or meetings, another 1 or 2 more before December, where we hope that in December we will have a physical meeting so that a reunion, a genuine reunion is possible in Bangalore. So I want to thank once again, all the speakers of yesterday, today, the chairs, for having participated, and we heard some wonderful science, both basic and applied, as Dr. Kishore was mentioning. It doesn't matter which one, but I think many of our students, or most of our students here, have benefited hearing such good talks. So thank you once again. Let's keep in touch. We will be writing to all of you again, and we would like your support and keep this interaction all the time alive for the next couple of years, if not for this year. Thank you very much. Good night, good morning, good evening, and we hope to see you again.
And thanks, Professor Rao, for organizing.
Thank you so much.
Biochemical
Thanks a lot.
Society students and volunteers who actually did the whole thing of coordinating on this internet and this whole Zoom call and YouTube connections that we have had. So thank the Biochemical Society for this. Thank you and goodbye.
Thanks a lot.